Good afternoon, everyone. Welcome to ROV SuBastian Divestream 912. We're currently at about 340 meters down, heading down, right at the very beginning of the dive. Once again, we're going to be having a dive full of imaging, scanning, and sampling of midwater organisms. So enjoy the dive, and I'll hand it over to our PI for an introduction. That's Karen Osborne.
I'm in 3 hours another time in the last 24 hours, and I'm like, I'm good to go.
Okay,
But,
Karen.
Okay, let's try.
all right. Good afternoon. We are aboard the Falkor 2, a research vessel operated by Schmidt Ocean Institute, and we are so excited to be here. We've been doing midwater exploration. We have a couple of objectives on this expedition. We want to image all of the animals that are down here. We brought a suite of tools to do that in new ways, because just looking at them with cameras gives us a lot of information, but there's other information that we just can't get from just a 2D picture. So we brought a suite of instruments with us to look at these animals in new and different ways. We are collecting animals so that we can describe them. We are working on the genetics of the animals that we see here, and just trying to learn as much as we can. Most of the animals that are out here are fairly unknown. When I give a talk about the work that I do, I always get these awesome questions like, what does that eat? Or how long does that live? Or what is the name of that? And oftentimes the answer is, I don't know. We haven't figured that out yet. I find that situation super exciting because every time we come out here and we look around, we learn new things. And
Did you want me to take over? I think I'm up first, or is this good?
I think you are up first. I think you are up first. If you want to take over, you can. If you're still working on animals.
Well, I just kept
Feel
getting
free
distracted
to-
by people asking me questions.
You're a resident expert of everything, Russ. And now we're down to all the weird little stuff that people.
Yeah, well,
I'm happy to do this for a bit if you want to.
just let me get my ears back in. I've just realized I haven't put them in yet for the day.
Okay.
They're of course anxious to take stuff to control and run for isotopes, but many of the things
Maybe the stuff we haven't
ID'd
done
or
yet.
run yet.
Yes, exactly.
So,
we need to grab some of that stuff.
which is
Before
the
they
priority? This
the-
or me figuring
You figuring out those things.
that out?
Yeah, for sure. Sorry. One of the things we're doing on this cruise is trying to build reference libraries for as broad a swath of the diversity that's out there as we can. And to be able to do that, ooh, look at that. Little Bisonect just went by. There's a low bait. We are at 430 meters. We're pretty much going to race down to 900 meters today. And then we're going to start hunting from there. So we want to build reference libraries for the animals that are out here, and that means we need to identify them. That is not an easy task. Most of these animals have not been looked at, especially from this region. We're just off the coast of Brazil, about 360 miles off, just about at the bump. Maybe somebody in the chat can tell me what they refer to that area of Brazil, like where it changes direction, so that I can have a proper term for that. So we want to build the reference libraries for the animals. That means we need to collect really broadly. We need to identify exactly what they are. As we do that, we are finding tons of new species. This area hasn't had a lot of exploration. We don't know a lot of what's out here. And so things look very similar to things other places, but they are not necessarily the same. A few are, most of them aren't. So far we're about a week into this, and we have found more species than I can keep track of that are confirmed new species. And so we really want to document those species really well. We want to get genetics, and an important part of that is identifying them. So we assembled a team of experts. We have Russ Hopcroft. He is a professor at the University of Alaska, Fairbanks, and he is into larvaceans and thaliaceans, salps and doliolids and larvaceans is his main interest. There's little hatchetfish. Got a couple of those in the tow this morning. Those are really fun to look at. They have incredible bioluminescence. They have incredible photophores. So Russ also is an incredible general midwater taxonomist. Can you turn the lights up just a little more?
Lights.
It's just really dark in our view.
Let me try this.
Or maybe iris, I don't know, whatever. I mean, it makes it so that we're not going to stop because I won't see anything. That was strategic, yeah. That's pretty good. I mean, it doesn't really matter because we're swirling, so, you know, but still, can at least get an idea of what's there. So Russ is doing chaetognathes and shrimp and a bunch of different groups that we don't have an expert specifically here for. We have Dave Fields. He is a researcher at Bigelow Laboratories in Maine, and he works on copepods. So he is diligently working his way through the hundreds and thousands probably of copepods that we are getting in our collections. We have Marcelo Mello, one of our Brazilian collaborators. He works on fish. He's been getting through all of the larval fish and all of the adult fish that we've been collecting. There's a scythe tail off to the right. Let's see, we have an awesome team. Heather Judkins is here from University of South Florida, St. Petersburg, and she is a mollusk expert. So every time we see a squid, she's our go-to person. She also does pteropods and heteropods, really interesting animals that we get out here, and generally she is keeping things running smoothly, scheduling everybody so that we get some sleep and we make sure there's somebody to be here and do the work all the time. Who else is here? Professor Jan Hemme from University of Western Australia is a neuroscientist. He is here to study the behavior and the vision of some of the animals that we're seeing. And then we have several engineers aboard. We have Kakani Khatija from Monterey Bay Aquarium Research Institute, and her bioinspiration lab is here. The instruments that you'll see today during the dive were their ideas and their equipment that they have built, and we'll talk about those when we start using them a little bit. They're just new ways of looking at the animals here, giving 3D information about those animals. Yeah, so it's a big team of a lot of different people. We have 25 people here from 13 different institutions. You know, this just hurts just a little bit, right? Okay. Right now we are bombing down to 900 meters so that we make sure oh, look at that. There was a big jelly in the background. So we are bombing down to about 900 meters because we want to look for animals in lots of different parts of the water column. It would be super easy to be distracted, and as soon as we're able to start working at about 200 meters to start looking at things, but there's so many things to look at, we would never get that far. And so we're setting ourselves up to see lots of different parts of the water column. Oh, what was that funny little squiggly thing? Yeah, it was like this very white, convoluted, like a worm, but super reflective. And I just no, something else, something squishier.
Do you intend that 900 will be the deepest, then it'll start coming up there, or you want to go down a bit deeper?
We may oscillate around. It depends on how. We're not going to go deeper than 1,000.
Okay.
You need to set something.
I'm supposed to just do a bottom depth to get a few milestones set up on final milestone on the down part.
So if we say 900 and we chase something a little deeper.
Well, that happened yesterday. I mean, it's okay as long as you don't go significantly deeper.
No, we won't go significantly deeper.
Okay.
Yeah.
Are we doing this in the bottom depth?
900 is the target.
I think
This in the 900 as well?
Oh, yes, yeah. One at 900, and then we'll do one at 450. And then 2 again. I think they want to maintain that pattern for the rest of the dives.
So we're going down first and then coming back up, or just?
No, we're going down, and then we'll search like slowly up. We don't make it that far when we start at the top. It's just too many good things. Starting to see the forams come up, Pastogen rhina. There's calicopherin. We are at 600. What's that? I've looked at this kind of video for a long time. When I was a grad student, it was my job to annotate transects, and transects are just like driving as fast as possible. There's a oik. Actually, I might have been a Siphonophore. It looked like I had 2 little stripy tails. Something over there to the right. Can't quite tell what it is. Wait a minute. What? Can we try to go back to the...
Faster.
Do you think that's what it was?
So I saw the thing in the little.
It definitely
Squiggly
had...
bits.
Yeah, okay. They just seems like... No, yeah, that's what I can go faster.
How do you want? Speed light.
No, actually.
Fast enough that we can't tell what it is.
Fast enough that I can't see anything. No. We've made such good progress. We're already at 600. Things are getting... So maybe we make a note that... I'm going to make a note that things looked pretty interesting here around 600. That's where we spent a lot of time.
Oh,
Or-
it's not. That's just a dismembered.
We need to get some of those libations today.
The ones out in the house.
Yeah. You know, the really hard, tiny ones.
Yeah.
Oh, another sparkly foreign.
100, 450, 50, 50.
I'm marking them down after I see them, but I think that means I'm frame grabbing them when they're out of the screen.
Yeah, that's okay. I mean, we generally know that you just need the time code somewhere near it, and we know that that's how that works, and so we always look a little ahead,
Ooh,
little
that was little
ahead
Siphonophore,
there.
right?
Is it a Ketognath or Cyclodony? Either way. Oh, there's a Sarah Volmer. We collected one of those yesterday.
What?
Sarah Volmer, S-E-R-R.
Oh, was it a eel? Oh, I think I know. We have those. Yeah.
I didn't see it, sorry. But
Terrible.
Coming up on 680 here. Oh,
Not trying anything.
that was libation free swimming. Given its size, probably really close to the camera. Jelly, probably a tricky.
No, you show me.
Medusa.
I don't know how to do that. I know how to stand in front of a monitor and yell out what out. That's my forte.
jellyfish taxonomist who's drowning in technology.
Stop the shadow cat. Otherwise,
Okay. I don't know what to do next.
yeah.
I tried to put in the like the next as thing tonight, returning lines.
Yeah.
Starting to die and stuff like that. Just
Yeah.
something that
That's a good video as well, just to see like how shaky
Oh, okay,
it is.
that's it.
So we can, yeah. Looks fine, honestly.
Definitely just.
it was working fine previously.
Yeah.
You know, when I think should be in the cluster,
Right area?
because our pump is a fixed placement pump, it's not like a space. You know, very like it's big. But you can do this in a proportional valve for the cluster. Regular cluster.
So as I sit here and try to avert my eyes from the feed so that I don't yell stop every 5 seconds when I see an animal go by, as we're trying to get down to depth to make sure we search through as much of the water column as we can.
Every now and again.
I'm looking at the profile, the hydrographic profile of the water, and at the surface it's about over 29 degrees, and then it quickly drops. Let me just make sure I'm following the right curve there. It quickly drops. By the time we got to about 400 meters, the temperature was down to about 6 degrees, and it continues to fall from there. I think that's a 5. Yeah, so we're just below 5 degrees now. Oh, 4.8. Yeah, you can see it better when it gives the actual readouts.
Manuel Diaz.
So
It sounds like
the animals that are migrating through this are experiencing huge temperature change. If they come from anywhere below 300 meters and get close to the surface, because the water is really warm up there in the top 200 meters. I think it's actually could, maybe not.
Not dying as much as it looked open. More than a better dragon. Not a gunner.
There's a fair amount of oxygen all the way through this water column. It gets down at the lowest to about 50% saturation, which is for most of these animals really pretty well oxygenated. So we don't see much structure within the water column associated with low oxygen. Oh, nice.
okay, so O doesn't mean stop. Right?
No, it just means, oh, we really wish we weren't going so fast. Oh, did we really say we were, did we really say we were going to go to 900 before we started looking? Are you sure that wasn't yesterday or something? Ah, darn.
There's another giant squid. Is that it?
Giant squid we can stop for. We're now at the depth. We could see a Vampyroteuthis potentially. I mean, if we saw a pig butt, that would be great. It'd be really nice to see a pig
We're gonna
butt.
stop just to wait for the winch.
Okay,
A little bit.
you're beating the wind. Super fast.
That it's an octave fish, maybe?
Probably. It's hanging the right
So
direction.
then for the
900. Yep. 900, 450, 50, 50 surface surface.
Oh,
Yeah.
is that the fish like
Good.
face?
Super. Yep. We got several of those in the tow this
Is
morning.
that a type of hatchet fish?
Hatchet fish, yes.
Perfect.
Some of the fish down here you can identify by the direction that they are swimming. So Loroglossus we see hanging in the water column or swimming kind of diagonally usually. Cyclothone is always aligned with the horizon basically, so they're horizontally arranged in the water column. Many of the fishes orient themselves vertically in the water column. Hatchet fishes tend to be oriented and a little halocreid. No, there was one Halosolmissus down below and a Halocreidae above. So hatchet fishes tend to be oriented horizontally, and you see the flashy silver sides of them from the ROV. All of our lights reflect beautifully off of those mirrored sides of theirs. The ones that we got in the tow, we were looking at their photophores. They have these incredible photophores that are very specifically oriented downwards, right? Because what hatchet fishes are doing is trying to erase their silhouette. Many predators in the midwater are hanging out looking up into the light that is coming down from solar illumination, whether it's the sun or the moon or the stars. That is the only major light source in the midwater, is that downwelling light. And so looking up into that downwelling light and looking for shadows is a great way to hunt. There is also bioluminescent light that is produced all throughout the water column, and up shallow bioluminescence is often used to counter illuminate. In other words, try to create enough light to match the downwelling light so that your shadow disappears. And that is what the hatchetfish are doing with its downward directed. Oh, what was that? You hear the angst in my voice. Oh, what is that? We're almost there. So close. 8:41.
I want turn the lights off.
No. Oh, there's something wrong with the iris. I don't know what happened. Just, oh, big mucus house. So many fun things to look at. We could just do this 24 hours a day for, I don't know, as long as you want.
What?
It would take a much larger team than we have to do this. Oh, there's something off to the right. Huh. Anyway, we could do this 24 hours a day for a long time and still find new things. Right, I can introduce myself again. I'm Karen Osborne. I'm an invertebrate evolutionary biologist from the Smithsonian National Museum of Natural History. I am the sacrificial lamb for this cruise, doing the organizing and making sure everybody gets what they need, when they need, where it's at, helping with everything, trying to pull this team together and facilitating making this science happen. So it is my pleasure to do that. We put together an awesome team, incredibly diverse, about half engineers, half biologists out here. And yeah, I'm here because I'm interested in what animals do to survive out here in the midwater. The midwater is the largest habitat on Earth, and a unique habitat that we have a hard time relating to because of the nature of it, right? We live in a 3D habitat, but it's kind of more of a 2D habitat because we're all tied to a surface. The animals that live out here are not tied to any surface. There isn't a place for them to hide. There isn't a place for them to rest. So they have to regulate their buoyancy, or they have to work. What is that? I think it's a Euchronia fowleri, an orange Chaetognath. Those guys are bioluminescent, which is pretty cool. So animals out here have a lot of challenges to survival. They want to do the same 3 things that everything else. Oh, I can't have open one. All this talking makes my throat dry. Let's see, where was I at? Right, talking about the water column. What it's like to live out there. It's dark, it's cold, really 3-dimensional. You have to worry about predators coming from all around you, below you, above you. You want to be able to see or sense what's around you, but you don't want things to see or sense you there. A lot of the work that we're doing these days is looking at vision in the midwater. One of the reasons we do that is because it is a really simple it's not an easy place to see, but it's a very simple system. There is downwelling light from above where you look for silhouettes, or there is bioluminescence around you, which is very unpredictable. Bioluminescent light can pop up anywhere something's disturbed or anywhere somebody is trying to signal someone else, something else. And so when we look around, like, for example, you look around the room, your eyes are focused on particular things and your vision is kind of ignoring all the rest of it, so it doesn't overload your brain with so many signals. In the midwater, the visual scene, or what there is to look at, is much simpler. It's mostly black, few smatterings of bioluminescence here and there. Almost there, 896 meters. Then we can start filling our buckets, getting beautiful video, measuring these guys with our instruments. We have a couple objectives today as far as collection. We've been doing midwater collections for the last few days here, trying to get representatives of the different types of animals that the experts on board can identify and are familiar with. You know, is it something new? Is it something we already know? How do we tell them apart? Okay.
Let me
900.
on my
Okay, buoyancy, sure. All right, we almost have the green light to hunt. So I'm interested generally on how animals survive down here. What do they have to do? Why do they oftentimes have such strange body forms, typically very different from their closest relatives that do not, if they don't live in the midwater? There's obviously a lot of things about living up in the midwater that are different than other habitats like the seafloor or the intertidal or a coral reef. And so I'm interested in how the traits that they have evolved. And if we study how animals function in the midwater and we study them across different groups, what can we learn about what it's like to try to survive in that habitat? That's fun. Greetings to everyone on the chat. It's nice to see you here today. Hope you're enjoying some of the video that we're getting. There's a nice little shrimp down to the left.
What was that?
This guy?
Yeah.
That's a little jelly Trachymedusa, probably. No, we don't need to look at it. So right now we are testing our trim to make sure that the vehicle is balanced. Flying the ROV in the midwater is a little bit different than flying it on the bottom. It's not very forgiving. The animal is oftentimes moving, trying to get away. The water is moving around. The vehicle is huge and pushes water around, that pushes the animals around. So there's a lot conceptually, dimensionally, there's a lot of things to keep in their heads. So the pilots have to do all of this, and the vehicle, it's really important that the vehicle is trimmed out so that they're not, if it's too heavy, then they constantly have to be, using the thrusters to come up. If it's too light, they constantly have to be using the thrusters to go down. And so it's really this balance. You want the vehicle completely and utterly neutrally buoyant. That's very similar to what a lot of the animals out there are doing. There's many different ways that midwater animals can adjust their buoyancy or maintain buoyancy so that they either sink when they want to, or they're neutral so they can rest and not have to swim all the time. Or if they want to ascend, they become buoyant so they don't have to physically swim their way all the way necessarily. So we do the same thing with our vehicle. Okay, we have the green light.
Was it a pair of Sarzarians just now?
I didn't see it. So some of the targets we have today are larvaceans. They look like a little tadpole. They secrete a mucus house. That mucus house has very specific sized filter mesh in there. They have very specific structures of how they direct water flow. The animal sits in the center or kind of oftentimes at the bottom of the mucus house, and they pump water through the house to separate out a particular size particle that they want to eat. One of the instruments that we have along today allows us to image those mucus houses and look at the structure. The structure is often distinct to each species, or at least each group.
That was a Rhyteria of some sort.
So we're looking for larvaceans, we're looking for radiolarians and forams today. We should be able to get several of those. We're looking for gossamer worms. Found at least one new species of gossamer worms, probably more here. What we're looking at now is probably Furligalma. That's from Dougal Lindsay, jellyfish expert extraordinaire from Jamstech in Japan. Although you'll notice it's not a Japanese accent that he has, it's an Australian accent. He lives in Japan, works in Japan.
Furligalma is a.
It's a fascinating Siphonophore.
So we don't need to image or collect this because we've gotten these guys before. Yep.
Go ahead.
Let's keep going.
Very young colony.
Okay.
It's
Are
way
you
deep.
moving within the south direction?
900 meters.
That's right. Yes. Yeah. Affirmative.
Okay, thank you.
We've been seeing these mostly at like 750 or so. 700
Good. So,
during this cruise.
Yeah, that's okay.
It's probably the deepest record.
We can let it go.
continue.
Yep.
And it's small. You expect the small guys to be developing
Tiny
on
little
their surface.
colony.
So, Australia, you know.
You can do a slight up and forward. Yesterday, I think we spent most of our time between 800 and 900, and we want to cover a little more ground today. You never know what you're going to find. It's one of the things I love about the Midwater. Just never know what's suddenly going to pop into your screen. Let's see what that
Right.
is over there. A super deep for Thalassocalaceae, isn't it? You can see the reflection of our lights in the tissue. There are two perfect reflections of all of our lights. Whoa. It's blown right up like a balloon. That's great. Ooh.
Looking at us.
I don't.
So let's make that a Lobata Lyria.
So Lyria is a group of lobate ctenophores. There's a lot of different species. They tend to be deep occurring. They tend to be incredibly delicate. They often have a very specific hyperid that lives only on them. That hyperid is called Glossocephalus, and they have, you can see the teen rows. Is it really far away or really close? Okay. Glossocephalus is a hyperid amphipod that lives exclusively, well, they live exclusively on a couple of different ones, but there's one species that lives exclusively on Lyria. And they were the first hyperid described just a few years ago, exclusively described based on the differences of their eyes from any other Glossocephalus, any other animal that belongs in that group, that genus.
It's the features are similar as well. Yeah.
Sorry.
It's no problem. No. Yeah, it's all good. I just checked.
That green light that you see there is our lights refracting off of the combos.
That's the most reflective ctenophore I've ever seen, like for the jelly itself.
So what looks like moving lights on there are just the reflection of our lights hitting them. Yep, we left from Salvador, Brazil, and we will end in a few days in Fortaleza, Brazil. Somebody asked where all the scientists on board are from. We have scientists here from Australia, from Japan, from Canada, from the US, from Brazil. Let's see, am I forgetting anyone? From the Netherlands, from India, what else am I forgetting? We have a lot of different language possibilities. So if you would, from Lebanon, if you would like to hear narration in Arabic or Hindi or Mandarin or Swiss or German or Dutch or Portuguese or Spanish, we can provide all of those things. Japanese. I think one of the funnest things about coming on cruises, coming out to sea, is to meet all the other people who are working on board. And this ship has a really wonderfully representative crew. I have met people from Philippines, from Mexico, from England, from Scotland. I don't even know where everyone is from, but it's really always really fun to talk to them and hear where they came from and how they ended up here. Dougal, do you want to do anything with this guy? Like get him settled and scan it, or?
If you saw that.
What's up?
Whoa!
Squid.
Came on the screen.
Big one or
Anna
just
suggested. Yeah, it's hard to say if it was just close or it looked pretty
Yeah,
huge, but it was also very close to us.
Camera.
Straight in. There's a really nice suggested out to the right. Yeah. I guess that answers the question of whether we're going to scan Illyria. Yeah, so that's a shrimp.
Is it shrimp or mysid?
Good question. Tail.
Shrimp, because long antenna?
Not necessarily. Mysids can also, they're often surgestids when they have this shape antenna, but I am not a decapod expert, so don't necessarily believe anything that I say. You want to talk paracarats? I can talk paracarats, but decapods are a different thing. That is really nice. Look at that eye shine. So you see the bright orange dot at the head, which is the part that's headed down. That is a reflective material behind the retina that helps it collect more light, just like a cat or a deer or an animal that needs to see at night. They let the light pass through their retina, they collect what they can. They have a reflective surface on the back that makes any light that makes it through the retina pass through again. It's called a tapetum, not necessarily in a crustacean, but basically the same type of structure, a mirrored structure behind the retina. And that's why you get this shine back from them. One adaptation we see from midwater animals oftentimes is nicely exemplified in this beautiful shrimp, and that is extending their sensory abilities. So these antennae are telling them what is moving around them, gives them a little forewarning if something is coming up, approaching them. We see this often in midwater animals where they have figured out a way to extend their sensory abilities in a larger sphere around them. We have researchers aboard also from Argentina. Brazil, Thailand, Singapore. Sorry, it's a really big team we put together. Try to remember where everyone comes from originally.
There's bright white spots on the tail.
Look at those antennal scales at the front. That is beautiful video. You can actually make out individual segments of the antenna,
Incredible.
the spines on the tail,
Do we have any data
the
on
legs
the
all folded up
descent of the
together.
vehicle
Like
at this
that.
point?
Yeah, we should be able to
Their
get
back
speed.
velocity.
Yeah, yeah.
We know how fast we're going down.
Yeah,
10.3.
I think this is one of the ones where when they shot the
2.1 meters per second.
0.1 meters.
Yeah, 0.1. That makes sense.
we
2.1.
are rocketing.
But that is amazing. Yeah, we can back calculate its migration time.
Using a lot of oxygen.
I think it's light and it thinks it's time to go down. It's like, I don't like this light. That's incredible. Rather than escaping, the response it takes is go dive deep. So that would give it how much range?
Metaconal swimming.
0.1
Yeah, that's what I was thinking.
per second. So that's a meter.
So watching those pleopods, I'm not sure if they're called pleopods on decapods as well. On amphipods, they would be called pleopods. The little swimming legs you see there, paddling away to push them. One of the engineers on board.
The key, not the tail.
This is yeah, no. They only use it for like a super quick escape response. They can do a tail flip and push themselves really fast. But no, these are their pleopods or their swimming legs. This they can probably sustain for a long time. Whereas a tail flip they can only do a couple of times. It takes a lot of energy to use those big muscles. But these, it just takes small muscles to move those appendages comparatively. So they don't get they can maintain that forever. Well, for a while.
Well, we're on well, 35 meters on now.
Insanely fast.
Right. I might have told Jimbo we weren't going deeper than 9.
Meter.
Guys,
Alright.
really not gonna ever believe anything I say.
Stop now and
No, let's follow this guy for a little while. This is a really nice video,
Yeah.
and I'm a little bit curious how far he will
But what's
keep going. I think it's interesting that he's got some
I think
gills or
this is good
something
data.
splayed out there on the side. Seems like those would create a lot of drag.
I have a
as he's
question.
yeah.
One antenna is longer than the other. Is that because somebody's taken a bite off one of them?
Probably. Probably.
You see, he's swimming slightly lopsided because of it as well.
Oh, near. So that's another super interesting thing with monopsids. So if you read that paper, that Marshall and Diepel paper, you see them striding through the water column and they do it with 6 legs. That's what they're supposed to have. They oftentimes will lose 1 or 2 or 3 or 4. They maintain the same stride no matter what. They just move the legs around to compensate so they can still keep that tripod gait. And I've seen them with as few as 2, and they may be, you know, the opposite pair, but they'll angle one really far forward and one really far back so that they can keep moving straight as they go. It's super cool, and how they're able to figure out like, oh, this is too lopsided. I need to like shift it at 15 degrees or something. I have no idea. It's pretty cool.
iris on this.
8,000 meters in a day.
I mean,
But they
a
migrate
meter
for
in
like an
10
hour.
seconds.
What do they do in an hour? How far can they go?
Yeah, that's how many seconds in
Can we do iris on this?
This is incredible.
We can try.
Yeah, that would be
We've been successful
360 meters.
Yeah.
I mean, until he decides to tail flip away,
I mean, we are way past 900 meters now.
that's okay.
Yeah, that's 360 meters an hour.
All right,
Okay.
gonna try to do.
So it'll
the-
take him an hour to get up to where he wants to go at this rate.
We know
Pretty easy.
Yeah.
Yeah.
Pretty easy.
See if I can manage. We have the camera. Here.
This is not a suggested trip.
All right.
No.
So this wonderful red glow that you see
That
to the
is
left,
incredibly
we
fast.
have four sets of light out there. And in the center, we have a camera with a planotropic lens, which means that it has 2,400 individual micro lenses within. Oh, red and yellow, that's good. Looking for some blues and greens too, I believe. So that camera is being, the camera in the center of Iris is being piped back to the vessel. So we have a single screen that we're looking at the results from that camera, and basically it's what we see in that is kind of a rainbow colored picture, and the colors mean different
too
things.
close,
They
right?
mean yeah, not too close, but the colors tell us how far away the organism is, or how far away the parts of the organism
Zoom
are.
out on that one. Zoom out on this one.
That's what they're seeing in the
So
pit.
it gives us it's a little jelly in the background there.
Keep going
I'm
down.
losing it.
Can you zoom out at the
Port on down. He's in there. Go to port. Yeah, there he is.
Can you zoom out on the
Thinks it's bright.
Yeah, there he is.
oh, there he is.
I think it's gonna keep going.
Because you must see the pressure
Here he
sensor
comes
must be-
again.
At some
So
point
when
of
we look at the
time,
readout for Iris, we see the
this is
colors blue and green mean far away, red and yellow mean really close up. We can actually measure the size of the animal from the single image that we get on Iris. This is super challenging to work. This animal has to be held in a very specific location relative to the camera. And the animal is swimming, the vehicle is driving. Trying to get it just in the sweet spot is really challenging for our pilots.
It'll knock everything else out.
Oh,
A little bit inside.
are we scanning Mr. Antenna?
We are scanning Mr. Antenna.
Port. Down. Down.
Tested one that was 3,000
Port.
rated. And this one's rated 4,500.
Head over.
Let's see, we are at 1,000
That is
meters. We have tracked him for,
Port.
that's got to be faster than that.
That is
Oh,
incredible.
that was almost 100 meters in.
Yeah, no, I think it makes
a
sense
few
if you think
minutes.
about the 3 hour, like
Yeah.
our dial migration.
Oh, beautiful,
The
Joel.
rates make perfect sense.
The steadiness that he's swimming at is impressive to me because
I
we
think
don't
just
usually
triggered
see
his
them.
Dial migration
Yeah.
cue.
We
That's what we
just told
did.
him it was
We
broad
just told him
daylight.
it's broad
It
daylight.
is it is daylight. He just doesn't
But
usually
not at this
expect
depth.
daylight here. Oh,
He's gonna
oh, he's getting
Oh, and he's like, something is not right.
But he's still
going.
He's like, no, I'm out of here.
He's like a long distance hurdler.
I am
Yeah.
not used to running into things out here.
No, but it makes perfect sense.
That's better for me.
The long distance
Okay.
hurdlers are the
Something-
ones
something
that
else.
are in our signatures right there. He just is off the group now.
Don't worry, man. Your co-pilot had you.
Wow,
Yeah.
great.
Okay, I should get in this one. I should get in. Where is it?
Go ahead, all of that.
That is a pretty cool
turbulence
behavior.
from the light, right?
Like, but the ROV can't do that.
No, but then it locks in. I think it's the intensity. There is a spot somewhere where the two beams
All right.
cross.
Let's call that good.
All right, now
I
it's
think we're
set
good.
turbo.
I think we're good.
That was incredible.
That was impressive.
Wow.
Very impressive
Oh my
flying.
goodness, we crossed
Very
our
impressive.
boundaries. We are at 1,020 following that.
We cannot go past 1,200 with some of the equipment
Yes.
that we've brought, so we're gonna be good.
Back up, huh?
We're going back up. We can work our way slowly up. Actually, let's trip a bottle here. This will be our deepest depth for the day. So let's trip one of the Niskins.
That's
Number one
This is
would
what
be great.
happens if you
Okay.
try following someone. You get out of your depth.
This is why when we're blue water diving, we are tethered so that you don't go, ooh, look at that worm. Just head on after it and
How many
end up.
bottles do you need?
Just one bottle. Can we do, yep, number one?
I saw that it was in the works. That
I'm
looks
just gonna
great.
update the depth here since it's not what we planned.
Yeah.
10, 23.
I'm gonna take for you.
Okay.
Shouldn't be here anyway.
It's okay. Russ
know.
is doing
Yeah, yeah.
more important things.
We just chatted.
Okay, good.
Just chat. Is there a goal, a mission?
We're gonna, go to 900 and then work our way up. Clearly,
Clearly, we're on path.
clearly
already today.
we are on track.
Excellent.
Good thing the chief scientist is in the chat, keeping us on track.
All right. Anything on a no list? Anything on the must-have list? Still larvations.
Doliolid.
Yeah.
Democrates,
Okay. Do you want this one?
whatever.
Okay. Any other things? All the squids.
Whatever you want to scan.
So
Adaptation houses would be ideal.
That was which bottle was that? Was that bottle one?
That was bottle
True.
one. That was bottle one.
Okay.
I have my pen.
You sure can. Is there no? Okay.
Oh,
And we have nothing in any canisters yet. It is hot up in here. I'm in the hot corner today. Okay. All right, everybody. Hi, I'm taking over for Karen Osborne. I'm Heather Judkins. I work with cephalopods and midwater snails on this expedition, but also work with other inverts through different projects. We are slowly moving up to 900, I am assuming, since we've already hit our target depth of 900, and a little bonus today down to 1,000. We're going to be collecting stuff, and we're on the lookout now. Hi. Okay.
Putting that inside larger.
I can go
Hey, where's Arian?
for days
Oh,
now.
right there, yeah.
4 a.m.?
What? Not paying attention. Not paying attention. This is the problem.
It was in the bottom left corner.
Yeah.
That was at 4 a.m., right? Oh, what's that in the bottom left corner? That's a Siphonophore.
Bottom left corner, stop. Nice eyes, nice eyes. Seeing that the Siphonophore, bottom left corner. Coming up, maybe. Here, I know I was gonna say,
There.
these sharp young eyes in the room are finding things now. Nice job, thanks, Woody. It is a Siphonophore of some sort. We are gonna zoom in when it settles.
Yeah, when wishing it.
Mike.
Great.
When we get
currently.
closer, if we can see the bracts on the cyphosome, we'll be able to tell the species. It looks like a Philogalma from this distance. You got to see that jagged edge that looks like a sawtooth pattern of one of those big lumber saws, handheld lumber saws. And if it's got that kind of a look to the colony,
Went over to the left on my screen.
did I get a framing over that? Waiting for a good shot.
Up top corner left. There he is.
That was a fish.
Yeah.
Probably a
That's a
cyclophony.
Philogalma.
Swam like a cyclophony.
Philogalma vitiazi.
Okay, so the
See how the bracts are all
gases.
pointy? They're
Yeah,
all
yeah,
like.
yeah,
on
on
top-
the
oh, oh, it's still got the larval bracts attached.
You want zoom in still if we can, yeah?
At the very tip of the tail, so to speak,
Yep.
you'll see two really long gelatinous shields that are poking out the bottom.
You went down.
That's the larval bracts that have been retained. It looks like this Siphonophore hasn't been injured throughout its however long its life has been up until now.
It's not on our screen yet.
Oh, there it is.
There he is.
this is a small animal compared to how it will grow.
I'm not sure how big Philogalma would get to, but I'd imagine only like 15
What
centimeters
is
or so.
that?
That was
a
a
fish.
fish. I just don't know which
Is
one.
that a rosarian? How do you think that was?
No, that was a fish.
Oh, that was a
Yeah, that was a fish. I just don't think it was a cyclothony.
funny fish.
Yeah. We're coming in, Dougal.
There's really pointy bracts on the end.
Oh,
Look at
I
that.
see.
You see
The
how
very
pointy
end
they
of
are?
the tail's what you want, right? The
Yeah.
feeding. Yep, tentacles right at the bottom. If
Because
possible.
when we catch them when they're larval, they're the only bracts that are on it. And when it was described with the mature bracts, not the larval bracts. So it's like,
Okay,
oh, that
better.
was a good one. Yep. That's good.
Definitely trucking.
Yep. I think we're good on that.
You're good?
Thank
Okay.
you.
Thank you guys. We're moving on. Again, the plan is up and slow, right?
Yep.
Kind of exploring. Great.
You're in charge.
I'm just making sure. I just
I-
walked into the room.
we
Didn't
are-
want to mess up the plan.
We only ever listen to you.
I don't know about that.
We never go running off doing other
I don't
things.
think that's true. We wouldn't have gotten to 1,000 had I known. I'm just kidding.
Yeah.
We weren't supposed to go this deep. We were supposed
Just
to go to 900.
going.
And
Just
then go
kidding.
up.
Hey, there's cool stuff under 1,000 too, you know. You need to study that bathypelagic a little more.
Is this where as deep as like the guys who live even deeper come up to feed on the guys who are coming down from the
some
surface?
come from
right?
the bathypelagic up to the mesopelagic to feed. Yep.
I think that's an interesting story our listeners might like to hear more about, Heather. Don't you think so?
Well, Dougal. So we've talked about over the past few nights how there are these deal vertical migrators, right? So there's a deep scattering layer, which is in a huge abundance of organisms that come up to the surface towards feed. There are also some of these organisms that don't make it all the way up to the surface, but they live in the bathypelagic, somewhere between 1,000 and almost 4,000 meters deep. And they'll move up in the water column, but they might not move all the way up towards the surface. So they might move up into the mesopelagic to feed, somewhere between 200 and 1,000 meters. So it's super interesting as we start to be able to find these patterns. And we do this by collecting these organisms in different nets and observations like this in the ROV.
Yeah, I think it's in the more productive regions.
We've been up half an hour.
Did you read our comet tail paper?
Middle, low.
Do you know about what we're now calling comet tails?
Really, really long. Yeah, I read
We
I
just
read
did this.
Still
and
lower.
We show is that
See it and keep
tail
going.
keeps extending, keep extending. It's a
Here he
physical
comes.
tail.
I don't know what it is.
It's a bacteria.
Gentlemen, Siphonophore.
I'm thinking
Worm?
pteropod or copepod? Copepod,
It's a
maybe?
copepod. Oh, that was a big copepod. We will zoom in on the. Oh no, it's not a copepod.
No, it's a larval
It's a
shrimp.
larval shrimp.
That's nice. Let's
That's
go take a
great.
look
Yeah,
at
if we
him,
can stay with that for a minute.
And he's gonna go down, and we go down with it.
Nope.
It's
Nope.
not swimming
I'm not
at
going below
all,
1,000.
is it?
We're
Oh,
still
here we
right
go.
near
It's not-
1,000, so we're
He's
good.
gonna swim. And the dive. Come on, dive. Turn.
Oh, he went the other way.
No,
Oh, he's
just
gonna-
orienting.
Ready.
Let's dive.
Is that?
You guys think you know
Really?
shrimp
Shrimp?
behavior now.
It's really flattened, isn't
Yeah,
it?
he's flat. Is it? It's not a.
It's a.
It's not a stomatopod. It's not
No.
an isopod. It's a shrimp. Let's stay with them if possible, guys. He seems to be getting pretty close.
Flatalophus.
Turn.
What is he?
Turn. Oh,
not a
look at the
mysid.
gut. Look
Not
at
a
the
mysid?
gut.
Oh, yeah. Well,
Look at
Maybe
his
a mysid?
eyes.
if we look at the tail and see if there's reflective structures inside, we might be able to tell. But yeah, his ears, so to speak, kind of suggest it, don't they?
Yeah, he doesn't feel like a crusty shrimp. A shrimpy shrimp.
Could be something
Wow.
benthic that's.
Sorry guys for whoever's listening to us right now. Oh,
Paracarad.
It was my alarm to wake up.
Paracarad. Here we go.
I'm going mysid. Aren't the mysids the flat ones like this?
They normally have the tail gets thinner
He is
rapidly,
different.
like almost suddenly from the cephalothorax. This doesn't have that feel, so I don't
Yeah.
think it's a mysid. If you look in the tail, the two bits on either side should have a bright
Yeah,
reflective
I didn't
status
see
in
it,
it if
yeah.
it's a mysid. I don't
Okay,
see that either.
not happy with that then.
So I think I would say it doesn't look like anything I know from the pelagic.
What
so
about
I'm thinking it's a
what are the
benthic.
chances of collection?
We'll try.
You saw those gonads?
No?
It's a
But
larva.
we've been naming a lot of the males, so
That's
I
right.
think we have to give the feminine names some shots.
Definitely.
Oh yeah, yeah. Of course he's small. You're trying to collect them, my friend. We're not going to give you anything large. Close though. Oh, we took the tape off the suction sampler. Oh.
tried to eat
Oh.
Yeah.
Now he's below. Yeah, he's below. Keep him, Sonny. I don't have him.
Go back with the tiny bit.
I see him.
There
There
he
he
is.
is.
There he is.
Coming back in. Yep,
All
yep.
right.
It's
Hold
the
it.
right height now.
Hold it steady.
Outside the side, yeah.
Wait for the water to calm down, and then.
Oh yeah, he is right in front. Yeah,
He doesn't seem
Little
that
low.
mobile with the
a little low.
ratio of his swimming
Going
legs
low
to
on
that
mine.
huge cephalothorax. It's got antenna creating draft.
Coming back in front. Just passed.
Drag, I mean. Does it have claws folded up on either side of the cephalothorax? Those white things.
Mm-hmm. Just on the outside. He's coming up a little. Okay, he's back.
Oh, he's over. He's too high.
Oh, it's an octopedema. Just trying to see the big picture here. That's a trachea, yeah.
Wait.
Come on, Cindy. Suck it up. Can you guess what it is?
yes.
Is it in? Oh,
Yeah!
nice one.
Nicely done. Loretta is in.
Loretta.
Hey, Jimbo. If you get a chance, can we put a quad over here on this screen with a collection bucket view?
I'm sorry. Yeah, it was there.
Oh.
Somebody's changed it.
Yeah, no worries. And he's in, so we can change buckets.
Sampled in SS1.
Perfect. Nicely done.
Good work. This is a
Unidentified Loretta.
crusty dive today, I reckon.
Unit
It's like a
Loretta.
crusty dive.
Okay, there you go, Heather.
Oh, that's
I don't
perfect.
know
Thank
why
you so much.
Somebody changed it from 4 screens to 1. I don't know why.
I don't know why anyone would do that.
It's
But
another.
it's okay.
Sampler 1 or
It was one.
1?
An
Mhm.
So you went the other way
oikoplurid to the right, just below the tail. That long string coming in. Oh, it's really.
Yeah, this guy right here.
Really small. He's inside there. I see his tail
Yep.
moving.
Okay. And then if we could follow up that string, right? Or do you want to see the
No,
end of the
down
string?
left. Down left to the oikoplurid.
Oh, he wants the oik below.
The oik,
Sorry. We don't want the string. My bad.
A little bit left.
I thought
little
you
bit
wanted the
down.
string.
And down from here. Down from here.
Yep, yep. I got
And
what
it
you,
should
I'm
be.
picking up what you put down.
Whoop, there he is to the right.
Oh, to the right. I see.
Yeah, can you see
Oops,
him?
I'm just saying.
This guy.
Yeah.
Sorry about that. We had a couple of things to go for here.
He looks like he's still home.
Okay, is this it? We have nothing in the decouplers yet, right? Or the desamplers?
Yeah,
Nothing in the D sample.
I keep calling them decouplers.
If we get
Go on.
he's pretty close, so it's going to be hard to keep him in his house probably.
Right. Let's see if we can get some
But
video
if
on
we
him.
edge away, we might. A couple of in focus shots of his house.
Oh, almost. There he is. He's in the home.
In his home. All right.
Mm-hmm.
Have a good.
Catching on the decent.
Yep. Let's do the D sampler when you're ready.
Sounds good.
All right.
Yep. Again, we only have pretty much one shot, right? Because
Yep,
these guys
one
are
shot.
gonna evacuate the second.
It's going to be one shot, one shot, one shot until we get one.
Yeah.
Get it.
Yeah. Do what you gotta do. Mm-mm.
Where is he? Where is he?
Where did he go?
Sorry, I was
You
looking
still have
down at the annotation.
Here he is. Here he is. That's him.
Oh yeah, he's there. I
He's
see.
here.
I
He's
see.
here. I got him, team. I got him. He will not evade us right now. He might not stay in his home, but we've got him. Oh.
That's it.
Come on.
And here
Yeah.
we go. Here we go. Yes.
Yeah,
Close.
close that, baby. Close it, close
Oh,
it.
He came out the bottom. I think.
Nice.
Oh yeah, he's in there. I see him.
And is he in the
Let's
home?
zoom in. He's in the middle, round about the middle where the grey bit of metal is, just above. He's coming up to the lid, coming up to us. He just went behind the bar,
Okay. Yep, just stay right.
and he's going to be out of sight probably
Yep,
now.
he's gonna hide. He's gonna be a little elusive, but we got him and the home, we think, right?
Yes.
Nicely done, Russ. We have got you.
There he
All
is. There he is. Can you
elevation.
see him? Zoom in
Zoom in where? Oh, down here.
and go down a little bit. Keep it in focus. Yeah.
Yeah.
And zoom
Joel's
right on
like,
in.
oh, that.
Can we zoom any more? Can we,
Some more.
are we
Can
able
we
to
zoom
zoom
any
all
more?
the way in? Try and get a shot of him. There we
Yeah,
go.
he's dropping.
I want to see the pigment in his trunk. There.
That's about as much as we can. Oh, maybe not.
That's
That
it.
dot. It's that little black. Oh, he's in the
Manual focus.
back in there.
You can really zoom in on there.
Manual.
Yeah, we can't do auto. It's gonna get the metal. He was in here.
Oh, yeah.
There he is to the left,
Yep.
bottom left. You can be able to see the inside of the
Here he
trunk.
is.
Look at that.
Oh my god.
Large is not how I would
Okay,
describe it.
we're good. We're good. Everybody good?
Yes.
All
He's
right.
still there.
He is still there.
That's
I
decent.
agree.
One.
And we decided
Okay.
it was an oikopleura.
Oikopleura, yeah.
Okay.
Ocean.
Oiko. Nicely
go.
done. Thank you, pilots. And house. So we had him and the house. Nicely done. And that was in sampler 1. Perfect. Well, we're just knocking off our wish list so far.
Deep sampler 1, right?
Yes.
Next up, colossal squid.
Nearly impossible.
We got a
since they're
pretty
not in
good
the
rate
Atlantic
of
Ocean.
catching stuff here now, haven't we? Yes, chunk, chunk, chunk.
Oh, Medusa?
Could have been a rise area.
And if we could come out a little more with this camera, side cam.
Did you port him?
I did. Or submarine.
Perfect. Thanks, you guys. For some reason, you are getting the smallest organisms almost every time. Maybe you're just our lucky charm.
They fit easier.
974 meters depth. We're gonna wager what comes on next.
Oh.
Bottom center.
Bottom center. Oh, I just saw it go off.
Yeah.
Oh,
Don't-
sorry.
Is it a rolled up?
Aggressive with the pointer.
Yeah.
This guy down low.
Yeah, behind that copepod.
Yep, I got you. Sorry, Dave Fields. We love copepods. Is it that worm thing we saw the other night? Did we ever figure out that worm from last night? Remember, there was one of the worms we collected.
Lepidorincid? Maybe? It looks just like mung. Sorry, false alarm.
Okay, moving on.
We had a perfectly good copepod and we just went straight past it, didn't we? Oh no.
So we can move slightly up and forward.
It's clear 900.
Well, we could be aspirational.
Rise
Oh,
area.
area. 4 a.m.? No, that's not a 4
Let's
a.m.
go take
that's
a look.
Yep, absolutely. Let's zoom in.
It's 20 something.
It's a 4.1. I think he's got 5.
Wow. Only in one plane, the spine.
Yeah, I was gonna say
Zoa?
the spines. That's what I would say. But again, I am no Manu. Down low.
I wonder if it has radiozoa.
That's what I think. Here he is.
You can hear the
Oh, that
delay.
is not
very
him.
strange
Yeah, it is. Oh,
Yeah,
Manu.
No, it's planar.
How did you know?
Oh, I always know. Dougal and I have a deal. I'm paying him on the side to make sure he's. No,
That
you
guy.
see,
There it
it's
is, there it is.
totally planar.
That's what we said.
So hold on.
Yeah.
So
Holding.
It's not
We're gonna focus in a little bit when we get a second.
Philodendrica. And
I don't think I've ever
Spore
seen one
symmetry
that's
is
completely.
very strange.
Mm,
We'll get there.
pretty small.
Here it comes. He's definitely
Look at
tiny.
those
Okay,
spots
as
on
Rise
it.
no,
area.
yeah, the spots. I think we should collect this, guys. It's
Okay.
definitely a Feodarian. You
Yeah,
can
it
say
is. It's a Theodarian, yeah.
Theodarian.
Feodarian. It has a
It
little
looks philodendrid-ish to me.
phaedocyst right at the center.
My money's on philodendrid.
I got
think
one.
it's
No,
that
no, no.
dog.
It's definitely not.
Ah, I reckon it is.
No.
I think it's
No. I'll tell you one
Arguing
that I
about protest identity.
isolated that had
Decent
jelly
too.
tentacles on it.
We'll get a zoom in.
we need to zoom a little more.
Yeah,
on this.
let's take some
Yep.
videos first.
I don't. Right. I don't know if it's gonna survive. You think D-sampler then?
It will survive. The core will always survive.
Okay.
It doesn't have the. I think what does not survive is our Hestigirella, the foram.
And that means there'll be no more D-samplers. Is everyone good with that? We have a larvation
No,
for
we could
rough.
do a suction sampler for this if that's okay.
Whatever you want. That's why I was
It'll be
asking what you
broken.
prefer.
I think D-sampler would be ideal because we want to get what those punctates are. And
Can we put
especially
anything
Those
else?
our
white spots are
symbionts.
weird.
What are the white
'cause they're-
spots
they're
on the-
equidistant as well.
I
That's
mean,
like
it's very strange.
What can we put anything else in with it?
There we go.
Yeah, I think the challenge is that once we do that,
We don't wanna lose it.
flushes out. Yeah.
Right. Yeah.
Yeah.
I'd say
It's a Feodarian.
Which means what?
it's a Proteus, a
It's
Rhizarian.
Rhizarian. It's
Single
a single
cell.
cell that makes these hollow silica shells.
Little.
And my money's on Coelodendrid cousin.
Yeah, no, no. I'll show you what I think it is because
Behind it is just some house pieces.
No,
Looks like-
behind
could
it
be a
is
Medusa.
oh,
a Trachymedusa.
I think it's it's Medusa behind it.
Yeah. Maybe D-sampler the Protist.
Yep. Let's go
Yeah.
D-sampler 2 with the protist. Just a walk
Those-
in the park. It's just a Thursday for Joel.
okay. Here,
Get all these tiny
Just another
things out of the way
single-celled
first.
organism I
Yeah.
have to put into a very
it's
small
not the
container.
species, but look at that. That's the tentacle of either a jelly wrapped in.
Mm.
I found this in the Tucker Troll.
I can't.
1, 2, 3, 4, 5, 6. What was the symmetry?
Only
there?
four.
Four.
Yeah, this is different. It
Well,
would be
sometimes
amazing
Crap,
if
did you get
it
him in
had
there?
symbionts. What
Okay.
depth are we?
Okay. You keep
1,000,
doing your thing.
Oh my
969.
goodness. That would be
Oh, is
crazy
this in?
if it did have. Did we catch it?
No,
No, not yet.
we're setting
we're
up,
just
testing the equipment before we go in for it.
I was like,
Calm down, man. Calm down.
I don't have 'em.
All will come
Anybody
to
have
those
'em?
who
Oh,
wait.
isn't that this thing?
This guy? This guy? This guy?
The,
Oh, no, I haven't.
Oh.
You
Got
see
it.
him?
See that?
Oh, yep. I do see it on your screen.
It's straight above the black.
Yep. You keep watching yours. I don't. Oh, here he is. There he is. Is that him? That's not him.
Right between the two black. Oh, about to go over the left-hand black. Whoa, he's going over the D-sampler.
Yeah, right
just
there.
past
It's
the
above the
D-sampler.
D-sampler. Past
coming
the
a-
D-sampler up above.
Come on,
Right
center
there.
screen
You-
now.
you do-
No, it's right
Bottom
there.
quarter.
This one. It's
That guy.
That's the
Okay,
guy.
thank you,
That's the
Manu.
guy. No, that's all Google.
Thank
The
you.
one who spotted
This
it.
guy right out here.
Yeah, coming.
Thanks, you guys. Many
back.
eyes. Don't lose them.
Yeah, this is man overboard. Keep that finger pointing on the person, or the animal, or the whatever
Yeah.
this is in this case, the rosaria.
Cell.
Cell.
He went off.
You carry
He's
on.
close. He's close.
It's just inside.
Yeah.
No,
he's in front.
It's inside. It's
Yeah,
under
oh,
the
he's
lid.
under the lid.
Was under the lid.
Okay, now he's right
Oh,
here.
he's got caught.
He's stuck on the.
He got caught
on the
that's how they
screw.
eat.
Yeah.
That's how
There
they
he
There
is.
he
He's
is.
up.
Yeah.
Oh,
There we go.
Yeah.
It's
Really?
nice.
Yeah.
Close.
he's in. He's in.
Close.
He's
Close.
in. Nice.
I think you got him.
Oh.
Hey,
Great work, team. Oh my.
Good stuff. And do a zoom
Something smaller
in
next
so
time?
we can see you.
Joel's
Something
requesting
smaller.
something smaller in the
Okay,
future.
Still too big for him. All right.
I think we're Rosarian.
I'm calling it a coelodendron.
Coelodendron cousin.
Oh, it is in. It is in. I can see it.
Here it is.
Of course you can.
Yes.
Oh, there he is.
He's
Oh,
there, he's
he's
there. Let's
beautiful.
Zoom in nice and close.
Yeah, let's get some zoom on this
I
because
think
the
that's
chances.
a great catch.
Do you want to give it a name? So we've got a name
yeah, let's
for the.
call it Theodarian star.
Just-
Darian Star.
I'm just gonna look in Heckle's drawings in a second, so I'll
Yep,
give you
yep.
possible names soon.
We should call him
Nice.
Asta.
Rosaria
Can we call him Asta?
Asta?
And then we can make.
Oh, look
Like
at
Asta.
the cap.
Then we
Yeah,
can
he's
make
got a
the.
big old
See the
circle.
hat?
Yep,
Yeah,
I
the
see it.
hat is asymmetric.
Asta,
This is a
all the way.
classic
Yep.
Theodarian.
No surprise there.
Asta la vista, baby.
That's all we can get for Zoom. You're good,
I
Manu?
think we're good.
Okay, we're good.
Guys,
Thank you, team. Nicely
Do
done.
two, right?
that might be our new
I'm just
species.
gonna giggle now because
You're gonna need
truly
glasses
you are
after this.
All right.
glasses. You're never gonna want to see
So
snow
now-
again.
Something
We're
less
gonna
than
ruin
4
you.
millimeters is the order.
Like these midwater samples never.
We've done the.
Don't you want to stay longer?
Yeah.
Have to
We're gonna have a pilot shift real quick.
urinate and have fun.
We've worn his eyes out. He's like, whoa, whoa,
Yeah,
whoa.
he just said he needs glasses.
Oh, and we could call him Theodarian X.
There you
Because
go.
we don't know yet.
X.
We
There
are
you go.
going up slow
Somebody
and
said that in
forward.
the chat.
Yeah?
Well, the
Theodarian
target
X.
was 900. They chased a shrimp for a while, which brought them down to 1,000, and we're working our way back up slow.
Oh, X. No. I meant X.
So we have
as
no
a-
more D samplers
as a
available,
variable.
and we've used one canister. We have 7 canisters and some Niskins.
They're all
Yeah.
Yeah, I think it went up.
So that's like, what do you reckon? 5 millimeters?
Let's a little bigger.
Good,
Little bit
better.
bigger.
Yeah,
The arms make it big.
Yeah, the arms are bigger.
it must have been something of that. It was. Yeah.
Let's
definitely
do
better.
something a little bit easier, like a 60-meter long Siphonophore.
Yeah. Or perhaps like a 20 centimeter cephalopod.
Yeah, there you go.
Just to get video because he's too big. That's a. I wouldn't feel comfortable
So do we have
saying crustacean or fish.
restrictions today? No,
yesterday
yesterday
was
was
like,
like no jellies. We kept seeing
We had to catch up, right? There was a lot of processing catch up that had to happen. So I believe everything is fair game today now, right?
We're
Are we
back.
allowed to collect jellies or siphonophores? Okay.
Everything's
And then
processed.
Yost, anything you guys need on DPRD?
Went to bed at
or do you have
7:30.
a wish list still or?
Anything that's new,
Okay, perfect.
Yes.
anything that
Very
excites
slow.
people above the base level.
Everything
it.
that excites me above the base level. Ah.
Middle?
What's my base level? Oh, yeah, bottom.
Oh.
But there was a rosarian there somewhere.
This guy?
No, it was the
Yeah.
bottom of the screen.
I don't know
So
if
that's
it's
a
just a
long,
long thing
thread.
attached or if it's just. Yeah, let's check out the bottom quick first if you could. And then if we could follow it up to see if it's connected to anything.
What makes a structure like
'Cause
this?
this is like the
length,
third or fourth
right?
one.
Yeah.
It's got to be something that falls off. Could it be a feeding tentacle of a siphonophore? Or a jelly? There's segments
in
to
the
it.
background there.
It's a bulb. Okay, if we could.
Oh, are they two eyes?
I don't think so. Can you go back to that bulb if possible at
Go,
the bottom?
yeah, zoom me in.
Thank you.
You guys keep finding cool things.
Is this it's not a rhizarian.
Let's zoom in a little bit.
Yeah, that's what we're working at.
The stiffness means that it's
But it's
mineralized.
super long. Like super long.
It's mineralized.
Yeah, let's do it.
Hold on. Whoa.
I think it's a tentacle of
Just
something.
a piece of mung at the
Yeah.
base.
I think, yeah, if we could follow it up.
It's real close to the ROV.
Yep,
Yeah, I
it's
think
good.
that's
Super
why.
long
Is that
though.
a dark body in the middle, or is it just
But look at
transparent and
these.
it just
No,
looks-
there are regular bodies there
Right.
Yeah.
all along.
Yeah, that's perfect.
I mean, it could be the
Keep going. I don't know when it's gonna end. This could go on for 50 meters up. We don't know. Just don't know.
and it's completely straight. Like the edge has no like.
And it's not retracting.
If it was a siphonophore, well, there's bits and
it's
pieces
just
on
gonna
it.
end.
Is that a copepod sitting on the strings around the middle
if we
there?
could get a zoom on the
Yeah.
end of it, if possible, then.
Can you see the copepod sitting in the middle of it, Heather?
Where do you see it? Oh, this
there.
this
Is
guy.
that a copepod, or is
If we
that
could zoom in on
that
that.
might give us an idea if it's a
Yep. I see what you're saying.
gastrozoid
Feeder.
or like
No, I don't think it's a
something else.
copepod. It looks like a little bulb coming from the body.
yeah, maybe a palpine. It looks like it used to be a siphonophore.
That's what I think. I think it's a
Yeah.
part of a siphonophore.
So maybe the stem
What
of a
about
siphonophore.
microplastic? Except not so micro.
That's decomposing.
Yeah.
Yeah, a decomposing stem of a siphonophore, and that's like one gastrozoid that's still hanging on because it's the one that ate last or
That
something.
makes more sense to me. This feels like what a.
It's pretty big too.
yeah.
I can't think of anything else
That
big
would
that
be
would
like
have
that, right?
things along its length.
Cool.
I've never seen that before.
Right.
It's crazy.
Can we drive it past the piv just to along the whole of its length? Will you get if it's a 3D model, you're going to get a length measurement for that, right?
Yeah.
My stereo cam's not in there right now to get length.
Should be able to do that.
I think so. DPIV drive along it. Oh,
Yeah, drive along it.
yeah.
What is the tick?
Sarah, not a challenge.
No, I am about it.
We are gonna challenge every pilot today. Apparently, that's how this one's gonna go. Yost, you're good and ready?
Sending.
Almost.
Ready.
Yeah, perfect. Thanks. All right. So try to get it sideways. I don't. Do you want them sideways or get them at all?
Anyway, as long as we get the entire length, so we can measure how long
you
it
want
was.
to do it as long as we get the whole
The
layer.
whole thing through.
Yep.
Yeah.
I'm sorry.
Just pretend you're sucking on ramen at a ski resort in Japan.
You're reminding me of the second meal now.
If it's easier,
Some
so we can
noodle
see
slurping
it,
going on here. Miso ramen, kudasai.
right? Here-ish, if possible. Yep. Do what you gotta do. Okay, coming in. I don't think there is a head. Yeah, so I think that's what's gonna happen. And they're just trying to get a size on it. So however it can make it all the way through would help, I think. Right, Connie?
Okay, here we go.
Yeah.
Before the thruster wash gets in.
Coming in. Moving along. Ooh, this is lovely. Oh. Got halfway. Got halfway. Nicely done.
I don't know if that was halfway. I think it's much longer.
Sarah's like, it felt like 10 meters.
I know. No, it felt like we barely.
We've got a lot of ROV light on. It's kind of a tricky one.
I think that's.
Kakani said the light
Yeah, I
could
can
be
just
on.
see the dot, I guess.
Yeah, it's.
Didn't
I'm
get
just
to
worried
her.
about.
Yeah, it's not gonna work.
Yeah.
It's way inside.
Already have lost that reference.
Too many angles.
Wasn't
Right.
continuous.
That's going to be too hard to reconstruct, eh?
Yeah, this one's not gonna be easy by any means.
But isn't this what your stereo cameras are for?
It's not on.
It's not on. You took
Yeah,
everything
we,
off again.
we couldn't download in time. Next time.
Just start down, go up, sideways, around.
Exactly. Exactly that pattern is perfect.
It's like the little ring you have to go around this rod and not touch it.
Yeah, it's a bit like that. That game operation.
Yeah.
If we miss it, there's an oncoplurid behind it.
Yeah, but it's curved back now. So.
Yeah.
Ugh.
I think it's a discarded Siphonophore something.
Yeah, I think we should like roll it into a little ball using the manipulator and then put it through at one time.
You could probably suction it.
I mean, we could suction it.
You could measure it really accurately.
Well, unless it breaks into 85 pieces.
All the specific.
Hi, guys.
What about 86?
Maybe
Next time we'll bring a ruler.
if the suction's on really strong. 86. Oh?
Okay.
All wonky.
But he's in there. He's. Yost.
I think it's too hard.
I'm not sure that this one.
Yeah.
Suction it?
I don't know.
No,
that
I
it's-
think we're cool.
I think we're okay.
We're okay. Oh,
wasn't too bad.
no, that wasn't bad. Okay,
All
let's
right,
move on. I'm
we're
pretty sure
good.
that was a gastrozoid hanging off it, so I'd say it's a siph.
Yep. Okay. Thank you guys
Dead
for the
siph.
attempt on that one.
Dying siph.
That was a challenge. Mm-hmm. Exactly.
There it goes.
Could be anybody's.
Oh, maybe you.
We could drop the light a
iris wouldn't be there.
smidge. Exactly. Top, middle, yeah,
Everything's together.
please.
Yep.
From iris.
Yeah, that's good.
Oh, I was gonna say, are there enough identifying features on that
I
to
bet this is an apolemia,
This guy, middle.
we could have gotten a size scale?
and after we zoom in on him for an ID, slightly to the right and further away, there appears to be a
A
fish.
vertical.
Yeah,
Yep, I got
vertical
you.
fish.
I got the vertical too. Yeah, let's get this guy first. I'll keep an eye on the vertical. Oh, you think it's apolemia?
Just a second. I think it's going to be an apolemia. Yeah, it's an apolemia.
Good. We're good there. You want any more bit on this?
Okay, I think we're fine.
Okay.
Okay.
Good, good.
Oh yeah,
Go.
that's good. That's a good one.
Zoom on out so that
By chance
vertical.
related to the thread.
there he is.
Apolemia. It's a Physinect Siphonophore. We're coming in on a cereboma, probably.
Yep. I think you
But
are
it could be
correct.
anything else.
Or
you know,
something in that.
Till we get close
There's a
enough.
couple of genera in that family.
Looks like a Cerebomiridae, is it?
Working on it.
Yep.
Looks like it. Swims like it.
And a Keatinite.
Of course, there's
Don't forget the
Keatinite.
Keatinite.
We could stay with the fish if possible. I don't know if that's going to go well or not. He's getting close.
Cerebomiridae.
There you go. Nice. Nice job,
Yeah,
pilot.
you can see the beak is.
Yeah.
Bend
Any
up.
chance for more zoom, or are we going to lose him if that's the case?
What is this thing? Is it an eel?
It's an
yeah,
eel.
it's one of the snipe eels.
Sawtooth eel, I think they're called.
Yep. Over to the right on my screen.
That's a skeena. No, is it
That's a
copepod or
hyperid
skeena? It's a
amphipod.
hybrid, right? Was that a skeena? Something else?
Up top. There he is. So let's take a look. If we can get a close up of the head, if he'll be willing to stay still for a minute. It's a good indicator. Oh yeah, this would be great. It's okay. It's okay. He's gonna come back.
Slowly.
Went over to the right.
Okay.
Yep, right. Nope, that's a Keating Nath.
Dang it.
We might have lost him. We got a quick close up.
Hmm.
Okay.
Oh,
Oh,
there he
you
is.
got him? Oh, yeah, he was right up in the cameras. Oh, there you go. Okay. Let's try one more time on the fish just to get a close up of his head, and then we'll move on after that.
Stay on the sticks. Zoom
Okay, his head is now currently pointed down.
in. Go for it.
He's going to decide to swim.
Good. Going down.
We want to check his
down.
dorsal fin compared to the anus.
Yep. Okay.
And
There you go.
count those vertebrae.
Oh, he's so close. He's like, you want a close up? I'll give you a close up.
It's like, dive, go
No.
fast, dive.
Okay, one last shot and then we'll move on. That's how he normally positions himself. Oh yeah, get him before he moves.
Close the iris a bit if possible.
No. Okay. Okay. I don't see the tip of the beak curling like the other one did the other day. Okay.
That's a money shot.
That's great.
There you go. That's good.
Great job, pilots. Okay. We're good.
Good.
Good deal. Thank you. He is cool.
Those fish and shrimp today for sure. And the tiny
far.
little things.
Yep.
I don't know about you, but I haven't seen that many jellies yet. What do you reckon?
Can we make it a smidge
There-
darker
Fewer of them down
on
here?
the side cam? Yeah. Yep. Thanks. Yeah, that'd be perfect. Like a forward and up slow, however you want to work that.
Gentle. s-
We're good. A gentle snowfall. That's what I like. Gentle.
Middle, bottom.
Back down.
It's Siphonophore, I think.
Good eyes, Dougal.
Looks like a lobate.
Lobate Siphonophore coming into view. Maybe.
Seems a little bit Bathysauroi-ish.
Nicely
It
done,
looks
pilots.
like it's
This
been
is
eaten
great.
once because one lobe's bigger than the other. We may be seeing a Siphonophore that's grown back part of its body after a predation event from the look of this. Look how different the lobes are.
Yeah, they don't seem symmetrical. Oh,
The tentacle bulbs are on either side of the top of the pharynx, so it's a Bathysauroi
Bathysauroi.
that has lived through some predation event. The comb rows are short, suggesting it's Bathysauroi fosteri. Is it completely healed? Take a little bit more of a zoom on
Yeah.
the...
Let's do it.
And
Better?
Whoops. Bathysauroi fosteri. Yeah, that second lobe seems
Compensating.
much an actividema since there's so many and we haven't caught
Sure.
one yet.
And is this a likely
That's
candidate?
that's a suction sampler after a
Okay.
zoom.
Yeah, let's zoom on this guy, please. We're gonna zoom on him for a bit and then try to suction sample. That's why he's been a challenge.
Yes. There'll probably be a heap more, so it doesn't have to be this particular
Right.
guy.
Okay, there he is. He's headed left. Oh, now he's headed in a swash.
So, prioritizing the next sequencing done on board.
Quick.
Uh-huh.
Try to prioritize.
They're so
things where we
small.
have
Yeah.
a lot of
No,
these
yeah, we're gonna
information.
go for this little Medusa
So,
right
This
here.
one, 905,
We've
maybe
seen a
the
bunch,
shadow
but we
graph
have
is
yet
still
to
working.
collect one. So
And
Dougal would like
then
to
we have
give it a
virus
shot.
and BPIV,
if
and I think this one I got a
possible.
lot of this liquid DNA from. It's this
Video
Siphonophore.
first?
Yeah, that'd be great. Yeah, we can give it one more shot at video and then we'll...
Video.
Oh my god! Woo!
A fish.
Sorry,
or-
your jelly's
alright.
out the window.
Out the window.
Sorry, go down.
Whoa.
Yay!
Oh, look at the eyes on
Oh
that.
my god.
Is that
follow him.
Taninja?
Follow him if possible.
Is it Taninja or something else?
No, it's not Taninja. I think, well, I have to look at the fins. The fins, if they're large and triangular, oh, I bet he's gone. He's already gone.
I didn't capture it.
We don't have room for him in any sampling device we have, so.
What can I call him?
Must have been
I-
a squid. I could hear it in the next room.
Here he is, wait. Maybe if we just stay steady for a minute.
Back and down.
Maybe if
I've
we
just
just stay
been-
steady? Yeah, I don't know.
I've just been going back and down. It's
Perfect. Yeah, that'd be great.
match, but
Turn everything off.
We're turning this car around right now, kids. So that was a squid. As to which one, I'd like some more video, if possible. We want to look for the fins here. That was a lovely crustacean. He might be done for. Yeah, I'll go back to the video. I can go back to the video and take a look, but you marked it as a squid, yeah?
Do you get
If you mark the
Octopachutus here in the
squid-
Atlantic?
I don't even know if I can get the genus. What?
Do you get Octopachutus in the
We
Atlantic?
get Octopoteuthis. We get the Thysanoteuthis.
It wasn't
We
a
get-
Thysano.
It wasn't Bathyteuthis.
Wasn't bad.
Right. So maybe I would just go other at this point.
Okay.
And just put in here, go to the comment and go dark red muscular. That's all I can give you at that point. Thank you guys for the attempt on that. Should we try to find that little Medusa again?
Thank you, Heather. Yeah.
I understand our target. I understand our target.
I mean, just if there's no squids around, that's fine.
Maybe a Taninja. We have to take a look at the video feed to see the fin size again. I didn't see any visible quick photophores. We could check for hooks or suckers on arms. So there's some things I can look at with the frame grabs when I can go back and look at the video.
At least we have some of
But
it.
very cool.
I think for Illyria,
Little
we have
finds. See, there are some big squid in the ocean. Nice eyes, pilots, on that.
Probably, but I don't know which one would be more exciting.
Spinoceros, there's a paper that's...
League man was like,
Well, thank you. I'll use that as the universal. That's a squid. How's that?
We don't like the baseline.
You want a genome?
The live stream.
Wasn't a Mastigoteuthis.
That one.
I'm gonna
that
keep
one
going through all the things that can't
is
be
the
now.
best potential out of everything so far. Jellyfish seem like they're too big. The other Ctenophore.
Yeah, we don't have Humboldt squid here in the Atlantic. Those are Pacific species. Yeah, please.
okay, well then I'll go
Talking
now.
about that,
So the whole genome...
So the Humboldts are over in the Pacific, off Mexico and up the west coast. Yeah. Is this his copepod? No.
Start now.
Awesome.
Risaria.
Skeena.
No, it is not a Rhizaria, my
No, there
love.
is on the right.
Oh.
You guys missed it.
I'm looking at the
I think
target we
this
were
is,
just going for.
My eyes
I
are
think
gone.
it's Skeena or a hybrid amphipod. Yeah, the elbows are coming out on the
Okay.
side there. I'm pretty sure that's what
He's
it is.
definitely not going to stay in this in
That
this
Rhizaria
screen.
is probably
Okay,
gonna eat
we
it.
can keep moving on. We're okay. We are good.
I don't know what Skeena. Let's make it Skeena Day.
The good news is the squid is probably right behind the ROV, just watching what we're doing. I think that's what he does. All right.
You're probably not wrong either.
No,
It
we have
probably
seen.
is.
There
Yeah,
have
they
been
hang around,
some-
right?
Yeah, where they have the double ROV systems. We've been able to see one ROV see the squid behind the other ROV, like out of the field of view. It's
Right,
pretty cool.
yeah. Top left.
Yep, good call.
Is that another string?
I
that
think it
used
looks
to
like
be a
another string.
Siphonophore?
What's going on with the Siphonophore?
What's going on? Oh, this. Yeah.
They were awesome.
There's a whole bunch of mucus
Yeah,
on this
zoom
one.
in on them and see if it's got gastrozoids hanging
Yep.
off it.
Take a look again for those little bulb-like structures we saw before.
Ooh, I know what this is.
What is it?
Snotulus elongata.
I think you could be correct, Yost. I don't see any of the bulbs like we saw in the other. Do you?
Yeah,
Do you
no,
go closer?
at the very bottom. See where it's just,
Here?
whoa, that one, yeah, it just moved.
that's
Or,
the bottom?
or it might be a
Yeah.
shrimp sitting on it, I'm not sure.
Are you looking at the mucus
Go
section?
down, whoa, whoa, whoa,
Yeah,
whoa,
you
whoa,
want
other way,
low.
other way, other way.
Okay, we need to go to the
Down
bottom
there,
of this.
yep.
Okay. Yeah.
And
You and me both.
pan camera right to bring it, well, bring ROV right, it's better.
Yeah. They're
Can you
coming
see
in.
that thing sitting, looks like it's sitting on top of the stem? Not those Fiadaria or whatever they are below.
Oh yeah. There's a little
But-
shrimp or something. on
Yeah,
there.
yeah.
You're
Want to
good?
see
No,
if
no,
that's a
don't
stomach
apologize.
or something else.
You're good. He's close, Google. Yeah, stand by. Yeah.
Someone suggesting a speed.
Thank you.
Big goo structure though, like it was a larvation house it was caught in or something.
And it's reminiscent of decaying tentacles or something. There we go. That's the money shot down there. That thing hanging off it.
Yep.
Yeah, it
That-
looks like there's a little tubular thing.
On it.
Yeah, on it,
Yeah.
like a stomach or a pelvic.
Agreed. And I see it a couple other places along the string. This is the same thing.
Yeah, I reckon it is.
Okay. Do you want to pan up just to the top by that mucus
Yeah, just
house that looks like it was just attached to it?
Oh,
Oh, let's take a zoom at the tip here just to see. So here you can see a couple of those, Dougal.
yeah.
The bumps.
I reckon it's the stem of a Siphonophore.
Yeah, I think you're right.
They must be.
Where's all the stuff gone? Is it all eject? Like the one we saw yesterday?
Yeah, like all the
All right,
zooids, I think.
we're good,
of-
you guys. Thank you.
And the gastrozoids.
Mm.
What a mess.
I see in the chat somebody said it looks vaguely like a Philodoteuthis. That's still an option on the list as well. This is
Philodoteuthis.
another deep sea squid. Philodoteuthis.
I thought it. Oh, sorry. I thought
Philido. No, not this guy.
Another shrimp. Another shrimp.
Another shrimp, which is great.
Pretty color.
Here's this guy. If we could just zoom on him for a second, if he will be agreeable. Lots of antenna on this guy.
That guy, yeah.
He's beautiful.
Very
Nice job,
different kind
pilot.
of swimming. I wouldn't mind a little iris on that. I know Sarah's been really itching to do something. It's gonna be the winner.
Okay.
That's great. Benthesema.
That's what I wanted to say.
Benthesema is most likely. But again, I would need my
Can you spell
crustacean
it?
field guide
Can
in
you
front of
spell
me.
it?
Benthosema. Great. That's great. Because those antennae are at the right angles. They're right angling those tentacle or the antenna. Nice. Yep. We're good. Okay, you guys, thank you very much. We can move on.
Were you not doing iris?
Oh, we are. I'm sorry. My bad. I thought you were talking about the camera. Right. You were literally talking out your device. He's right here. He's in your iris almost. Sorry, Yost. I thought they were talking about the camera settings for the.
Don't worry, you're not being ignored, mate.
No, he wasn't. I didn't think that's what it was. The discussion.
We're not just ignoring what you're saying, don't worry.
No. I don't want to be that girl. No. Conversation.
This thing they have
You
on the
got it?
camera is an aperture, not an iris, actually.
Oh, see.
Common misconception.
There he
Where's
is. You
my
got him,
colors at?
Yost.
Where's my colors?
Too close though, right?
Yeah, it's a bit
We'll
too
get there.
close.
We'll get there. She's on it.
Too close.
Oh.
There we go. Yeah.
Backwards.
Yeah, there for a minute.
Oh, you want to stay in, if
Yeah,
possible.
I'm trying to get the flapping legs.
Okay.
See that movement.
Okay.
So some orientations are better than others. We don't really control that so much, probably. But see
Yeah,
here
so just give a
more
few
of the
passes
legs.
and see how it goes.
Yeah.
Like that.
Oh yeah, yeah.
That's
yeah.
what. That's great.
And hopefully it'll rotate upward or downward to get. Yes, there. There we go.
That's it. Thank you, Crustacean, for agreeing with us. Being a willing participant.
All right,
yeah,
one more
he's-
because I changed the frame rate.
Yep. Yep, we'll get him again. Yep, he'll back up if either you or him back up. Stand by, here he comes again, Yost.
Go, yeah. Now, little guy.
Yeah,
swim up,
different
swim up,
swimming
little
angle.
guy. Yeah, keep swimming.
Too close.
Swimming too close.
Yep. He's like, come on.
It's a good angle. It's a good angle.
Yeah, we could if he wants to stay in this
Yeah,
position.
Here we
Yeah,
go.
there we
Money shots coming. Money
There
shots
we go.
coming. There we go. Can we try and suck him up? He's going to be really hard to catch, but if you've got that swimming information, it'd be nice to actually
Yeah.
know what it was.
Put
right?
it with it. Yep.
Even though there's that beautiful looking Siphonophore at
As
the
I
bottom
say,
of
there's
the
a
screen.
Siphonophore back
yeah.
there.
Yeah.
But I'm game for whatever you guys want. We've only used one. Did we use one suction
Nice body
sampler
shot
so
there,
far?
perfect.
It's a low bait of some sort. Like big Siphonophore.
Almost. Come back in, little guy. There he is.
Oh,
He's like, how about this angle?
Yeah.
I'll give you a little side view.
All right, I got what I want.
Okay.
Thank you.
Let's try to suction sample him. You're gonna have only one shot because the second those antennas touch or feel something, he's
Oh,
gonna be
yeah.
out of here.
And a
Max up,
full-
right?
yeah,
or should
max
we go no
up.
suck till the end?
No suck until he's right in the
Right.
funnel and then smash.
Yeah, try to get him in the funnel and then suction.
While
'Cause if he
driving
feels the
forward
suction,
and
he's
ramming.
gonna I think the second he feels the movement, he's gonna ditch. But you guys decide. Dougal, what do you think? No suction?
No suction until he's right close
Yeah.
and then
I'm gonna
boom.
need you to.
Me or you? Him? Cut it.
I have suction at the moment.
Yep.
You want the nozzle out more? Or?
Nozzle out
Remember,
more
it
is
doesn't
probably
quite come on,
better.
like,
Yeah.
instantly, you know what I mean?
Yeah.
So I'm worried about bonking.
Nope, whatever you guys want to do. I'll just give it a shot.
Okay.
We'll just chase him another 1,000 meters or
He's
so if we need to.
Too far left.
Yeah. He's going off over.
Nope.
Now he's like,
Oh,
He's
I thought he was
gone.
gonna
Just
go in.
underneath.
yeah.
Down, go back.
It'd be great if he just backed right into the funnel on his own.
Yeah,
That'd be
right.
Very helpful. Here he is, up in front. Or on the side. You got him. A little low, maybe? He's not in my view currently, but I see you got him. Here he is. He's jetting off the other direction.
So technically, DPIV is made to look at, the flow around
Okay,
legs.
he's
of
right
these
at the
types
top of my
Yeah, yeah.
screen. He's coming back.
It's pretty tough to line up. Turns out,
There's not enough particles really to.
not many particles. So you do particle tracking, not really PIV.
Come on, buddy.
So what do we just shred? So it's just a vehicle coffee.
Mm-hmm. Mm-hmm.
Oh, thought he almost went into the funnel then by himself.
I know.
I think this might be a bit hard. Oh, there he goes.
He went way up and over.
Yeah. I think this might be a bit.
That might be it for
He
that
might
guy.
be a bit fast. Yeah. All right. Well, it was just in case.
All right. We said we gave it a fair shot, right? That
Yeah,
you guys
we did. We tried. Yeah.
Okay. It's all right. It's all right. We knew that would be a tricky one. All right. Back to searching, my friends.
Oh, there's an Octobedema.
Where?
Left, center now, a little bit off to the left. Down here. It's about a quarter of the way
I don't
from the bottom of the screen.
see it, my friend.
Slightly to the left of center, driving forward.
Yeah. Yep. I see what he's saying.
Coming towards the center a little bit down.
Not that.
Left a bit
Do you
more.
still see it? We
Yep.
don't see it
It's
up
in the
here.
center now. Center up down wise. It's going up to the top of the screen now. Almost hit out of the screen now to the left.
I know. I still
And
haven't.
turn the ROV left.
I haven't seen it.
Up and left.
That's right.
Oh, this guy maybe.
Maybe this
That guy.
Yeah, not sure, but I think so. Oh, it's because my glasses are off. I was cleaning them.
We both did like.
Like, wait a second.
Is it
This?
just an oik?
I don't know.
Oh, is that the Bathurst Row we were on just before with the?
I don't know. Maybe.
Or
We haven't
no,
gone very
it's
far.
a Doliolid.
Doliolid. That's what this guy is. Yeah. Now that I
Take
got
a
it.
closer look.
Yep. Of course. Zoom in on this guy. You guys are doing great.
It's a solitary Doliolid.
Correct. Yep.
Did we get a Doliopsid before?
Wash. I got too close.
Yep.
Okay.
Okay.
All
That's
right.
cool.
Moving
Next.
on. Doliolid for sure. Solitary. Yes.
Doliopsid. Yeah.
Yep.
Oh, there it is. It's right out.
It's in the middle.
Oh, there he is.
There we go.
We good? You want to
Go
zoom?
take another zoom.
Yep. Absolutely.
I think Russ has caught the like he's got under the video, the microscope video, this solitary guy.
Yeah.
So it's the colony that we really.
Right. That's what
I'm
I had
looking for,
heard.
right.
There's no room for the colony on this dive now, unless we ditch your oikoplurid or a rhizarian.
We can try a suction. Like if we microblast it, it might work.
On a colony?
Yeah.
Okay.
The stolons are fairly strong.
Got it.
Good.
Okay. This is great.
They're so reflective.
Uh-huh. Perfect.
Oh, it's got babies.
Yeah. Great.
Thank you.
Okay. We can move ahead.
Move ahead.
Yeah.
Yep.
Like back to search mode. Yeah, no.
All right.
If there is this guy in a colony, we're going to try for a colony.
They're weird because the colony's not like linear. It's like Christmas lights that are where the bulbs are hanging
Yeah.
down from it, right?
We saw one of them, was
The
that
ones you get
yesterday or the day
from Costco.
before?
Costco Christmas lights. There's a Doliolid nurse.
Where do you see that? Or are you just
It's-
passing?
it
Are you
was
just commenting?
passing. I was just commenting.
Okay. Fabulous.
I didn't scream.
That's true. I should have recognized. Like my oh my god scream for the squid.
Yeah, yeah.
I understand.
Well, look, we do have people from the Netherlands in the
Yes,
chat.
I was gonna say.
I can totally speak Dutch.
Joost,
They
Go
can come
for it.
to
This
you
is your
because
time.
clearly they struggle with English.
Doc.
Yost, go for it, my friend.
Doc.
This is your
Ja,
time to
goedemiddag.
shine.
Groeten daar in Nederland.
Yep. Go for it. That guy?
Don't say more than hello, though.
Oh, I think
I
it's
don't
a
think I need
cute.
to.
Nah. I think this is a Keating-Nath, but I asked her to slow down. Yeah, I think it is. We're good.
Okay.
Bye-bye. Yeah. Nope. Good. Fabulous. Mm-hmm. This little guy?
You hold.
Oh. Here you go, Dougal. Somebody coming up on the right. Middle.
Oh, nanja korea. It looks like a Pseudusa, I think, the carnivorous doliolid. Get a nice zoom and see if it's carrying a baby. Wow, the edge of the bell is
Pointed.
all bunched up and white. Oh, nice. So it looks like a medusa, but it's actually a tunicate,
Oh, really? That's cool.
a doliolid. The name Pseudusa because it's a pseudo medusa kind of a
Right.
shape, right? And they sometimes have buds of little babies sitting on top of that bell, but it looks like this one doesn't.
Yeah, I don't see anything that looks like that.
That orange thing in there is the gut. Wow, look, even the pleating around the edge of the bell makes it look like it's tentilla. It's like, don't eat me, I'm a venomous jellyfish.
So stingy.
Yeah, but it's actually
Very
like
cool.
not at all, right?
Great video.
Mimicry.
All right. We're good with this?
Oh, it's upside down.
How come it's upside down? Good question. Maybe because it's heavy at the bottom? I think it kind of waits there for marine snow to fall in it as if it's like a sediment
That
trap.
would be very clever.
Yeah.
That would be very
And then
clever.
when it can catch something that's bigger, it'll eat
Yeah.
it. Otherwise, it just
Yeah.
kind of
Yeah.
What a
munches.
great
Not
use
sam-
of a bell.
Lobate's
You're
good
not
too.
sampling very much volume that way.
Hmm.
Yeah,
True.
Your
you
most
know.
success would be from fast falling particles, of which there aren't that many.
Yeah.
You really hit the jackpot, and then a larvation sinker just falls into your mouth.
Yeah, or like one of those shrimp that was just swimming down. It goes,
Yeah. All right.
You guys
Free
good?
here.
Yep, thank
Okay.
you.
Good to go.
Thank you,
Well, so
team.
you're gonna do at least you don't have to move.
We could keep
Yeah.
moving up and
right? It's a
slow.
trade-off
What are you gonna
of,
do with
See what
that
other
energy?
exciting organisms
Yeah.
we're coming
Sit
across
there.
today.
something cool? What? Trouble.
Oh, yeah.
Okay.
Of course.
Trouble or something cool? Either two
It's got to
things.
be one or two things.
I came to see, I'm imaging right now, animal number 0.
A little Leslie?
Oh, yeah, DO059.
Yeah.
I need to look in my notebook to know what that is.
I
Yeah.
need to know the ID
Middle
Okay.
right here.
because it's super happy and I got
You know what's
some
weird? It doesn't
incredible
look as noisy as it did
footage.
yesterday.
Oh,
Even at the same
is it the one with
ionization.
the little
Yeah.
palpons?
There's a very clear wave that
We
propagates.
went
Well,
down to
yesterday was
160
a
yesterday, and it still looked
hard-
It's an
noisy.
It's a hard
apolemia.
day
Yeah.
for the SciCamp.
I was
It
blaming
is an
the-
apolemia?
Yeah.
Or what
Yep,
you're
you're
seeing
good.
is
Oh.
apolemia. Wait.
Yeah,
It does.
whenever
Oh,
you're
what
ready.
is this?
Whenever you're
What
ready,
is this?
Jelly
Is it
experts.
jelly
I've got
or
one
it's
for you.
just a- Oh, it's a
Can
jelly.
we
It's a jelly.
zoom in on that? I think it looks
Yeah.
like
We're working on that, my friend.
Does it have a little nipple on the top?
I don't
Probably.
know.
Is it like
Is that a ruby? No.
Colobenema apicatum, maybe? Let's get a bit closer and find out.
It's a nice spin for us.
It could be a young Patacogon. We'd have to check out the shape of the
Siphonophore
muscle-free zone as we look down on the apex. It's definitely a repellent nematode.
Maybe we have more lights on. Could be.
It's something with the lights.
But
It
then
is-
it would have been just dark, too.
Okay, great job. Let's zoom in if we can. You want the bell or do you want the top?
Yeah, if he turns around, I want to look down on the bell from above
Okay.
to see what the muscle-free zone looks like. It
Oh, that's
looks
great.
like it might be Colobenema apicatum, but
See if he'll turn around for us a little bit. It does look pointed.
want to see if the mussel fields there's a transparent circle in the middle, or whether they come up and it's like a star in between each of the radial canals. Here we go. That's good. That's good. And
Nice.
zoom
Pretty small,
in.
right?
It's pretty small.
You're telling me it's smaller than the predation?
How's that, Dougal?
That's
Better?
looking good.
Yeah, I was gonna say, no, you're good.
All right, so
I would like to collect this dude.
Sure.
I'm gonna collect them. I was gonna say I would like to try this with iris. I don't think it is gonna work well.
Right, we can give it a shot. Whatever you want to do.
Yeah, if we think we can keep track of it, otherwise we should just collect it.
Well,
Yeah.
you two
It's
decide.
pretty small.
Iris,
when it does,
or do you
when
think?
it does go, it'll be boom, boom, like this. That's how they swim.
Okay, so you're saying we should pass on?
I think, I don't think we
We
should.
just want to go to collection. It
We
sounds
just
like.
want to go and
Okay.
collect.
Yeah.
It's got a very voluminous bell, so when it does escape, it's going to drop all its tentacles. It's going to change direction suddenly, and it's going to go whoop in one direction and
No
then whoop
pressure,
in a
pilots.
different direction.
No pressure.
Just so you know.
Exactly. Be ready. Be
The
ready.
baseline is failure. You can only deliver like above expectations.
I have a little bit on
Just-
right now.
So much less noise in the image today.
Yeah, this guy with suction should be okay.
Forget about some of the lights.
This is in Mbari's deep sea guide. I think that's where I've seen it before.
I'd only ever seen a line drawing of it before then.
Oh. Yeah. Yeah.
Is he
Got
in?
him.
All right,
Yep.
stop
Awesome.
and move
Number
forward.
2 canister is where it's going.
Moving forward.
Google didn't do all these things you said it would do.
It didn't, no. It was like, yeah, okay.
It's like I saw you struggle in the last 2 attempts of stuff. That was-
Do we have the shadow cam on today? Shadowgraph?
Yes.
Okay. Because I haven't seen anybody
Is
come
it
in.
on?
I haven't.
Still?
I haven't seen anybody fall into this container yet.
It's a bummer we can't tell right now.
Yeah.
The live feed would be cool.
Yeah, to follow him through. Definitely a Rhizarian coming at the
Fleshing
screen
about
while we
in the
wait
shadow
for
cam.
canister 2. Yeah.
Another little micro bit.
Pushing or little micro.
Yeah, little. Haven't
That
seen
wasn't
him.
anything, right? Normally I see the canisters.
Well,
Yeah, I
Shake.
see the canister move, but I didn't see any animal appear.
It's done 20 laps around the shadow
It
camera.
clearly looked like it went in the funnel. Oh, there he is.
Ooh la la.
Okay, give it a second. Yep, second. Okay, you can switch.
which way am I going?
To 3.
Oh, yeah.
Nicely done. Woohoo!
Yes.
Excellent.
That's the first one I've ever caught.
The Trachymedusa thropalia, right?
First one you've ever caught, at least.
Yeah, he looked good.
First one
That's
I'm gonna get my
caught.
hands on under a microscope at least.
Great.
I've only ever seen a photo from someone's video feed.
All right. Let's keep
Nice.
searching.
What
Great
did
stuff.
you think it was?
I think it's Colobonema. I think it's Apicata or Apicatum. I saw it on the deep sea Monterey deep sea guide. Up until then, I'm like, oh, is it real? Is it not? And I said, whoa, wow, looks like the thing. Is it a Coelodendron? No, Siphonophore maybe.
I was
So-
saying, is it? Do you like it? Do you guys? Is it?
I love it.
I don't
Oh,
know.
Siphonophore.
Stand middle.
Stop.
Middle. I always wonder. I'm like, because I know each one has its challenge, right? Because you guys have done a bunch of benthic,
Looks like it has a
but
dark
the midwater is
gut.
pretty cool.
Yeah, this is.
Yes. It's different. It's like
like I was telling somebody, this is normally
That's
just kind
cool.
of like zoning out time because
Right.
Can you turn your red scanning laser on and see if you can get
Yep,
the
so you just
tentacles
to get down
as
to the
they're
spot you need to get
expanded?
to or up.
well, no, we
right.
like to stop.
Right. Yes, Jimbo, Tina 4.
We like to stop, but we like to do videos.
But we are
Right.
not. It's not.
That's not the goal of the
know where
other
it is
trips.
relative
I
to
know
Yeah.
where it is,
Doesn't
yeah,
pay
right.
that much attention, unless we find something really big.
Yeah.
Spectacular.
Of course.
It's probably too high.
This is like kind of a combination of fun and terrifying.
So you guys stop for rhizarians all the time, those single-celled things? They're important. They're important.
They are important?
They are.
Are
All of these are important.
we anywhere near where we would need to be to get a scan with the deep PIV?
Do you want an actual scan or do you want to what do you?
I want an actual scan
We
of
do an
those
actual scan.
tentilla while it's in fishing position before it
That's all I
decides to
do.
haul them in.
Yeah. DPIV is what we're hearing, yes?
Yep.
Deep PIV.
Yep. Okay. Working on it.
The P stands for please.
Absolutely. It's a very polite way to name a piece of equipment. Here he
I think he
is.
might be pulling his tentacles in.
Did he pull in? I can't see his tentacles from here.
I think he might have pulled them in.
Start turning some.
Doing quite the somersaults.
Mm.
Here he comes. His tentacles are still out. Maybe.
It's turning around a lot.
One's
isn't it?
out. One's out for
Coming
sure.
through, coming through. Yep.
Yeah.
Backing up.
There's much there. All
One
I'm
tentacle
gonna
is
zoom
still
in
out.
about 1,000,000 times.
You want to do that again?
Yeah, now
Oh,
I'm
no,
like
we might
super
lose
zoomed.
him, mightn't we? Would?
You're gonna zoom?
I'm quite zoomed in now.
Okay. Let's try it again. Okay. Yeah.
Who did I do that to yesterday? Leo? I zoomed in so far that like we kept getting good scans except for not in the camera view.
Yeah.
It's pretty close. It's a little far out. Now he's down low. Coming back. He's in the field, moving
About good, probably.
almost the right
Yeah,
direction.
There comes.
He's up on top
Promising.
now.
Might be a little bit too far
Yeah,
to the right.
right. A little to the left if possible. Yeah. Like in there somewhere.
Overshot. Yeah. Wow.
Yeah.
Sorry.
He's
Wow.
tiny though.
He's tiny.
And I think he has pulled in his tentacles now.
Yeah.
He's about the width
He had
of the
one out in the
laser.
beginning, and now we don't have any. Slower.
Yeah, I got a little blob there.
Slower.
All right. And so
Yeah.
after we do this, we'll get him a little bit away from the ROV again, and then zoom in nicely for
He's
some
down
4K
low.
I think we should probably
video.
just do that
Down
now.
low.
We should do that now. Yeah.
Let's
Okay.
stop the scan.
Okay. Camera is done, but you want to get
I
another
want
close up?
Close up, yeah. 4K
Yeah.
zoom.
We can go back to a zoom view when you guys are good. There he is. Middle.
You want some zoom on it, go then?
Yeah.
We do.
You want to look at the
please.
tentacles
I
that are
want it
retracted,
from-
I'm assuming?
Yeah, I want it from all angles. I want him to turn you. I want you to turn him
I'd
around
like
for
him
me.
to extend all tentacles. The two tentacles extended
Nice and
slowly.
zoomed in. Yep.
Okay, coming in. Nice.
Not too far.
Good. Dougal, closer.
Closer, yes.
Yep. There's a tentacle coming out.
Yeah, still on it. Oh, I saw the radial canal. The
Stand by.
radial canals.
Sorry, guys. Stand by.
That's a good shot.
Okay.
Switching some more.
Shoot a turn. Turn around. That's it. That's it. That's it. That's it. Yeah.
There we go.
Just keep turning. Just keep turning. And a shot from above. It's slightly flattened.
You guys are good. You guys are good. I know. It's all over the place.
It's got ridges in between the comb rows.
Okay. Coming back to the middle tubal.
Oh yeah.
Okay.
Focus,
Oh,
focus,
oh,
focus.
that would be fabulous. How's that, Dougal?
Okay, that's looking good.
Good.
This
Okay.
is good. This is good. This is good. Yes, keep rotating slowly. That's good. Oh yeah, all different angles. Okay.
You
We
good?
slurp him up, please.
You would like to slurp him up?
I would like to
Yes.
slurp him up.
Let's
nice
give it a
and
shot, my
slow.
friends. We are in canister number 3. He's out here.
I'm not sure quite what to call him.
Okay, here is right here.
Sidipidoris.
Mhm.
See it?
I mean, I get
No,
Wait,
it's behind.
oh.
Just on the outside.
missed
Gray is
him.
just the outside. There's Siphonophore
Yep. Yeah. Yeah, nicely done.
Alright, stop
Do, do,
suction.
do.
Stop function.
The littlest Siphonophore. Oh, he's in. He's in. Hold on a
Awesome.
second. Hold on. He just went back up. Hold on. He went back out. He's up. Okay, now you can turn it. Turn it. Turn it.
Section sample 3.
Thank you, section 3. Section 3. Nicely done.
What are we gonna call him?
Hi, sunshine.
How's it going here?
Good. We
Brown-
made a whopping.
Brown gut
75
tensid, I
meters
guess,
or
for
so,
now.
100
Could be a
meters, but there's some
Bathytina,
stuff,
but
That's alright, I went a negative 150, so.
If it takes us a little time, we're okay. Yeah, we're doing good.
brown.
Yep, we have an
Not brown,
oikoplurid,
but
we have a
brown gut.
rhizarian.
Got an oikoplurid.
We did with the house.
Nice.
There's an unidentified shrimp we're calling Loretta.
Sweet.
We have a roparia and we have a Siphonophore.
Cool.
That's our. We've been unsuccessful in a couple of the crustaceans we have attempted, but
And a big squid.
The squid.
oh yeah, the
Couldn't-
big squid.
Couldn't get
Oh,
that one either.
huge.
The one that photobombed us?
Yeah, the photobombed big one. I just need to look at the video to see which one he is.
And we
He went
chased
by
an
off the
eel.
pass.
Yeah, he was there. He was creepy slow in the beginning, and then he decided to.
Oh wait, there's another one?
Uh-huh.
Not the one we saw when we were looking at something else in Gerald.
No, there was
No,
another one out there.
no, like big.
Oh, my
pretty
goodness.
good.
Like he was gorgeous, like a deep dark red.
He was definitely in this one and then like skirted
Rise
through that.
area.
Yeah.
No, that's the forearm, right?
4M over on the right. So yeah,
Waiting for
there
that
he is.
good lariat shot.
We'll see.
That might be a little bit higher up.
We'll
Mm.
see.
This is the guy for good for the lariats again. I can feel it.
Okay. I like you manifesting
Ooh, long
it.
thread.
Do it.
Oh,
Yep,
that's
we're
pretty
slowing.
long.
We're slowing.
That's really long.
Might get a little swirly here. Sorry, guys.
Yep, we're okay. If you want to go up, isn't he up? you
I'm
think?
not
I saw
sure.
something Down.
I'm
You
not
think
even
he's-
sure that's a Siphonophore. What on earth is
Okay,
that?
Oh
don't.
man.
Yep, yep. Maybe.
Go up to the right,
yeah, I would
maybe.
go up
yeah.
to the right. That's what I would do if possible.
Could it
this
be an
does
ink-
look like ink.
Is it an ink
actually.
trail?
This is ink.
Oh yeah.
Evidence, perhaps, of another. Oh.
All the way up the
Oh,
top.
yeah.
Where's the squid on the
Where's
other
the
end?
squid?
That was
Yeah.
squid for sure.
Must be a very
That
long
was squid
piece of
ink,
squid.
my friends. That was a lovely amount of squid ink, meaning a medium-sized to larger animal. Maybe he'll come back into frame in a minute.
You have to go
Check us
down
out.
there.
Mm-hmm. I encourage him to come check us out.
No.
Cyclothone.
Yeah, I think it was a cyclothone.
They really don't care if they're like back.
No, they don't care
upwards
what
and-
position they're
Yeah,
in.
they're just like
Not at all.
whoa.
Uh-huh. Going with the flow.
What's the speed?
Good.
This is good, yeah.
Yeah.
Great.
Color looks good. Speed is good.
Absolutely awesome, in fact.
Perfect.
Perfect.
That, I like
Dark
that
zone.
answer.
Down in the comments and find where that big squid was. I just want the timestamp so I can go back in the video. Begin.
Cyclophones here and there, but
Oop,
and
there he is.
copepods.
What's this one, right? 18, 26. Don't move. 18, 6. Thank you. I got it. We're up 895 meters. We're about where our target depth was for today. I think
Barely
this is a
recovered
win.
from a
Barely
shrimp.
recovered from that shrimp.
I'm calling it a win.
In the middle,
Middle?
down, just going off the screen. Don't worry, it's
Mm.
cool. We keep going as we are.
Are we good?
Yeah, we're good. We're good.
Okay. All right. Thanks.
These mugs are a little bit too good. I keep burning my tongue.
Yeah.
Keeps everything too hot.
Russ has been putting his in front of the fan on my microscope light to keep it warm. So it's like it stays warm forever.
Oh, wow. At some point, you gotta just drink it.
Yeah, yeah, right.
And then you just get new.
Good pod.
Yeah, copepod. Yeah.
In the middle of the screen.
Low, middle.
So yeah. Is it a fish? I think, kind of flashing silver.
Let's check it out.
Really flashy stuff.
Definitely flashing. What is
It's
this?
a beating tail. Yep. The head's
Yep.
up.
Yep. Agreed.
Fins are splayed.
Worm? Fish?
Fish, I think. I'm saying bathylagged, but let's have a closer look.
Southern.
Yep. Working on zoom in.
Oh, he's little and
Oh, it's a
stumpy,
fish.
isn't he?
All right. I'll go with your fish. It's not a cyclothony.
Nice and slow. Look at that. He's just treading water.
Yeah.
Not going up
Wow.
or down. Does he have
Poor
barrel
guy.
eyes?
He
Poor
might,
guy
if we
needs a
could
swim
zoom in on
bladder.
his head. I mean, the whole thing, obviously. Yeah, just go slow. Yep. Creep up. Stealthy.
He's using a lot of energy.
Yeah, I want his.
Oh, wow.
Don't you don't.
Oh, that's better. Actually, I can just float.
There.
Oh, yeah.
Wow, look, it swims like a shark. See that?
Yeah.
I know it's not a shark, but
No,
the way
right.
it
I see what you mean.
swims.
Something close to Revettus.
Ooh.
Don't do it, dude. Hang out with us just a little longer.
Where's Marcelo?
I would say, I think Marcelo finally might be sleeping, or is he in the room?
I
No.
said he was swimming like a shark.
Is it like Ruvetus or something similar? No.
Silly little guy.
Yeah,
Argentinoid?
but I don't think it is.
It's
No.
not. The mouth is
Yeah,
not
the
big
mouth
enough.
isn't right.
Argentinoidea?
Like the meanest
No?
fish I've
Right.
ever seen.
Those would be.
I don't recognize it at all.
I don't know
These
this
are
one.
also huge.
Right.
Oh, it
I can't even get
turns
you to
just
the
as
family.
eye
Look,
is
he opened
getting-
his mouth. Oh, no, he didn't. That was his big old eyeball.
Oh, look at the ease of the
I
fin.
know
These are big, like, I know.
Oh, that is
Fins,
Marcelo.
though.
Marcelo's in the room.
Why is this such a struggle to go up?
He is tiny. This
Like,
is a fabulous
spread his
zoom, you
fins
guys.
and be kind
Thank
of
you.
a-
That's
Let's
a
just
great
really
shot.
negative
See if I can
buoyant.
pick.
Which is a bummer.
Yep, over left, or over right bottom.
Wait a little, he's out.
I don't know.
Maybe, we can try.
Did you bring a fishing pole?
Right. Agreed.
I'll back in again.
Yep, that'd be great.
Get some more video. I think we can get video without it
Big
escaping.
fins though. Looks like a baby.
He's zooming away from us. Come on back.
Someone give me a name. Anyone on the chat? Is it?
They're just asking if it's a McTow fish.
I don't.
No,
It
it's not a
doesn't feel like a
McTophid.
McTophid. It's not
Definitely
the right body
not.
shape. Here he is.
Scopella.
Bonius.
It's a. I really want to be a flying fish. I'm an aspirational
Well, his
flying
fins are
fish.
huge.
Deep sea flying fish. Yeah.
Yeah. Challenged by living in depth.
Challenged by not having an air-water interface to practice jumping through, but it's got those fins going.
Oceanographer in Tennessee.
Like
We
a
should know this.
marine biologist
I
in
feel like I should
Fairbanks,
know this.
Alaska.
Those fins. It's a juvenile somebody, right, with those fins that big?
I think so.
I'm gonna text someone.
Google images.
I know
Call
a guy.
friend.
I know a guy. My pictures are not fabulous.
Do we
but
have
I know a
enough
guy.
video to suck him up yet, or?
We can try. I don't know. I don't know how he'll do.
Backing up. Ooh. Yep, now you wanna.
Tell me what you guys would like to do. We have 5 canisters. We have 5 canisters available.
Yep, let's catch a fish for the
All right,
fishy
let's do
people.
it.
Oh, where's he going? He's going
There he
inside.
is. Whoa, whoa, whoa. Oh no.
Right at the last second.
Did we lose him? What's coming out?
If we
What's
get
that
if
long?
we get like a tail or a piece of
Well,
something coming in,
There's something coming.
I think he's gone. Yeah, and that
What's
looks like
coming
an,
into frame though,
that's
my
an
friend?
apolemia coming
Okay.
into frame.
Oh, there he is.
Oh no,
top
it's
middle.
a. All
We still got
right.
him. There's still a chance.
All right, you can get the fish. It's an apolemia, but you can get.
Thank
the
you, Dougal.
fish.
Yeah, absolutely. I think you can. Oh, I think he went up.
He's to the right.
Here he is.
To the right. Here. There you are. There you are. Come on, buddy.
We're on jar 4 now, okay?
Description: 5 new species
Correct.
of Ancestrocephalus.
That's anyways. Down. Just getting swirled around is what's
Could be Ancestrocephalus. That's what Google Lens is.
Here he comes. He's back in mine.
No, that's
Sweating.
Jurassic.
You got this. You got
Oh.
this.
Just
I don't
let
know.
you calm down.
Have a picture of it?
Pull straight in. Just need a little pop top.
Have the parallelopsis.
Yeah.
Oh, I just took.
Yeah,
That goes to the right.
Not very good.
Still better than
I don't think it's a parallel, but it's too short.
Is that worth sending?
Yes.
Oh, that was close.
Let's see if we can get him back.
He can swim when he wants to, can't he? Did you
I
see
was like,
that?
He had some. He's right here, middle.
Alright.
All right, let's try one more time.
He looked like he wasn't that strong a swimmer, but as soon as he's like,
Looks are
I'm about to
deceiving.
die.
Coming straight at us.
He's like, yeah. See you.
He's trying to scare you off now.
He has the- You want this? You want this? You're gonna get all of it. Just go right in the
Just go into the fun.
into the fun.
Oh, there's a bonus cuting there. Oh.
And you've got pretty good suction on?
Yep.
Okay.
I put the image on the group WhatsApp. Random.
Like I will outswim you. He's
Yeah.
back way back here, yep. Yep, out.
Seducer, yeah.
I don't see him. Oh,
Extra
here he
balloon
is.
like this one.
Out of the lights, in and out of the lights.
There's a different fish that just crossed in front.
More fish. We've hit the fish zone.
Yeah, we've got him here. That's not gonna happen, I don't think. He's been pretty active this whole time. Oh. Down in between.
I think
the
he's toying
campfires.
with us.
I think he is too.
He's like, come on,
Nice try,
have a
humans.
try.
Nice
I think
try.
the next thing he's gonna like pretend to like actually swim in there and then just swim.
And then just swim back out.
and then fly
Oh,
away.
I like Howella. What about
There's a
Howella?
house. Hello, Ella.
That one of the chat folks.
Hello, Ella.
Howella.
Oh.
Those are, we catch them in the Gulf. This is 350 miles off Brazil's coast
Do you
in the
release
South
them in
Atlantic.
Brazil?
Not sure if the body's. Oh, so close.
Just stops.
Yep, he did
Like-
stop. Okay,
Psych.
back.
Oh, through the DPIP. Nice shot.
Did you scan him?
Maybe we can herd him in with some lights.
I think this
All right.
is the longest
What do you
chase
think
I've
for the
ever
tactic?
been a part of.
Good luck. You're staying strong.
At some point, he's just gonna stop. though.
Right. Or at one point, we may have to say he wins.
Yeah, tire him out. Just keep him
We
going.
thought we were doing that.
Really, man.
There he is.
Where's that little
Not a
harpoon
big
gun?
scale. It's not a big scale. Fins are wrong.
Where's our non-intrusive sampler?
Good. It should be okay. Try and catch this fish.
It's like the test of the
Fishful
day.
thinking.
Oh,
Uh,
Want this?
All right.
I got him.
Marcelo, can I tell that this will be the last chance?
here he comes in.
Yo.
Okay. Marcelo's like, no, we can use the rest of the dive to collect this
I think
fish."
he's tired. I think
He
he's
is
tired
tired.
himself out.
He
How
is?
about a baby Cubiceps?
A baby what?
Cubiceps?
Cubiceps.
Maybe. Maybe.
No?
I don't know. I don't know well enough. He's like, haha.
Yeah, it looks tired.
He does look a little more tired, but I don't think that's gonna necessarily call that a win for us.
As long as Sebastian's not tired, then it's fine.
Nice and calm.
Oh yes! Oh yeah!
Nicely done. Holy crap.
Suction's still on, and I'm going for
Yeah! He's
Okay,
in. Give
he's
it a
in.
second. Give it a second.
Oh, I'm going.
Yep. Nicely done. Nicely
Wow.
done. Yeah, it's in 4. It's in 4.
Oh, that was
Look at
awesome.
Howella. Did you look at Howella? It's Howella.
So does that mean we have to pull the ROV now? Otherwise we won't get it ready in time.
Sushi
We
all
still-
around.
We have a couple of minutes.
Yeah.
Phew. All right, you good? Nicely done.
that
Nicely
was
done, team.
Excellent.
Nicely done.
Still waiting
All
for that
eyes
fill
were on
area
that
house.
What?
We're still waiting
Trying to
for that
help
fill
out and
area
keep an
house.
eye.
Haven't seen
A few times there, it was like, oh no,
Did you get a good,
just outside.
did you get a good shot of it?
There's beautiful
Beautiful.
video
I know,
shots.
but did you get one? So I can-
Not beautiful. I would not call what I got as a. I did get 2 pics. I sent them to Tracy, but they're very blurry.
I can always go back to YouTube. Don't worry.
I would look at, yeah, let's
Yeah.
find the guide of Howella, because I know it's in that mesopelagic book
it can
of
be,
the
yeah, it
Central
can
and
be.
South Atlantic. Sarah's like, I'm done.
I'm loving it. Nope, I'll stand.
No, look. Oh, I like this. Did you look at it?
I'm having a blast.
Look at the fins.
Yes.
And yesterday it was
Extremely long.
And the, yeah,
I'm watching on YouTube.
yep, excellent. And I'm looking at the images from various, look at worms, the various spots. That might be it. Thank you. Whoever suggested that in the chat. I can't see who that was now.
Which one? The holiday?
Howella, maybe. We're gonna let the fish expert clarify that when we get a chance to look at them later.
Still swimming in the jug.
Yeah, he's gonna be pretty active. I'm quite confident. He didn't stop swimming once.
very
Down
lucky
middle?
day.
Sorry.
Oh,
Oh,
you're
no,
good.
we're okay.
We're good?
It looked like a cyclophonia. I think we're good. Yeah.
Got lucky that he stayed in our
Uh-huh.
This.
View.
Yeah.
Right. Mm-hmm.
for as long as he did, because
Right. He could have ditched. Top left. This guy up here, it's Medusa of some kind.
Jelly.
Yep, we can get a good zoom. Kakani, any group you want to put on this if you know?
No, where's Dougal when you need him?
I know. Dougal has walked out of the room.
Swims like Octopodina.
Okay. I'm hearing a swims like an octopodema.
Thank you guys.
Octopodema?
Oh, that's what. Nicely done, team.
that was excellent.
Fabulous.
That's what we want. That's what we want.
We want one of these guys, yeah?
We do want one of these guys.
Yep.
Did you? I saw repellent nematodes, but I didn't see an octopodema yet.
Up to the left.
Suction?
Yeah. Do you want video
Yep.
first, Dougal?
I think we got enough video. These are really hard
so
to
yes,
see the.
suction sampler, please. You did not
Do
get
it.
much of a break,
I love it.
but he is a different swimming type of structure to him. So
Are
we'll
we
see.
gonna film it first, or
He said he's
We
got
just
film of
rated.
him
We just
already.
got some, yeah.
you got some film?
Super tiny.
Enough for me to know what it was from the monitor and come running in here. So I think that's probably okay.
That's your we have fun. Going into number 5.
See how big it is.
Nicely done.
Oh.
Nicely
Oh yeah.
done first
Nice
try.
one.
No.
Suction off.
Yep, moving
There
through.
we go.
No, I don't think we have it.
Yeah.
No, we do. We do.
We got it.
Much better than the fish.
It's gonna take a minute.
Corner.
I think it went in on you. You could see it. In the,
It was sucked in.
at least it's shaped form.
Sampled in which one?
Keep an eye.
5.
Yep, I'm watching
Side.
canister. Oh, keep an eye on the side too.
Keep an eye on the side.
I think it went in.
My feeling is it hit the side.
Yeah, it keeps changing. People are making schedules after I make a schedule, so I don't know who to follow, which I don't care, as long as you guys are on the same page.
I think Lily can go.
She's probably working.
She's
Yeah.
doing the medical.
Yep. Yep. Thanks, you guys. Yeah, however it switches,
Yep.
it's all good as long as everybody's good with what they're doing. I haven't seen him go in yet.
Side and flip.
Do you want to give a tiny little pulse?
There it is.
There he is.
Oh.
There he is.
Alright.
Yep, you can switch canisters.
Alright.
Thank you. Nicely done.
Right.
Woo-hoo.
All right.
That is very very small.
I don't need anything else for the rest of the ride now.
Okay. Noted. Noted when you claim
I'm
you
good.
Yeah.
I will.
Did I just say that?
Take a
Wait a
video.
minute.
How much
Take a
time
video.
is left in this dive? Let's see how
Yeah, exactly.
generous.
We have 3 canisters left, and
I'm-
we're at 867 meters.
I better
Just
step
as a
out of
heads
the room,
up.
right?
Yeah, I think best is to be out.
Sorry, sorry. I'll move over this
I think we need to do a lot more
They
scanning,
go. Yeah.
right?
Do you want to go up to a depth, or should we just
Activity
do this way?
again.
I'm up for options here.
I don't know. I feel like the last 2 stations we started to see bigger larvations.
So it doesn't matter as long
Yeah,
as
she asked me to
Yeah.
change with her in
Okay,
the
perfect.
in the second
the second dive.
dive.
Yeah, perfect.
And I'm say yes, but I don't know why she make another.
I'm not sure.
This is sleeping.
All good?
There you go.
All good.
So,
Yeah, no, it's good. As long as we can all cover for each other, it's perfect. Thank you, Julieta.
Straight ahead.
Yep, straight ahead. Oh, are we
Slow
stopping?
down.
We're not.
Yes.
You want to? Yep.
Please.
Yep, yep.
Yes.
Sorry. Yep, we're getting there, Kakani.
I think. Oh, where's Karen?
Now we're going left or
We're gonna go down a little bit because
Just to see about
think
this
Is
house.
a larvation.
Yep, I just don't know
house.
if he's hound. This looks
One
like
of
a
the big
sinker to me,
ones.
but let's take a look if there's a
Can I-
larvation inside.
Yeah, if we can zoom in.
Yep, perfect. Perfect, thank you.
Yeah, if we can, whenever you're comfortable.
Do you want just the house, or do you want if he
I-
has a larvation in
I
it?
would like to zoom in and see if there is
Yep.
an animal anywhere,
Yep, yep.
but I think
It's
that
not that
opportunity is probably
not that included.
gone.
Yep, I don't. We're breaking up the house. That would be
we're
it,
not
it's him.
inside.
Yeah.
I think I
Uh-uh.
see.
Oh,
No, it's something
no.
else in there.
no, I think it was a sinker. Like, I think he had left, maybe?
Maybe. Well,
Okay.
that means we're below them, which is good.
Yep, all right. Headed back up to 700, please.
Heather, another guess. Epigonid.
Wait, a what agonid?
Epigonid?
Epigonid for the fish.
You spell it?
E-P-I-G-O-D.
Oh, I don't know that. What's a species in that group?
Epigonus. Brinkmanella. Brynthostoma.
Okay. Maybe.
Floresiella. Microictis. Rosenblattia. Spheranopsis.
I like Howella better so far based on Marcos's picture.
Equaga. Epigon.
Sem H.
Só E.
Epigonus.
Mm-mm.
Epigonas.
Mm-hmm. Mm-hmm.
Heather, are we gonna stop on anything big?
If
going
you
up,
want to, sure.
Or do you want to just go to 7?
We had said go to 700 unless there was something super.
Okay.
So if you claim there's something super,
That is such a great
is he deep sea?
Epigon.
descriptor.
Maybe.
Yeah.
Maybe. Epigonus.
They
Well,
have
yeah.
Yeah.
So
Basically, we're
Look
not
at the-
stopping unless anybody wants to stop.
Exactly. I would look at. Yeah. At least you guys have a couple of options in there
Yeah.
too. That's great.
And we have the fish.
Yeah,
That's the most
that's what I'm
important.
saying.
Yeah.
Exactly. That's the best.
Don't say anything.
Nope. Nope.
Oh,
There's a lot right here.
There's another one of those.
Yep.
Rosaria. There you go.
Yep. Some rhizarians. We're in a rhizarian layer. We're gonna go up to 700
Well, at some point.
and
the
slow down at
rhizarians,
that time.
Yeah.
I see a question in the chat about what happens to our samples when they get up to our pressure environment. Dougal, is there a big pressure issue with your animals for the gelatinous organisms?
Siphonophores, which have a float filled with carbon monoxide, often the float will rupture as the gas expands, as pressure decreases. The other jellyfish, it's more of a problem with the temperatures getting warmer, is pretty lethal for a lot of the species. So it's just the Siphonophores for me. Fish as well, obviously if they have a swim bladder, they don't like it. But even if they don't, the gases in their gastric tract or their stomach or their intestines can expand, and they can die from that as well sometimes, correct?
Correct.
Yeah.
Yep,
I guess shrimp as well, if it happens to.
Yeah, some can adapt. As again, some of them are these vertical migrators, so they are adapted to a wide range in pressure, and others aren't. So it's very species specific about how they deal with the pressure changes as they move up in the water column.
A little bit chenophobe. Frilligoma.
We're not stopping.
I know. Just calling them out.
I like it. Call as much as you'd want. It's fine.
Oh, we really haven't done much scanning.
No.
There hasn't been.
They've all been
There's
pretty
not been a lot of
small.
jellies, or they've been super small.
There haven't been many jellies, and they've been really small.
Right. Yeah, very small.
We've only seen one really large Siphonophore so far. The one that appeared at the same time as the fish that we just kind of went past and went after the fish.
Your
That one.
priorities.
We're at 750 meters. We're gonna go up to 700 and then hang there for a bit to search out some more animals.
700's Frilligelma land, but you've got Frilligelma, right?
I think so. I was making my way through the second dive of data and pleasantly surprised by the number of scans that
Oh,
we
good.
had even on the first day. So
Yeah. First few days, you were like,
cranking.
yeah,
We were cranking.
that's why there was
It's fine. That's training. Because of that commitment of time, though, I think it's been helpful because now it doesn't take very long.
Be nice to get a few. I guess you already have Thalassocalis and Lyria and like the main big.
The ones I looked at for those, I hear people say Thalassocalis, so that's why I'm gonna pronounce it that way. But the animal, like the body, is just so deformed, right?
Yeah, yeah.
Because so I haven't been that happy with the scans I've looked at so far.
Right.
But,
We did a Sudusa earlier, didn't we?
I vaguely remember that, maybe the second or third dive.
Because some of these guys are so reflective, you can see with just the lights off the ROV. The Lyria, for example, and Thalassocalis, both of them have this bright white spot just where the reflection from the ROV is coming back to the camera at that perfect angle.
Yeah.
Right. Dun dun dun.
Doesn't look like jelly.
Team, I'm very impressed with your restraint. We have made it to 700 meters. Nicely
is my
done.
middle name.
We sacrificed for you.
Dougal Restraint
Not for
Lindsay.
me.
That's
It's for all of us. Dougal restraint what?
Yeah.
Uh-huh. That's it. All right. Slow and steady scan, my friends.
Dougal being restrained Lindsay, maybe.
Yeah.
Yeah, no. Yeah.
By someone else. Yeah. Straight
Had to
jacket.
cut you off. All right.
Right up
Let's
there.
find something
Scannable.
big and amazing.
Right up there next to Manu restrained.
I thought yesterday was so funny when we all had a different interpretation of what we were seeing with that mysid and
Oh yeah.
there's a squid.
Cyclophone. Where's the squid?
No, that's a cyclophone.
Yeah.
Don't tease me like that, Connie. I was like, wait a second. That could be a Tomopteris or a fish.
Oh, we haven't seen a Tomopteris.
We
yet,
haven't
have
seen
we?
a Tomopteris today. This is a fish, I think.
Yeah. Oh.
Is he another one of the
Oh,
same
it's a
guy?
different fish.
Little
That's a different one.
Can we zoom in, please?
Zoom in. Oh, it looks to be the same.
He's got.
doesn't
It's the same. This is this. Well, no, he's different.
different
No.
species, but
No.
looks really
Probably it's the light.
similar.
Maybe.
The coloration.
Okay, if we stay on him for a minute.
Those bright white patches.
Those are
on the
photophore
ventral-
patches, aren't they?
Yes.
Yeah, but they're not single.
But he's not right.
Photophores,
It's
right?
it's like patches.
What's that white thing on his dorsal? Is that a
Look at that.
copepod?
That is cool. These
But
are.
with the another
wanting to
fish.
mate with us.
They look like They look like photophore patches.
Yes.
Photophores are bioluminescent organs
Was
that
it
these
the book in his
animals,
eye?
some animals
Yes.
have. And this guy most likely uses it, what they
Probably.
use for counter illumination. So he's dark coloration on the top, and he uses that bioluminescence on the bottom to blend in. So he's harder for predators to find from below. Very cool fish. I don't know this fish.
Get him to poke his tongue out and see if it's bioluminescent.
I don't. Yeah, let's get some good video on this. I'm not sure. We're not
Keeping that.
collecting. We only have 3 canisters
that.
left.
I mean, it was the keeping bath wasn't.
Yeah,
Different species, same
it is different
Male or
species.
female?
Yeah.
What is it?
Is that a parasitic copepod on its dorsal side? A
Wait.
bright white
É
thing?
bom é melhor quando ele vira de barriga pra cá.
Could be.
Can't
Olha lá, tá vendo?
see very well. Could zoom in a little.
Marcelo, any ideas?
Can I put that in the same?
No.
I think the
There's
other guy.
another
You want to try to put him in the same container you just put your other fish in?
Yes, can I do that?
I don't.
I would-
Well,
I probably.
the other guy may swim out.
Can we zoom in
Oh.
to see if that's a parasitic copepod on the
On his
fin?
head.
On
The dorsal.
the
Head?
dorsal fin?
Oh, the
second
dorsal fin. He's looking to see this.
We'll try, but he's quite small and he's moving around. a lot.
Yeah, yeah.
I don't think you're gonna get a whole lot of zoom success with it.
Yep. We'll give it a
It does look
shot.
to be sticking out.
Yeah, it doesn't look
Yeah.
to be.
Yeah, you can do it.
We may have a pinnellid copepod.
It might be a copepod on there.
Yeah.
In the dorsal.
Yeah, on the back of the dorsal, this big white patch.
And we should
Could be a
definitely get it.
parasitic copepod. There's lots of copepods and different arthropod species that are acting as parasites on some of these deep. No, I don't know.
It's not dragon.
I don't.
It doesn't have a egg sacs dragon behind it, for example.
Yep.
Poor thing. Saying all the time.
Yeah.
Yep. Okay.
Yep.
So you want to index it back to the
The
other
only
fish
the
and-
only question mark is you might lose the first fish and you might not get the second.
Keep
Right, I
the suction
understand.
on.
Right. We might lose
We
it.
can keep the
they
suction
can
on. Yeah, keep the suction on the whole
You
time.
got.
Yeah.
I'm good. If you would, are we gonna? We won't mess up anything because it's back to canisters. We have to go back to canister.
We got to go past the Octopodema to get to your fish, man. Okay. I'll
We
do it
need
for you.
canister 4. If we can index to canister 4 without losing anything on the way.
Please.
We're gonna try this, Marcelo. Yeah.
Sure, let's try.
Okay.
Here. Because you got.
I'm just gonna watch him for now.
Yep. You keep watching because I was not.
How many things do you have now?
Okay. We're at canister 4. Your fish is currently at the bottom, and we're gonna suction this guy into the same container.
One cyclophone A, one eel, one.
We're gonna need suction as much as possible. He's on the other side.
Two from the last day.
You think he's in?
I'm just thinking in terms of like.
Did he go in?
She thinks he went in.
Really?
I couldn't see if he went backwards behind it or in.
There's only one.
Yep, so far. Oh no, he's in. He's in. Close. Oh, they might yell. There might be a predation event. Yeah, we have to go to 5. We have to go to 6.
It was tiny.
Yeah,
Maybe.
6.
Are they mating now?
There might be a
Are they mating?
Or
might be a predation
it might
event.
be a predation event.
Well, I can open it. We will recover it.
Thank you guys. Nice job.
You got a
Nice
scalpel,
job.
right?
Yeah.
And one
Was it that
more.
much smaller?
It was
That guy was tiny.
small.
Yeah, he was tiny.
Wow.
Okay. Let's do it. That was a great catch.
So,
Pilots are in
Can
for
you
some
turn-
champagne today.
Just fill that bucket of fish, okay?
That is Marcella's one fishing bucket.
Okay, blue shark next.
That
Another
would be
fish.
another fish. Looks cyclothonia,
It's a
so
cyclothonia,
yeah.
They're not really fish.
I think
they
we're
just
okay.
pretend
Yeah,
to
we're
be.
good.
So
Thank
To
you.
the right. To the right.
To the right. This guy right up
Starvation
here.
house. Possible
Thank you.
scan.
Stay off, stay back for a little
Yep.
bit.
Yep.
Okay.
Don't get too close, so we don't want to break up the house.
Looks like a well, it's an Oikopluridae.
Over. So, have we collected one already for Russ?
We did
collected
with the
one
very
before.
first one we collected with the house.
Was it a clubby one or?
It was similar to this, I
regular
think.
one,
Okay, could we zoom in now?
This looks like a clubby one, doesn't it?
Yeah, it does look clubby.
Yeah, I'm gonna try and
Oh,
stay
we've
away
already
a little
filled the
bit.
D
Let you
sample and
just.
stuff, which is-
Okay, he is home.
Wait, why can't we just put it in?
Okay.
Sampler?
Do you want to DPIV this?
Yeah, we'll scan it.
We do.
And then could we attempt to collect it with a suction sampler?
You're okay with a suction sampler?
Yeah.
Okay. It's gonna break up.
Well, I'm not. Yeah, the house will break up. The animals should be fine.
Okay. Yep. Yep.
So
Let's do it.
I will get.
Be suction 6 if we get there. Let's DPIV it first, right?
Yeah.
Yeah, because this guy will break up and the animal might leave. So there's not a lot of room to play here.
Yeah. So Sarah will want to try and do maybe one scan.
Just one shot.
No pressure, my friend. There's a lobate ctenophore on the other side.
Who's on that?
Yeah, I don't see him.
Only gonna keep.
One shot Sam.
There you go.
I don't see him.
Need those outers on.
Opportunity comes.
Too much.
I've lost it.
Yeah, I had lost him as well. Stand by.
Well, do we want to move
That
a
is
little bit forward?
Yeah.
Can find another. Yeah, there should be plenty of them.
I think I saw
Yeah,
the second
we're in a
one right
good
behind
zone.
it.
We're at 699 meters.
They're all hanging out at 697 now.
It was like
They
nicely
tend to do
lined
that.
up.
All right. I don't know, you guys. This may have been lost.
Yeah.
We'll probably
They
keep
find
the
it.
All right.
outers on until the end.
Okay.
Yeah.
We can go back to scan.
Yeah.
Or
Let's
upper.
drive through
Yeah.
that thing, that protist that's about to go through
We're
the
having to keep
laser.
those far away.
Here we go.
All right.
We can
Oh,
go
it's not a protist, is it?
back to
It's
scanning
a
mode.
Siphonophore.
Where are you looking, Dougal?
It was under the laser. Just
Oh,
Oh, there is a safe there.
there was
another
a-
side for praying.
Straight ahead.
straight ahead.
What is that? Is that a jelly?
It's a jelly, it's a halicreatid. Is it botulinum? Oh, halicreus maybe?
Let's get some
Let's
zoom
take a
and we can let
zoom.
you know, Dougal. We'll get you some detail.
This would be a nice PIV scan if it is a halicreus. Just because they got such
There's a
cool
cyclothonia in
shape
here.
on them. Yeah, it is. It's a halicreus.
Yep. Halicreus is a which category again, Dougal?
Trachymedusa, halicreatidae. Yeah, can you scan him?
Yep.
Well, it's got the
Yeah.
tentacles out like
I
that if
can.
possible.
Yep.
I can. How
We're
big
gonna
is it?
DPIV him.
Should I be zoomed all the way out or? Sorry, give me a second. I have to get the laser on.
Yep.
Okay, lasers on.
Okay, lasers are on. DPIV when everybody's ready. Connie, you're good?
Yep.
Okay.
Full halicreus.
Yep.
I might zoom in just a little bit before you get started. Oh, a little bit.
It's a little to the right.
A little bit low it was.
Up above.
Oh,
Oh, wait, this guy creepy.
Siphonophore.
That's Ctenophore.
Oh, that was a ctenophore? Oh, that was another.
Tetrophilia, maybe.
Yeah.
Yeah.
Sorry about that.
It looked like a Siphonophore until you could see the comb rows with the
Yeah, now he's swimming.
rainbow colors.
I'm losing him. I've got him.
Here.
Yep, I see him now. Oh, he's in a nice spot if he would be agreeable. He's moving a lot though.
He's swimming, so I guess he's probably not so great.
And down a little bit.
Gotta wait till he stops swimming.
Yeah.
Going down out of my screen.
Might be better looking for the next one.
Yeah, maybe. We
He's
might want to.
down off my screen still.
Because once they
I
start
see,
swimming,
you've
I
got
haven't
him.
seen them
Yep.
stop. Proteus and piece of Siphonophore, yeah.
I don't have him. Oh, there he is. Now he's back. Still swimming though.
Yeah, we might want to go find another target.
Yeah.
'cause
Okay.
they
You
may-
sure?
Yeah, they might swim for a while.
Once they
Okay.
start swimming, they
They're
never
not
give
gonna
up.
stop?
Yeah, they're
Okay.
pretty.
All right. We will call this a nice attempt and we will move on. Jelly 1, Team Sebastian 0. That's okay though. That's not too bad so far.
Zero, Team
Yes,
Sebastian, 2.
exactly. That's right. Depends on the canister we're looking at.
The cursed canister has been okay today, right?
Don't talk about it. We're on it right now. This is the one we're on. We don't know yet, Tuco. Not sure.
We haven't gone to 6 yet?
We're on 6 right now.
Oh, no.
That's why I was like, dude,
Touch wood,
what
touch wood.
are you saying? Don't say anything, my friend.
You've done.
Exactly. Don't do it.
The canister that must not be named.
Shall we name it Bob?
Is that a tiny little Medusa?
Little.
Do you want to stop or go?
Chucky Medusa. Itty bitty bitty. I think it's an Octobedema.
Okay, we're gonna keep moving.
Sure.
Oh, he didn't say stop quick enough or sounded.
I didn't yell.
Yeah.
Yeah.
There was no
It
yelling.
was like it's fast.
I didn't
it's
hear any
already
yelling,
moving.
so.
Changes direction a lot. A couple of fish in the background there. The Keatingnathe. Another Keatingnathe. Cyclothone top right.
Got something down on the right.
Yeah, to the
Yep.
right.
Oh, to the right? Yeah. Let's
Yeah.
take a look at that.
Looks like a
It's an oink.
location to me.
Try and keep it
To
away.
elevation.
Keep it away.
Alright, yeah. And if we could zoom in to try and do an ID before we get
Zoom from far.
any closer.
Oh, there's many cyclotones.
Yeah.
Yeah. But we're not looking at them right now, okay?
Keep plugging away though, Marcus.
Yeah,
Keep
plugging
plugging
away.
for
Yeah,
him.
We might.
Keep plugging.
Oh, he's just
Oh, he just left.
Even.
He didn't even want a picture
You wanna look at the
taken.
house? There he goes.
Oh, and it was a Nobby too.
So this was a larvation in a house.
Can we look
Got a
at
little
the-
nervous.
Can we look at the house a little bit?
Sure.
Because I'm really curious.
Yep. If we could zoom on the house.
Started far away.
Mm-hmm.
Good.
He was nervous. He's camera shy.
They're so different.
They abandoned the house like.
They did, yeah.
Yeah, he abandoned.
Why?
What?
Probably
They have
sensed our
a lot of
presence.
work to construct this and
He thought we were debt collectors.
They do shed their houses, so it is normal for them to shed a house, but that
they
seemed a
run
little.
away and come back. Or
No, they'll make a new
construct
one.
a new one?
Yeah, they'll construct a new one. There are some larvations that I read about that can construct 8 to 10 houses in a day, like in a 24-hour period. But I don't know what species that was. Google may know.
They keep them folded up underneath their chin, ready to
To
inflate.
glue a new one.
For nets.
And sometimes they get super heavy with particles, so that's maybe why they ditch. This case, we don't know about that, but in
I think
general, we probably scared
did
a little bit.
certainly,
Yes.
Uh-oh.
Anything you want with this Kakani?
A
little
little
bit
more
more zoom.
Sure. I figured. Nice.
That looks so different.
Fleets
Come
are
back.
amazing, aren't they?
He's coming back. He's coming back.
Yeah.
You should, we have a larvation house model online that you can look at.
Cool.
Just
Well,
Google
the edges almost look to be straight on, like,
Yeah.
You know,
I'm kind of just tempted to do a scan anyway.
Want to give it a shot?
Can we do
Yeah.
just one?
Yeah,
one-
yeah.
one go?
DPIV.
It's in good condition.
Let's try
And
it.
it's still inflated.
It is still
so-
inflated. Again, it's kind of a one-shot deal.
Yeah.
Thanks, you guys.
you know, they have the audio push
So yeah.
and like the gold proportion.
Ah,
They
yeah.
have
I'm not sure. Pythagoras, Archimedes proportion was it, or I forget. But
Still inflated, so we're good.
I don't see it in our crossing camera yet.
Nope, not yet. We're getting there.
golden ratio. That's it.
Oh, keep
He
going.
should be. He's right there.
Maybe a little close.
Maybe.
Oh, keep it. Oh no. One more try. When
Yeah.
you're backing the vehicle
You
out.
can go back through.
Oh, too far to the right.
Yep. Stand by. Super slow.
Too far to the right.
Yep. We don't want to use any thruster on this.
Nice area.
Okay.
Little low.
I find talking to it
Self
helps.
talks.
Yeah, I think it helps.
A little low.
Yep, he's lower. And now we need to go left a little.
No, I don't know if they're in front
Maybe,
of the laser
maybe.
or
It's in front.
Yep.
Yep.
He's going over to the right again.
does the
Now
house look
it's-
like
You
it's
see him?
deflated?
He's down here.
Yeah.
Okay. Yep. Yeah, exactly.
One more try before it deflates.
Maybe. Might be in
Oh,
front.
Nope, it's right. It's right.
Yep,
Keep
he's
going
coming, he's coming.
straight. Oh no.
He's too far right. All right, should we try one more time? He still looks
No,
inflated.
the vehicle, just one that reversed. Ah!
Okay.
Oh, yikes,
Everything's
yikes, yikes.
Fine. Fine. We're good.
Look away from that.
We're good, we're good. We're still good. He's inflated.
Yeah, it's still inflated
front?
somehow. I'm impressed.
Or is he behind?
He's between us.
Yeah,
Some
yeah,
sort
yeah.
of valve on it that
Okay,
prevents-
so you can go back through that way. While he's very tolerant of staying inflated for us.
Oh, it's starting to
Oh.
go. I think it's the vehicle wash now.
Yeah.
It's too close.
Yeah, he's in between the field and us. Starting to break.
Yeah.
Yeah.
Is that a fish? or-
Really close.
Just stay. Oh. Darn it.
Button.
Still too close.
Yep.
Let me back. Let me see if I can.
Walk a little bit.
So start breaking up. He's okay. Okay.
Too rotating too much
Take the
now.
smallest little. Well, you're up top now.
He hasn't broken up yet, which is some of the mucus is broken, but the big structure part hasn't.
But it's rotating and deforming, so it
Yeah,
might
it's
be
starting
better
to.
off someone else.
See if
Might
we
be
want to give one more shot.
I mean,
You choose.
Bump back.
I I would say no.
You're okay?
It's starting to
Break up.
Yeah.
Okay.
All right.
In the wash.
Yep. Okay. Thank you guys. We will continue on towards 600, just nice and slow.
Straight ahead, slightly to the left.
What are you looking at there,
Oh,
Dougal?
it's okay, don't worry.
We did not get him in DPIV at the perfect scenario. The house started to break. Hey, it's all good. When you're playing with mucus in the middle of the ocean.
When you're playing with mucus.
You gotta expect you're not gonna get
Watch out, when
everyone.
you're playing with mucus, it's not all pretty.
Yep. Can never count on it.
Another one to the right, but it was really small.
Really small.
He was small.
And a beautiful looking Keating knife. No, is it fish?
Where?
Oh, straight ahead.
Straight ahead middle?
You're down a little bit and a little bit left.
Left. Oh.
Big
Oh.
left and
This
down.
guy, maybe.
Something really big and far away.
Yep, I got you.
Maybe a Siphonophore. No, it looked.
Yeah.
Oh, maybe
Jelly.
the tentacles are all out, like that guy the other day. It's a huge, massive,
It's a chain?
massive. Is that a
No,
solmissus?
no.
It's a solmissus.
Is it?
Yeah.
Oh my gosh.
Oh, it's called a Ctenophore.
Oh, can we stand
Oh,
off? Can we stand off?
wow.
Stay with
Can we
this.
stand off for a little bit?
Yep. Yep.
So the solmissus is trying to eat the Ctenophore. The
Okay,
Ctenophore
so the
is like,
Solmissus is the Medusa. The Ctenophore is on the left for everybody viewing. And Dougal, what do you think's happening?
2, 3, 4, 5, 6, 7, 8, 9,
He's
10,
counting.
11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. It's a solmissus incisor, like the regular.
Yep. Solmissus incisa. And you think he's eating the Ctenophore or attempting to
eat the
eat?
Ctenophore. He's attempting to eat it. The Ctenophore can grow back even if it's damaged. So it's like, no, well, you can have that bit of meat. Yeah, I'm trying to get out of here. And the solmissus is trying to grab a bit more. I think
That's
it's,
cool. So what's trapped in the tentacles of the Solmissus is parts of the Ctenophore that it was able to
mucus.
capture.
Ctenophore? What is the name of it?
The
Very cool.
Ctenophore, yep. The Ctenophore
It's a lobate?
is... What are those yellow?
Yeah, what's the yellow in there?
No, Dougal, they're not eating. He's farming.
He's farming.
Come on. I'm missing it.
It's like a repeat of yesterday.
I know.
Do you want to?
I don't know what those.
Do you want to? Can we zoom in on the Ctenophore for a second on those yellow?
Yeah.
Was that because of us or?
We don't want to lose either one if possible,
good,
but
you
if
already
we could
see
zoom on the Ctenophore.
Bits of it on the
Ooh, I
Solmissus
see,
tentacles.
I
So,
see.
and those are already like
They want me to
falling
stand up.
apart.
Hy-
See it on the
Hyperids?
other side.
It could be.
I got too much.
Yep. No, that's
I'm
fine.
thinking tentacles, but they don't.
No.
No, the one on the left is definitely tentacles.
They
Yeah,
only
that
be
looks
there.
tentacle to me.
Oh yeah, it is.
It's yeah. Okay. We can zoom back out just so we don't lose the other.
Normally they're to either side of the
Thanks,
mouth,
Edith.
but I can't see what's
It's our
left of
first
the
feeding event
stromidium.
for this trip.
Yeah, I was gonna say we haven't.
Second feeding.
What is the class of Ctenophore?
Well, I mean, it depends on how you were interpreting it
I
last
think it's
night.
gonna be. Is it a
Who was
sedipid
eating who?
Ctenophore because
No,
of the
it's a
tentacles?
lobate. It's
Oh, it is a
lobate.
lobate Ctenophore. Okay, so Ctenophore.
But do you have any idea
Yep,
what the
this
gene
one
is?
right up
Maybe?
here. CTE.
I have no idea right now. I'd like to zoom back in on the Ctenophore.
Ctenophore.
We
And
zoom in on the
then
Ctenophore from
away
the
from the Solmissus.
where the bottom of the screen
Lobate.
is the
Lobata.
yellow tentacles and the top is the top of the head, kind of zooms.
Yep, they're starting to separate. Do you have a preference of which
The-
to focus on?
The lobate.
Okay, so the Ctenophore in front of us. Good answer. It's nice and in the middle. And what would you like us to do? Just good zoom?
Good zoom for now.
Perfect.
Trying to ID what it is.
That is pretty. It's nice. Alright, it's probably as close.
Okay, there's
Yep.
the stomatium.
It's about as close as we can do, Dougal.
Way out. Okay. Focus through the animal. It's a weird place for tentacles.
It's not. That's the shape.
Well,
The Medusa
he did
goes-
just go through a pretty aggressive
The Medusa goes.
meeting. What was that?
The Medusa.
Oh, you can see the.
He Medusa
radial
went on this way,
canals
on the
have these
side.
little
Yeah, they wanted to focus
side
on that.
canals coming out of them, quite long actually.
Oh, that's great, you
they're
guys.
Particular. What
This is
if this
great.
could be intacta?
Wow,
This is great.
It's
Fabulous video, you
already
guys.
Degraded, started to degrade.
Yeah, but just
And these
look at it.
ctenophores can regenerate, correct?
They can, yeah. You can see where the
this?
canals
Oh,
going
it's-
from the stomach are heading out and reaching the meridional canals, but the stomadeum is like very large and it's like Oceropsis shaped. This is like a
It's
weird
huge.
mishmash of like all sorts of things.
Well,
Yeah.
I imagine you want to collect it, right?
I imagine I do,
Can I do
yeah.
a scan then?
I knew those questions were coming up.
Can I do a scan?
That's fine by me. Dougal, you
And
good?
what's that green fluorescent
Yeah.
stuff on the end?
On the
of it?
end, right.
This is just so weird.
Okay, so we're gonna do DPIV, and then we're gonna attempt a collection, and a suction sampler is fine, right?
And well, yeah.
That's our only
Like,
option.
Yeah, microburst and
Okay.
hope we get some tissue kind of a thing. What on earth
All
is
right, we
it?
should be ready.
Okay. All right.
Is that's not the end of their?
Starting to.
No, that has nothing to do with their tentacles.
No, it's the end of the meridional canal. Seems
Right.
to be
Yeah, this guy's
so,
bigger.
green
guys,
fluorescent.
He's larger. Hey,
This is a giant.
you said scan.
T4. I mean, we'll see something really interesting, I'm sure.
Yeah,
Get
Okay.
ready for
Yeah,
some
any record then the
fireworks.
internal structure you
It's
have.
gonna
'cause
be
it's
pretty.
so soft. We're just gonna get a little bit of DNA,
A little bit
even
to
if
the
we're
right.
successful. So this
Yeah.
scan
A little
is
bit more to the right.
our
Okay,
only way
great.
to record.
Perfecto,
its
where you're
Now
at,
you wanna
if
go
you
a
can
little to the
go.
left because you're gonna lose some laser real estate.
Oh yeah, due to size of
Oh,
animal.
a little bit lower.
Yeah, he's low now.
Yeah, you're gonna want to suck everything up.
This is
Yeah.
so weird.
Water, gentle suction should do it.
Amazing scan of large decomposing.
Maybe.
It's not decomposing. It's just injured.
We're good. We're
Injured.
good. We're
Injured.
close. We're close.
Wait, that would regenerate if it wasn't being sucked up by another.
Oh, he's below it.
Do you want me to turn the laser on to help?
No.
I'm gonna do it anyway.
Oh,
Oh shit.
Uh-oh.
Rock.
Can you suck a little all of that really quick?
That's it.
Yep.
Turn her on. Suck him up.
Suck him,
up,
suck him,
and then
suck him, suck
And
him.
then stop the pump. There you
Oh,
go,
we're
there you
sucking
go,
some
there you
in.
go.
We got him,
That's
we got
that's
him.
Keep him going.
He's
keep him
oh,
going.
and he's on the laser as well.
Okay.
Exactly. All right,
And then
he's
stop,
in. Oh, and he's
stop.
coming
Okay,
in.
It's in. It's in. Stop.
Stop it.
Stop the
Yep.
pump.
Stop.
And stop.
Forward.
And rotate.
Rotate.
Yep.
Yep,
All right.
go for it.
We got some
Great.
tissue
Thank
in
you guys.
number
We definitely have
6.
tissue for sure.
We'll probably get
That's
a little
all.
tissue.
Yeah. I mean, that was all we were
That
going
was
to be getting
good.
anyway. So.
Large ctenophore.
All right, number 6.
Woo, we broke it. Nicely
Yay.
done. We broke that problem.
Well, hold on. I want to wait.
He's in there.
When we get back.
Well, all right.
I hope the
Now
show
you're
cam's still running.
now you're asking for a lot.
Yeah, that would.
We have 2 canisters left.
Where
We're
you
at
going?
684. What do you think? Should we go up to 600? Do you want to hang out
Can
here?
we keep hanging out here?
Sure.
There's a lot of
Yeah, there's a lot of stuff.
that we've seen.
Stop.
Oh.
Sorry, or somebody stop. If you guys need to switch. Okay.
Is that a Halascara bigelowi?
Yeah. He's little.
Looks to be a Halascara bigelowi. Caught one of them already.
Okay. So we'll get a good vid and we're good.
Yep. Especially want to see the shape of the radial canals close to the stomach. Are they rounded or is it straight the whole way down? Just hard to see what he's. Oh, they're straight the whole way
Sorry,
from the
son.
look of it.
Não, eu tô separando o bicho lá.
Não, vai lá de vai.
Everybody needs to decide what schedule. You guys need to decide what schedule you're going to follow.
Why are there 2 schedules?
There's a schedule?
I did. Google, I do have a schedule for you.
She did.
I do have a
I've
schedule.
seen it.
I create the schedule and then I think the sea log folks switch it due to what they're doing for work. So it gets switched, but I'm not sure everybody sees the switch or knows which one to follow. So I think we're getting 2 now, and folks are getting not sure which one to do.
Could you make it like an editable Google Doc so they can change it?
Whoa, broke up already. Might have just been a tentacle. It's basically, if I've slept, I'm in here the whole dive. If I haven't, I'm not.
2 canisters left. And I do have people for you to rotate. I have it on the schedule.
Yeah, they're definitely.
Oh.
Great.
Is that a colour copper? Where are we going live?
We're doing a pilot
We're gonna be
switch.
doing a little,
We're going to continue to explore in this
send the
area
video
for a little
feed
bit.
to the Fathomverse
We have a couple
Discord
hours
server.
left of the dive. It's about 5 o'clock,
don't change
and we should be on the surface at 7 o'clock tonight. We're 350 miles
like,
off
where are
the
these
coast of Brazil in the South Atlantic Ocean, and we are exploring the Midwater column.
Wait, Dougal, you're leaving?
You two ready? You good? We've been having fun, collecting lots of things today. We have 2 canisters left. We're on 7, and we do have Niskins to fire at 450, 50, 50, and then 2 at the surface. So I can repeat that. We've gotten 1 already. So then Niskin 2 will be at 450 meters.
We have 500.
Yeah, I think it changed because we ended up deeper than the original target. So there was some math that had changed.
Shrimp.
So it could be 500 or 450. You choose.
Oh, you tell us.
I'm going to go 500 because we went to 1,000. And then 2 at 50 and then 2 at the surface.
Good.
Awesome. Thank you for that. And again, no samplers left, only 2 canisters left. Thank you.
7 and 8.
We're hanging out, searching away. Yep, and the goal is to go just forward. This is a something. Let's take a look at this.
Oh, it's another one of those.
Yeah, it
Siphonophore.
is. What's going on?
What's going on?
There's a lot.
Someone's out
If we
there. after
could
these
scan
Siphonophores.
slowly to the right with this. We've seen this a few times today already. Trying to keep it in focus, which is not an easy task because we don't know how long it is. Yeah, sames. Yeah.
I don't think I've ever seen so many. You see the
Not in other
random
dives.
string here
Yeah.
and there, but
I haven't seen this. Okay, we're good. We can keep moving. Up, slow, searching. Perfect.
do we have main targets? Oh yes, the doliolid
Doliolid chain.
right?
Yep. If there's a doliolid chain, that's still on. And we don't have not seen any Tomopterus this one either.
Yeah, not even one, right?
Right. So a doliolid chain.
Oh, straight ahead, right in the
Straight.
middle.
Yep. Thank you.
Go forward.
I was writing our.
There was a
options.
Desmorphes there as well.
Tomopterus. Yep. Thank you.
Oh, it's a hybrid. It's that cystosoma thing.
Oh, we're gonna have to get this.
Yeah.
We're gonna have to dump
They'll be
Do we
wanting
just
this.
collect it? Let's get a video for a second. Cystosoma?
Is this okay?
Yes.
Yeah.
Is this okay in a section sampler?
You could go zoom.
Yeah, if you.
I think we're gonna
really
need just to
careful with it.
collect. Can I just collect, or do you want a DPIV?
No, let's. It's
I think
too
we just
valuable.
collect.
Yeah.
An amphipod.
We're collecting.
Yep. We're just gonna go collect this one right now.
Cystosoma something or other, right?
Cystosoma.
On this guy.
Yep.
For a scan, yeah.
We didn't know if we should scan or just collect.
We should do a little bit of video first.
You think we
Just
need to do
native.
more video?
You know the orientation. You have the video.
Okay. We're gonna collect.
What's native orientation?
Well, it's not really native with this.
It's kind of swimming around.
Yeah, not with the stuff going on here.
I mean, they
Way
do.
too much
They
fun.
We're
just
collecting
tend to
it.
do
Suction
some
sampler.
Just
Gonna go into canister number 7.
It's not.
This would be
close
the.
enough.
We hope it's the.
We
zoom in.
have gotten a shot of the head.
We haven't had a zoom.
straight down.
We haven't had a
It's not
zoom
square,
in.
is it?
Yeah.
We
It's
haven't
not
really
square.
zoomed and observed.
Same as always.
It's like a weird echo.
Yeah, I heard the echo.
This is the.
On the other side.
It's not found under Discord.
Yeah.
There you go.
Yeah. You got him. You got him. Suction down. Why is it echoing? Somebody have it on somewhere else?
Has it on.
Yes.
No. There was an audio.
Yep.
Connie had it
Yep.
on her phone.
It's an undescribed.
Hasn't come into 7 yet.
Family, genus, species.
Right. Oh, it'll be nice in the shadow bath.
So
Yeah.
Leary is like a common name that people are calling it, but it's only it's not even
Made it yet.
No. It's gonna be pretty slow. We shut that suction pretty quick.
That's pretty cool. All right, I'll figure out how to do that.
SS7. What are we gonna call it then?
These are great. Yeah, I was gonna say we've got them in the toes.
Do you have a?
2 of them were tiny, the tiniest film I've ever seen. Like 3 little babies.
Maybe.
Yeah. But I'm not sure where it goes.
Maybe give it a little.
Like a
tiny
microburst.
bit of-
Oh, there he is.
There Never mind.
Yeah.
Stand by. You can switch it. Switch containers.
I mean, I guess Magna, but
Stand by.
need to be seen when
Let
we get
him. These might not go down.
He's happy.
If we could
Should be okay to
switch.
it.
Yeah. Thanks, you guys. Perfect. Nicely done.
Super. Yeah, buddy.
We have 1 canister left. We have 2 hours still to go. We'll see how this goes. If the canisters fill, we go
The
up.
canister's filled and we can make it up before dinner. We can,
Okay.
We can't. It's 5:30.
Yep.
We
Right.
always eat it.
I'm resetting
Right,
my
always
eyes
just
to
do-
only react to Dolly chains and come up.
Okay. I was gonna say Doliola chain. We have not seen one Tomopterus yet.
I haven't gotten a chance to process the ones from yesterday yet.
Okay. So Dolly chains all the way down, all the way up, all the way sideways.
Oh, okay.
If available.
Okay.
Not to worry if not.
I'll find somebody else.
Yeah, no worries.
Oik.
Just commenting, right?
Just commenting, yep.
Okay. We can keep moving.
If I see a fritillarid, I'll yell.
Okay.
Okay, there you go.
I was gonna say audio. Mute that baby.
If I see her, 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 fritillarid, I'll yell.
Ignore Doodle.
Doodle.
Doodle or Dougal.
I totally did.
Totally did.
Gotta get a
Totally
caricature
did.
artist to do a caricature of Dougal, so we got a Doodle.
There you go. That's exactly what we need. Low right.
Oh, still misses. Oh, sorry, that wasn't my search pattern for a Dolly chain, but I still want to zoom.
No, we
if
can zoom in on things, of
Yeah.
course. Yeah.
Want to count the tentacles, look at the shape of the stomach pouches.
Sounds good.
And see whether there are any brooding sacs on each side of the
We have to be on
stomach
deck
pouch.
at 7.
Yeah. By 7.
And
we have
if
to
the
be on deck
canisters
by
fill.
7.
Oh, yep.
So we have time because we have to wait.
2 hours.
Correct. And we have 1 canister left.
But mostly now is a great time to scan. You guys still interested in scanning?
Oh, yeah.
We can.
Do you have targets?
We
Any-
do.
Larvation
And
houses.
we can always go up by 100. We can go up to. We need to get some Niskins, so maybe we stop at 500 for a bit.
Do you know which Somasis this is?
You have to get.
It's one
Yes,
of the
Niskins are here.
incisor
Pilots know them
species.
as well.
Do
Are you
want
coming into
to
this
scan
one?
it then?
Fabulous.
Okay.
I'm handing it over to Manu.
Without the DNA,
we may
though,
be
it's
stopping on
like
every
you can't
protist
really
in the next
tell
600 meters.
which they are yet.
But they're
Unless you're putting
fun.
together all the DNA stuff to
Don't
find the
overdo it.
morphology that
The word of
matches
caution
the
from the
DNA.
chief scientist is
Yeah,
don't
it would be
overdo it. That's fun. That's hard to do,
Suck
chief
up the
scientist.
small
I know.
stuff.
Okay.
Had parents on things.
Yep. Would
Yeah, I
that be
think
the Doliola chain?
Scanning is good.
No.
Okay.
I don't really change with.
Hello, everybody.
Okay.
This is Manu. We are ascending upwards.
We do a
and
deep POV scan, please.
if you've heard the chit chatter for a while, we'll try spending some time on the small world. We're going to be deep PIV this Medusa first.
Yeah. I'll pop back in too. You got it. You got
Okay,
it.
and zoom in from the top like that. That's good. Here we go. Here we go. Here we go.
I think a couple of things about these scans. If you've all noticed
But
that
if we can scan it, I'd like to. Okay. Can we scan this guy now, please?
Yeah.
in the laser
Oh,
sheet,
that's really-
you'll all see when it passes through these little sparkles. These are scattering from the small scale particles that are in the water column. Cells, detritus, marine snow. So the only reason the PIV works is because there is an incredible amount of smaller scale objects that you don't see in the camera, but you see them as a scattering in the laser.
Oh, a little bit to the right. And then these pretty.
Right there. And that laser sheet gives you a very, very thin depth of focus. It's an artificial depth of focus because the red light is only present in this very thin sheet. We call this optical sectioning. You know, the animal is not reacting to it, although it is laser light, because it's red. And you can see on your screen now the scattering from the little particles are all the small scale things that are in the water column.
Narrator.
I see a question about when will the expedition reach the seafloor. Not this one. The expedition's primary goal is to map the midwaters. The deepest dives we'll be doing for the last couple of days that we have been. The next few days will be till 1,200 meters. There is a lot that we don't know about this volume of water, and although it is true we know very little about the seafloor as well, but much of this volume is uncharacterized. So the primary purpose of this expedition is to do biodiversity map in this water column.
What? Oh, yeah, the pauses.
Oh, that was a great scan. Did you see the bell, for
Yeah, there's a
example,
lot of
was
movement
beating
in
during
the
that
sheet.
time? So these jellies, when they swim, have a strange way of propulsion. You can see that little pulse. That's coming from the fact that they are shooting these vortex rings down
Going and it's
below.
rotating, so that's unfortunate. And then now we're reversing. Like,
You can see that bell pulse from time to time.
Does it have to be a little bit faster? Going through?
oh, no, it just has to be continuous.
There was a very strange shade in the inside. Did you see that one?
the inside of the jelly.
Yeah.
Yeah. We get a little bit of refraction.
Yeah, you
inside,
get
like the-
scattering from the
body.
yeah, from the body inside.
Keep it going, keep it going.
See that?
There was a researcher in Japan who was looking at the structure inside gelatinous things like this by reflecting light internally through it to try and reconstruct it.
Ah, the animal's rotating.
Still rotating a little bit.
Yeah.
It's just a little tricky. I just walked in and I see that there's some questions on both on the Discord and the live stream around the laser still about how it actually works. I think Manu just explained how we can see just slices of the animals here. I work a lot with the actually operating this laser as well. So actually what we do, it's a two-part system on the ROV. One system is a large housing that's mounted on the core of the vehicle that holds the laser. The instrument's called DPIV and the laser is actually in the large housing. And then we have a cable that transmits just the light through an optical fiber towards this thing at the end of the, on the front of the ROV where you can see that red light coming out of. And when we have a little bit more light in, you might even see a red cable headed towards that end, and that's where the laser light is actually being reflected through. And what happens in that little housing that we covered with black masking tape
We're
is
gonna keep looking for other animals.
Keep going forward, yeah.
it's we're
kind of in
collimating the
this
light
area
into a
or this
laser pointer-like beam and then spreading it out like a fan. Oh yeah, I should have introduced myself. I'm Joost. I'm a research engineer at Mbari, and Discord might know me as Dr. Yeast. And that is because of an expedition with Schmidt Ocean Institute on the previous Falkor vessel. Back in 2021, I got that nickname from the YouTube live stream. Thanks, guys. I looked up those references too. It was like jokes around Dr. Yeast and the Yeasty Boys and lots of other things. The rise of Dr. Yeast. It was some good memories. Yeah, the rise of Dr. Yeast.
There's a Medusa.
That's probably my favorite.
Top left corner. Are we allowed to call you Dr. Yeast?
You're a real
Absolutely,
fun guy,
yes.
right?
That's doctor for you. Joost is fine too.
Dr. Yeast is
Dr. Yeast, yeah. I've only used it on Discord, I think, that as a nickname, but I'm pretty happy with it actually.
Makes you sound like a real fun guy.
Oh, there is. Wait. We have a beautiful rhizarian in front of us. This is a Feodarian, a Cylindridae. And you can see the two tentacles have stuck to each other. We've been collecting a few of them and studying them in the lab. And one thing that we've found so far is they are extremely sticky on the tentacles. We know that Feodarians feed on animals, on metazoans. So let me say this one more time. It's a single cell that will feed on animals. Most of the time I've caught them, they feed on small copepods. Oh, you can see the spines that come out from the central capsule. Feodarians are called Feodarians because they make phytocysts, which could be a waste material of a single cell.
It's trying to communicate with us. It's making a Moebius strip.
Oh, that is so beautiful. You can see the curve, and the thing that's been puzzling us for a while is in this expedition we've seen multiple, it just got washed, we've seen them in pairs, but seeing a single cell in pairs in the open ocean, we don't believe that these organisms can swim. They can definitely control their buoyancy. That means they must be pairs when they divided. We know very little about the life cycle of these single cells. They're found in all the way down to 8,000 meters, but nobody's been able to culture them, so they remain a mystery. We cannot tell how old this cell, for example, is. So it's quite puzzling to see them in pairs. So let's keep moving. We're gonna keep going up forward. We are at 640 meters. Oh, Medusa on the left. Is that the same animal? Google, is that the same animal on the left?
It's all misses? Yeah.
Yeah.
some questions from
So we
earlier.
push forward and try and find some
Yeah.
fresh water with some undisturbed animals.
Hopefully no fresh water, but yes, undisturbed animals.
Some fresh seawater.
Yeah. So while we go out on our journey to search for fresh seawater, I want you to pay attention to this Star Wars-like particle mist that's coming through. I think it's very important to talk about this mist in the context of climate change. What you're looking at is marine snow.
Oh, there's a praying at the top right.
And Rachel Carson, who wrote The Silent Spring and many other science communication book, described this after this was discovered as the most stupendous snowfall the planet has ever seen. So this is not directly snow, but we call it marine snow because it's made of thousands of organisms. Every little dot is an ecosystem in itself. And what you're watching is the ocean. Oh, another is passed through. And this is what captures carbon in the ocean. So between 30 to 35% of every molecule of CO2 that we emit in our atmosphere is being captured by the ocean and stored for long periods of time. And we don't understand either the capacity. There's another rhizarian on the left. Either the capacity or the limit associated with carbon capture in the ocean.
So miss this incisor. Yeah. This is important. It look definite. This is important. It look definite stomach pouches. And if it'll turn around for us so we can see the rim of the bell. Come on, baby.
Oh, it did turn.
Yay. Let me see. Yes, it does. So this is oh, beautiful.
Wow.
Beautiful. Also, I'm gonna pretend I don't know what it is yet, so we can keep zooming on it and focusing.
Is there no space for collections?
The stomach pouch shape.
I think we have one. We have.
Did you want to collect it,
number
Dougal?
8.
Can we still collect it? It's a cunina. We haven't had one yet on this trip.
They're beautiful.
One,
Yeah,
right?
we should definitely
We
scan
should.
it.
I think it's fairly big. We could probably scan it as well.
Yeah.
It is a
8 is empty, right? Yeah.
All right.
So Dougal,
Do you
it's
think
while
it's
we're
big
doing
enough
that,
to scan with the deep POV?
we're gonna be scanning this animal.
Try it.
My name is Kikani Katija. I'm a principal engineer at Abari. I'm also a denizen of the deep for Fathomverse, and so I'll do my best to also try and answer some questions. It's starting to get a little intense in the control room anytime we do scans or collect animals. You know, people get pretty excited. So any animals that we're especially hoping to find, for us, it's pretty open-ended. We're being really opportunistic. There's a number of different animals that we are, hoping to describe. The goal of the expedition is to come across animals that is unknown to science and quickly collect all the information that we need to actually do a species description. And so what you're seeing now is a scanning of an animal where we'll get three-dimensional models from that data. And then that combined with the high-resolution 4K imaging you're seeing from the vehicle. There's also imaging going on through the suction sampler. And then any DNA that we collect from the animals when we, collect them will also be used to complete the species description. So the goal here is to try and do that as rapidly as possible and provide as much information as we can to do that. I think for me, the most surprising find on the expedition thus far has been how many animals we've come across that are, at least based on the experts that are in the room or on the ship, are undescribed. But, this is an area where not a lot of expedition work like this has happened. Not a lot of people look at midwater or midwater animals, and so we have a real opportunity to see a lot of new things. So it's really exciting. And for those of you on the live stream, we're not only doing our best to answer questions there, we're also connecting via the Fathomverse Discord server, so that so feel free to ask any questions and we'll do our best to answer them. And after, when we have a quiet moment in the ROV dive, maybe when we're starting to pull the vehicle, we have a number of models that we can pull up and share. If you want to take a look at them in advance, recommend Googling or searching for Mbari, Sketchfab, and Deep PIV, and you'll come across all the models that we've already made publicly available. Keep in mind, though, that those are models that weren't generated from this expedition yet. We haven't processed that data, and so stay tuned for more as they come out.
I see a question about keeping some of these animals alive. We do have a crystal aquarium which is currently housing a Portuguese man-of-war, and we have a few of the jellies that are still alive, including the helmet jelly that we caught 2 days ago. And we've been studying bioluminescence in that jelly for the last 2 days, which is quite remarkable. So some of the animals have to be processed right away for genetic sequencing and for keeping them as museum specimens, but some of the organisms are kept alive as long as we can on the ship, so we can study the physiology of those organisms.
All right, there's a question about the name of this jelly. I heard from the expert in the room, Dougal Lindsay, that it's Cunina. And in terms of being able to see the names of, because whether there's a place to see the names of the animals encountered, that's a really great question. I know we've been taking lots of notes. The data are going to be pushed to Fathomverse app so that you can start looking at those images. But the real species IDs, for some of them anyways, could take time since they're not described.
See? Thank you for proving me right. Look at this. So whenever we learn, we say we give up. This is never going to work. All right. I'll turn the laser down.
So let's take this on sampler number 8. One of the important things to understand is why are there animals in the midwater to begin with? And much of that is the primary producers in the top layers are the ones that are producing the food that most of these animals depend on. Again, so close. That is primarily what we call as plankton or photosynthetic plankton. So many of you have heard of diatoms, algae, literally tens of thousands of species. Now this water column is very interesting because it's mostly the bluest waters, which implies that there is very little nutrients in the top water column for organisms. So it's quite surprising that we really do have this biodiversity still, and there is a significant amount of biomass in this midwater to begin with. That happens because of blooms, and these phytoplankton primarily divert that converting sunlight and nutrients back into food for the midwater column. We're going to try to suction this jelly in, but as the temperatures start to change, as many of you have noticed, if you look at the temperature, surface temperature is 29 degrees Celsius.
Great job.
Yay.
Actually, Dougal, there's a really good question here. How does the process work to go from observation to naming and publication?
And what do you do with the names?
And
here?
hopefully it
Like,
doesn't
we just
take
named
20
something
years.
Loretta.
How many years?
What's the relevance of that?
Just let me write down the section. It's section sample 8, right? We need to document things very meticulously, as in just now, knowing which canister it went into.
Section sampler 8.
So we make sure we have a proper set of in situ images, talking about ROV work in particular right now. Proper set of in situ images documenting what it looks like alive and happy, and making sure that any photos associated with the specimen, videos associated with the specimen, sequences associated with the specimen are actually of that one individual, because there are so many cryptic species out there in the deep sea that if you catch something that looks to your eyes, your regular eyes, as if it's the same species, but actually it's not. And you can mix up the sequences and the photos and everything becomes a mishmash where you're not really sure what is what. So everything needs to be documented at the individual level and having sets of molecular data as well as morphological data. So we'll catch this animal, we'll bring it into the lab on the ship. While it's still alive and kicking, we'll document it under the microscope, take photos of all the things that are very small that probably you could not see in the video. And then we'll try and do photography using one of the larger camera systems. There's a system here that takes images of the side view, the top view, and the lower view of the animal all at the same time. So trying to get a feel for it in 3D through those images. And then just to make sure it's not an already described species that has a name but not a very good description, we have to go back through the taxonomic literature. So in some cases, this can be 300 years worth of literature. You have to read papers in Latin, in German, in Italian, in French, in Chinese, in Russian, etc., which is a lot easier now with AI to give a hand. Has not always been like that. But get all the information you can to make sure it is not a previously described species. This is the most difficult part of taxonomy. It's easy to name a new species if you ignore the work that's come before you, but that defeats the purpose of taxonomy. So this going through all this old literature, which can sometimes be very hard to get your hands on, buying at secondhand bookstores online, etc., etc. And when you know that it's a new species that doesn't have a name, then you start to describe it. And there's a format for the kind of text you use, the kinds of plates that you need. You need a line drawing usually. And so you document it so other people can know what it is as well, and lay out in a short paragraph what are the exact features that can tell it from another animal, as well as the longer description that tells you more information about the actual individual you had, but which may not be specific diagnostic for the species, just the individual that you had in your hand.
So what we have on the screen is both a Siphonophore and a Phaeodarian. Now
Copepod.
Dougal and I will have to fight it out. Zoom out a little bit so we can see both of them at the same time.
Yeah.
There's a Rhizaria at the bottom which has 3.
Oh, that's Bathysauroi. We know what that is.
All right, so we can focus on the Rhizaria, Dougal?
Yeah. Yes.
All right, so let's zoom in.
Fosteri.
I think what's surprising is we've caught a few of these Phaeodarians and the Tuscodarian, for example, but this happens to not have the sphere that we often look for. Now, Phaeodarians are interesting because they got kicked out of the big Radiolarian group into their own cryozoa recently. And one of the things is that the fact that amongst them, These are some of the largest protists in the midwater column. It's quite puzzling to see how they form this colonial form, because it is very unlikely nature of them finding each other in the water column, so they must have actually divided at some point of time. They're using the tentacles to feed, and yeah, this is a tough one. It's quite small. Yeah, we can keep moving up actually. If this is hard to zoom in on.
Push forward and see if we can find some larger things.
Yeah. Can you squiddle this, Dougal?
I did.
Awesome.
What did you want to call it? By the way, I only put Rosarian.
Yeah, I think that's a Feodarian.
Feodarian?
Yeah. Another interesting fact about Feodarians, and I want to bring Haeckel to the conversation. Ernst Haeckel, who described more than 3,000 species of radiolaran, has said that this is probably Feodarians, and especially Solidendrian, which we've been seeing quite a lot, is possibly the most complex protist he's ever seen in form. Yeah.
Oh no, it's a doliolid nurse. Oh yeah, but not a nurse. We won't collect this because you can't tell morphologically what it is. But definitely videotape it. You see, this is a doliolid that's got all these little babies hanging off its tail there.
How old is this animal?
It doesn't actually eat. It's got no stomach. It just serves as a way to propagate under the microscope. They may be able to tell what it is from that.
Put it in?
Do we want to collect it?
Yeah.
Yeah. So let's put it in jar number 1. I think it has a shrimp in it, and we will double it. And that would be it probably for the collections. I don't see anything else we could double in.
Is there something in 8?
There is.
There is already. We
We're not
just got
looking for the doliolid
Narcomedusa.
Still? We don't have a chain yet, but
There's multiples there. We just need enough to get a decent.
Oh.
Could you reach around and index it?
Looks like we're going to try collecting them. Yeah, you can see around the main part of the body, there's these kind of blue-ish muscle bands that wrap all the way around it, several in this view. And it's those that contract, compresses the body, reduces the internal cavity volume, and generates propulsion.
That's the thing, the life cycle of these guys is like unknown.
that's what I was confused how long it's been floating. As far as we know, this could be like Sudusa or like some other thing. Could also have a nurse day. Who knows?
They have a complex life cycle that we really don't know.
Okay, this is in 1. Since we are in the middle of our expedition,
It is worth mentioning that we're expecting to be at sea for several more days. So if you want to continue following along, just check back. The Schmidt Ocean Institute has been streaming the dives to YouTube and Twitch.
So since we're in the middle of the expedition, we've been discussing what animals we have seen, collected, and what still remains to be seen. And so there is a little bit of a priority list in terms of what we're starting to collect now, halfway through. The shrimp is definitely still there in 1, and let's see if it shows up.
Don't see anything yet.
Is it there?
Oh, it is, yeah.
It's there. Okay.
You can.
Oh yeah, it's
In between one
Yes, please.
Okay, let's double up with Violet tunicate.
I could do it if you want.
Unfortunately, we, don't
on the
really
left.
have a schedule or plan. There's a lot of things that can change while we're at sea. So, the hope is that we'll be doing a series of dives for the next couple of days.
Dougal, there seems to be 2 tentacles coming out of it. Very
If
long.
all goes well, we might have another dive later this evening.
Oh. Oh, that was incredible to see. You could notice the contraction in those tentacles. We were just
Yost.
discussing what causes the contraction in these tentacles.
I'm being targeted. I can take that question from Sarah. I got to speak some Dutch earlier already too. This is my opportunity to use my other language. All right, I got a question there. Oh, if I went to university in the Netherlands, I did. Eindhoven University of Technology, or Technische Universiteit Eindhoven, where I studied applied physics. I did my bachelor's degree there, my master's degree there, and then I did a PhD through that same university in Berkeley, California. Worked on making laser-based particle accelerators, which is basically the same thing as doing deep sea research. And I'll tell you why. Because to do that, we used high-power lasers. Anybody see a laser today in the live stream? I did. We also used pressure vessels. We used vacuum chambers because the intense laser light we were using and the particles we wanted to accelerate needed to be in vacuum chambers, which are pressure vessels. Well, the ROV is basically one big pressure vessels with other pressure vessels attached to it. And then in order to do the science we needed to do, we used a lot of remote imaging, so cameras in places where we couldn't physically be because of radiation or vacuum or temperatures. And once again, we're doing remote imaging right now through all those different camera systems. I don't know if anyone counted them today, how many cameras are right now on the ROV, but I bet it's at least a dozen. So there's a lot of overlap there with my previous experience, and I was always interested in marine biology, marine science. Actually considered going to university in Groningen. Just decided against that kind of at the last minute, but I'd always been interested in marine life, worked as a dive instructor, so just sort of morphed back into the marine science space through engineering. So yeah, it is not always the most obvious path to get to do something like this. And let's see, and how did it get you to SOI? Well, through my work at Mbari, where I ended up after my PhD, now I get to do these amazing things out at sea with the Bioinspiration Lab. And yes, there's a difference in laser power. The laser we're seeing here is what we call a CW laser, or a continuous wave laser, meaning it's always on when we press the button, and not like a flashing laser like the ones I used to work with. So the laser here is about 2 watts, which is less than a light bulb. The laser I used to use back in Berkeley was about a petawatt, which is for a brief fraction of a second more than all the power consumed in the entire world through electricity. So it's a very powerful laser we used to use there, but very short pulses.
Did we see one of these today?
We snuck in a nice scan of it at the end of the dive yesterday.
So they're currently just answering questions and just move the mic around as you know.
You have to look at the bracts to see whether the dorsal brachial canal leaves from the left or the right branch of some other canals. And yesterday, ship is vibrating, animals are still swimming, hadn't had enough sleep, so I put it in formalin to look at later. So I can't really tell you, but I think it's Rosacea plicata.
So I see a question about if we expect to see things around the corner, the northeast corner of Brazil. There's a lot of currents here, and I think one thing that we noticed in the satellite images is if you look at the current satellite images, there is a little bit of a Sahara dust storm that's turned downwards, and we're starting to see on the surface water some of the particles landing. So clearly the geochemistry of the water is going to change. I don't know how farther up we will go in this expedition to see that change, because we've had to have change of plans. But definitely many of these conditions are transient, and for some of us who go out to the ocean, you never see the same ocean again.
Thank you.
Find one that looks like that but is red.
Starting to get hungry.
Yeah, the scattering layer, there is one above 250, 200.
Big pepper pot.
There's
What's that on the right?
chain on the right.
Is that a Brachycobia maybe? No. Yes? Very small.
Look at the second one then, huh?
Wow, that's big.
It looks Nanomia-ish, doesn't it? Yeah, it looks more Nanomia-like.
Someone's saying on the Discord that oftentimes it's shocking how small things end up being. We also are sometimes caught off guard when we get closer and closer, and at some point the lights no longer hit the animal. It becomes dark. It means it's just about on the camera lens. So that happens to us pretty frequently, actually, when we want to get a closer look, and we realize just how small it is when we're about to hit it with the camera lens.
Yeah, in fact, we had that happen earlier. We collected 2 fish kind of back to back. And the second one we collected was really, really small, and I think that surprised everybody in the control room.
Oh. Wait. Is that a No, that was a jelly. There's a green copepod. Is that a fish?
Fairly large one.
Yeah.
Let's go take a look at him.
Right there. Just. So while we're zooming in, I think.
It's too short for a Samia, wouldn't you say? Looks like a McTurfid to me.
Oh,
Nice.
there's a shrimp to the right.
In one view.
There's a comment about the nutrients. Indeed, when iron dust lands up in the ocean, it does actually perturb, and it's believed that iron is one of the most critical nutrient that gets deficient in the top waters. So off the coast of Africa, the west coast, often there are blooms that are driven by the Sahara dust. So the hope is that if the dust makes it all the way, we might actually see blooms further up north. But down here, it's the water column is actually, at least as far as phytoplankton are considered, very, very low. The most common thing that we see are acantharians, which are another radiolarans, but very, very few diatoms and dinoflagellates. Although we do have some bioluminescent dinoflagellates up in the top water column, mostly Paracystis. Paracystis actually has an interesting story about deeper waters, not this deep, but when we cross around 200, 300 meters. Oh, to the left. Yeah, right there.
Take a look at the spines on that guy to see if
Yeah, let's count the spines because we have 1 spine, 2 spine, and 3 spine so far. So that is another Phaeodarian right there, spinning like a ball in front of us. And it's very unfortunate we have no bottles to collect.
You'd like
What?
dive in and follow the cable down.
Oh, that is beautiful. So let's just admire this object for a second. What you're looking at is a perfect sphere, which is triangulated by a silicaceous mesh, and the cells are sitting up on the top. And what remains a puzzle, if some of you have taken some classes in crystallography, that tiling a sphere or a curved surface is quite hard. And they seem to have a triangular mesh on its surface. Now, how on earth does it calculate where to put this mineralized structure? And you can see a faint triangular mesh right there, but the finer mesh is the little sparkle that you actually see. And again, we have this particular species collected. You can see just the one tusk coming out. The cells are on the periphery, and the object that you're looking at is roughly of the order of 2 centimeters. So it's quite large. Most phaeodarians have a hollow shell, so these are kind of coaxial glass structures that are built. And the fine mesh is this beautiful triangular mesh on top. Oh, that's just such an incredible video. We have no idea how they eat, no idea how they would grow, and also no idea how this would divide. Many of them form swarmers, so the stages for the same single cell in its cell cycle might be very different in morphology. You can think of it as an analogy to metamorphosis, but for single cells, where you could have a swarmer stage, which is swimming, and an adult stage that would look like this. And again, they are sequestering most of these minerals. Yeah, we can move up. They're sequestering all this mineral from the water column itself, as many of the diatoms do, for example.
Can we go to the next one, Manu?
Yeah, we can move up. We let it sink.
Okay, move on.
Thank you. Shrimp? Suggested shrimp, it looks like.
Oh. Are we going to chase it down now?
No.
Oh, it's climbing up because it's nighttime. How awesome would that be?
Oh.
Look.
It's not a suggested shrimp.
No.
Something else. So,
And it's gone.
I think there is a comment about volvox. Indeed, volvox is a colonial protist, so the analogy is quite interesting. Mostly found in fresh water, and those colonies also form remarkable spherical shapes. Yeah, we can keep moving.
Get some air there, Sonny.
There's a fish in the other screen. We're gonna have a quick pilot change so they can eat.
Okay. Okay, cool, thank you.
Oh, I see a medusa on the left, far left corner. Another foram that just passed by.
Foram.
Yeah.
Still haven't seen a curious Coelodendridae.
Yep. So I know a couple of days ago there was a big discussion on whether we were looking at a foram or a phaeodarian. We collected one of them, and we can confirm at this point that this is Hestigirella pelagica. There are multiple species. We've also caught a Hestigirella digitata. Both of them are forams. They have an incredible record in the fossil records. And many of our fossil resources, and the reason radiolarians are mapped so well in the fossil record is because they are used to predict oil fields. So there are many geologists that study fossils for them. Something that passed on the right. There's a question about nutrients for the ROV pilots in captivity. I'll let the ROV pilots answer that.
What kind of nutrients we need in captivity?
Yes, yes. It is indeed true that the vehicle has to be piloted at all times, other than the 10 seconds pilot change. Oh, I see a marine snow sinking with a comet tail at the bottom. That comet tail reminded me of a story that was recently uncovered. Oh, a jelly on the right. That many of the bacteria that are eating this marine snow also have a capacity to secrete mucus to slow down the flux. So remember, I talked a little bit about how the ocean is capturing 30 to 40% of all of the carbon that's making to the midwater. If a particle makes it to the midwater, you have locked carbon for around 10,000, 15,000 years. So that's a long amount of time. But bacteria slow this flux down and leads to the capacity for the ocean to not be able to absorb that much carbon. So the ability to study these particles have a huge implication on how our planet will be able to cope up with more carbon in the atmosphere.
There's not much at this depth.
Oh, another Risaria.
Oh, did you see that bioluminescent spew?
On the left?
Yeah, something just escaped
Flashed.
and spewed bioluminescence.
Oh, that could have been an ostracod.
Could have been.
We haven't seen that many ostracods in our toes. Oh, I think up on top there was a chain. I think we missed it. Ah, yeah.
Siphonophore?
Dougal, do we want to focus on that? Look
Yeah,
at it?
please.
Yeah.
Zoom in.
Okay.
Right there. And this is one of the reasons to study the midwater, is to understand what actually happens to these particles. Because most of this carbon does not make it to the benthic, and it's actually consumed in this midwater.
Might have lost it. That's okay.
Yeah, we can keep moving?
Yep. Keep
Yeah. I think there is another Rhizarian to the right. Just focus on that for a second.
Right, straight in front of us in the center. Thank you.
And I'll mute the audio.
Oh.
Slightly to the left.
Another
Philodendron.
Philodendrium.
Is it a squid?
Oh,
No. Oh, it's just a colony of...
right there.
That's what we're looking for, yeah. There it is, the chain of Doliopsidae.
That's the tunicates we've been looking for a while. So could we
No, you can.
Let's do
Could
some
we double this up?
observations of it first.
Yeah.
And then
put it in the same chamber?
have a look at which chamber we might have a chance of.
I think the best chance is the one that we have a shrimp in, and we have another tunicate in.
Shrimp will probably chew it up. What else have we got in canisters?
I'm just looking.
That's easier.
There's a Siphonophore, there's a fish,
Neither of them will work.
there is a Narcomedusa.
That might eat them.
Let's zoom in on this. This is the one we want to collect.
What do we have in there?
We have one Rhizaria, and we have one.
That would be challenging.
The larvation.
Is this a particularly unique one?
Yeah, this is a new one. We don't have this for sure. What about we double it in the same chamber that we just got it in?
The
one with the mix.
Probably be destroyed. These guys are super delicate. So we can try. See if we get anything out of it.
What about some of the other?
Chamber
You want to look through?
Yesterday we put them halfway between.
Oh, you can do that?
Ooh.
I would put
It's
it.
a dolly.
I mean,
It's a dolly opposite.
What about
Yeah.
the team force?
just the. Okay, I would just put it on the shore.
I think there was
Take
one.
a chance of losing things the more we go past other buckets, so
I see.
I would
Yeah.
just put it on there.
Yeah. So
Okay,
pilots,
cool.
we're gonna put this in bucket number 1, but let's just observe it for. Dougal, did we get any good shots already?
I was over there.
I didn't see this.
Yeah.
Okay.
I'll take
It's
some
kind
time
of
to-
looking a little beat up now.
I'm gonna see if it's got red pigment spots scattered over the body or not.
We're trying to look a close look, but this was one of the high targets priority items for today's dive, so we're going to collect it.
It's a chain, yeah. How tight can we get in there?
Good.
Uh, Most of these organisms are incredibly delicate, so even when we handle them in the lab, they rupture, and you can see it was to the right.
Blow apart.
I think it.
It's going up.
Right there.
Okay, got it.
Oh no.
To the left of that guy.
Oh, a little further back.
It's going up, it's going up. It's going up. Follow
The other up.
Okay, and turn
We're all
right.
pointing. Is it that one or no?
No, it's in the center somewhere.
Forward.
You have to move forward.
There it is.
There it is. Okay. There we go.
The right. Can you find it
Oh.
again?
Yep, it's right here.
There.
I'd like to go. I think you want to go too, right? Go first.
Canister one, I heard.
Are you in it yet? You can
Canister
go up
1, yes.
Canister one.
Yeah,
there. If
canister
you
1.
see anyone
Yeah.
up there, can you ask that they come and change?
We are on the water.
Okay, that's fine.
Manu, I'll go there because I already set up my. I already set up.
Yeah, you can come in. Yeah.
We're just gonna do
Yeah.
very light suction.
So that's good.
All right, I'm gonna head out, and I'm gonna pass the mic to Kakani.
So, would you bring the camera down to the phone?
I think we have to be at the deck on 7.
Yeah, so we don't have to do. What are we sampling?
We are sampling on what?
On one.
We're putting it on top of one. Discuss that.
There. Okay,
And
but
one
then the
person
skin model,
just
just
is
doing that.
firing at 4.
450, 2 at 50, and then one at the
One on the
surface.
surface.
I thought Russ would be down here. It's still yours.
I don't know how many they'll. Maybe just one. Yeah, let's just stay with that. I don't think they need.
So the 2 at the surface.
You haven't been there, did you?
I'll check upstairs.
You need a replacement.
Yeah. Marco 2 replacements. 2 replacements. 3, right?
No, I just got plate 3 yesterday.
Okay. For me and
Google, there's a great question about why are Animals in midwaters.
What
Mostly
about,
jelly.
what about animals in midwater possibly?
Mostly jelly.
Mostly jelly, yeah.
Why are they mostly jelly?
Oh, why are they mostly jelly?
Yeah. Talk to the microphone.
Well, there's not a lot of food in the midwater, so one of the strategies is, well, try not to use too much energy, make your body as large as possible with the least amount of input, and making organic content to make your body big. So making your body gelatinous and watery allows you to increase the amount of volume you can spread your tentacles out or gather your prey. There's no waves, or you don't suddenly hit a school of tuna that might break you apart, so it doesn't matter if you're very fragile. There's nowhere to hide in the midwater, so being transparent allows you to kind of blend into the background, the surroundings, to stop predation. So all of these things are reasons why transparent gelatinous animals are so common. It's not just jellyfish, cnidarian stinging cell jellies, but the phylum Siphonophore, comb jellies. There are transparent amphipods, transparent octopods and squid, transparent arrowworms, all sorts of things are transparent and gelatinous down here in the midwater because of those reasons. And maybe they're just being nice to us taxonomists so that we don't have to dissect them and can just look through their bodies to see how they're put together.
Yeah.
Oh my.
Small and almost invisible.
What did you guys just collect?
We were trying to get all the opposite.
Okay. I
Did
just
it
walked
go
in.
in or no?
I can't tell.
It did? So we might
I
go
don't.
first?
I can't tell if it's on the.
Oh, is that it?
There it is.
Yep.
Oh,
no, he's in. So inside. Okay.
There it is.
We've got three sides.
I think halfway. I think.
Yes. Have you eaten?
Yes.
Here we go. Halfway.
You want
Great.
thank you for the assistance?
Yes. Are we? Do you want to sit here, Karen?
Yeah, I want to eat dinner first.
I haven't eaten either, but I'm
They're set
ready
aside.
to eat.
Plate set aside for us already.
Yeah, that's on the very time.
Do we? Are we?
To be able to download.
No.
Yeah.
So
We are. We are done.
we are done with collecting.
Yeah.
It's all about videoing, scanning, whatever can be done without.
Okay.
So, I'm.
They're just not here. Well, I think
biggest
we
one we saw was the
one.
one that was weird. The one we tried to scan. That had the like this Marcos. Do you want
Well, we
dinner?
saw one later as well.
I'll take over for Marcos.
Did you find out anything from the Tina farm? Or which Tina?
Yeah, you have to say you guys go grab food. Is the net okay?
Try those lights.
No. But we
The
have
sewing
one.
kit's
A
there.
little bit.
I already sewed. Sewing
The-
is done.
the-
The sewing machine, John just told me a little while ago.
Yep, and there's Fiona, who apparently is really, really good if you needed help. I don't know who that is, but that's what I've heard.
Okay. Yeah, no, I haven't had a chance to do that. It's been-
Okay.
Totally making me.
Oh, I see it.
There, would you rather this or seal off?
I've done
Doesn't
this, I
matter.
haven't done that one yet. Have you done that?
This one, yeah.
I have not done that yet.
I've been
Yeah,
helping
always a good call.
Google this the whole time. Yeah.
Perfect. Let's do it the way we've got it. What did we get in the last container?
We were stacking.
Oh.
Pretty jelly.
Easy enough.
Okay, you can go ahead and zoom.
Ooh.
Go
think so.
Looks like a Columbinema.
We've gotten a few good scans of this. Do we want to get another one with the tentacles kind of deployed this way?
It's sitting
Before
still
it
very
starts,
nice.
yeah,
I'd
before it
scan
starts
if we
swimming.
can. I'll
Which
hop on it.
group is this? Do you know? Narco, trachea. Okay. We are not sure.
What is that, Heather?
We will put Medusa other. And then are you guys going to scan it?
Yeah, we're gonna scan it really.
DPIV?
Yeah.
Yeah.
Unless you want more video first.
No, I think we're good on video. It'd be great for us to get a scan before he tucks his tentacles away and starts swimming.
Stop a little and we'll start following it down. The center spot.
It is.
Turning off.
Oop, it's inwards in DPIV.
Well, keep the uppers up until you're ready.
Emily, you can take this seat. You haven't eaten yet, and we have to get you food.
It's 1:15 already. 6.
I'll get you. Here. Seat.
Maybe he'll settle. Maybe he'll settle.
It's spooked it. Almost to the phone. There's like a tentacle or two. Back through it. Close. A little too much vertical movement there. I think I saw a question in the chat too about the refraction of the Ctenophore we saw earlier. Now these different type of gelatinous animals can have a different index of refraction, and that can indeed result in slightly different behavior of the laser. This is why some of these jellies that look very transparent with very thin tissues work so well with this laser system. So it doesn't get like all refracted and the laser doesn't get pointed the wrong way, because we really want to just illuminate one little sheet. And you see that here too. This is a good example actually where all the tissues are extremely thin. So the laser mostly shines right through and reflects just enough back to the camera that we can pick that up. Oh, just above. Say bye-bye, Columbinema.
Bye-bye, Columbinema.
Thank you, Marco.
Looks like we. Oh, okay. Yeah, happy to introduce myself again. This is Emily Clark speaking. I work on the Fathomverse team in mainly a content management role. Game including our animal videos, and badges. Just to mention a few. got in the center? Are there Medusae? Can we get a look at that
We have another Medusa.
in the room? The safe behind it. Okay, we're following that.
Like the top of the bell, and where the tentacles meet the bell. We could zoom in some more if it's agreeable. So then that way he can get a confirmation on it.
All right, if we follow this just for a little. Good zoom. It's like a 4M just passed by as well.
Another one?
Yeah. Yeah, do we go for that one or do we? Oh, it's right behind Iris now. The expedition is going well. We've settled into kind of a schedule now, but it's a ton of dives and then also nets that go out once the ROV's back on board. And when those come in, we also have a lab-based imaging system. So I have a few macro photography cameras that are all hooked up together. So we can stream 3 at once, trained on a little tank. And so I can take images of the animals that we're collecting with the ROV and also that we're collecting in the trawls. So that's been really cool to see the kind of variety of animals that come up. Most surprising thing.
Coming up.
And I'm watching from Mission Control.
So 450 meters, right? We're gonna do
500. Because it went to 1,000. But yes.
fun.
Yost, do you know what distinguishes a scyphozoan versus hydrozoan?
Yes, that's a perfect question for Dougal.
Yeah.
That's a real brief thing.
Oh, can we get a
I
little
think I'm live
zoom
on the
maybe on
Discord
the
stream here, so everyone can see what mission control looks like from the inside.
Hi, everybody.
All right. Emily, stick your hand up. That's your speaker right now.
Hello.
We've got the pilots in the middle. And we've got the intrepid Alex Havens there, making sure the streams work.
Well, try
Which are we looking at? Is it that little?
We don't need the fish.
Okay,
That was
no
a
more
cyclophony.
fish?
Well,
Okay.
that was a cyclophony, so we don't need
Catching
the
up.
fish.
No,
If it's not a
goes down
cyclophony,
at the
yes. But
bottom.
if it is a cyclophony,
Its fins looked different, but
That
maybe
guy
I'm
was
just not as
cyclophony.
familiar.
Fish. Yeah.
No, not a fish. It was like a house or something.
Oh.
It was just right below us. I thought that's what you guys were stopping on.
A little
Hello.
Siphonophore maybe down there, or sinker. And keep moving though. Keep searching. Okay.
Radiolarian, Rhizarian.
All of the samplers are full, right?
That's what I've been told.
Yes,
All the buckets are
they're all full.
I think there's even a double in one of them.
Oh, really?
Yeah. A shrimp and a jelly together.
And a double fish.
And 2 fish.
Yeah.
Oh, wow.
Some fish that our fish
There might only
experts
be
haven't
one
been able
fish.
to identify.
There might only be one fish now.
Are there any animals the team especially hopes to find? Well, we're always looking for large larvation houses because to get nice scans with DPIV. But I know asking around on another ROV dive, there's a lot of folks looking for anglerfish, including our pilot Sarah over there. She's looking for that. Dougal's looking for all things jelly. Behind me, Heather is looking for cephalopods of any kind. We had a really nice midwater octopus the other night. I know that can be a tricky one in Fathomverse because they look so much like squid. So we had one of those. That was exciting to see. But we have a variety of scientists on board in terms of specialties. So there's a lot of different animal groups that people are looking for all at once. It can be kind of a, let's say, competition in the control room to choose what we focus on.
Yeah, I was going for this. Oh, yeah. That's what I saw.
Yeah. It's like a Ctenophore there. See, it's comb rose pulsing. Oh yeah, looks like a big Siphonophore out there. What do you think, you guys? Siphonophore or Ctenophore?
I think we need to zoom in on the middle first, just to get a good quick zoom, and then we can go to the. Right.
So there's a nice trick. If this is really special, then it usually takes about 10 seconds before Dougal storms in. 7.
Start the countdown.
6.
As
5. 4.
3.
Well, I guess this is actually a really common animal.
Okay.
No, we did see one of these earlier.
Yeah. Yep, it's a lobate Ctenophore. I think the one we saw earlier was the one with the one wing that didn't, they were unequal.
Interesting.
One was regenerating.
For the badge question on YouTube, the border colors are grouped generally by the type of badge, but
CT,
not really any symbolism beyond that.
So it's too big.
I'll work on it. I'll come up with symbolism for you. Surprising find. There's a really surprising to me. Hi everyone. Gulper eel the other night, which is exciting because it's on the Mbari logo. So we saw it in person. That was really fun. We even got it to look right at the camera, see its little eyeballs. So cute. Fish around. We are at 520 meters, and it looks like based on we have kind of echo sounder data, which is a little bit like echolocation or a fish finder if you've ever used one of those, and we can see that in the midwater zone there are these diel vertical migrations, basically a daily migration upwards when it gets dark out, and then the scattering upwards, and then a kind of band that forms around 300 to 500 meters during the day when it's bright out. And so a lot of the deep sea comes shallower, especially about this time. What is it, 7 p.m. our time? So it's been like an hour since it's been fully dark at the surface. So we're seeing that scattering. And that, besides humans commuting to work, is thought to be the biggest migration daily of animals. So we are getting a look at that now. We're a little below where the scattering's happening currently, but there is a thin band of animals that looks like still there. Yeah, but just in the, there's still some hanging out right, kind of right where we are.
Yeah.
But yeah, mostly at the surface currently. In that first 250 meters or so. House on the right.
It's a really faint house.
Looks maybe empty, yeah.
Yeah, that doesn't look like
Pretty
there's
empty, just an outer house. So this is from larvation.
Can we just run a laser through it real quick? Because, well, now it's...
Maybe not.
Now we can't. Never mind.
Because one of the first times we came across one of these, we couldn't see the inner filter at all, and it wasn't until we hit it with the laser that we could.
And I think, folks, you just got a nice little gallery of the various screens came through the stream and the picture-in-picture that Alex put up. And those are all the different screens that we get to see in the control room as well. People commented on it earlier, how cool it is to see all those different views. And yes, we do have a lot of different views, but here in the mission control, we also actually have the same view a lot of different times. So that wherever we look, we can see the main science camera. We can see it on slightly different sized screens, which interestingly, sometimes it's easier to detect things from a little further away when they look smaller or bigger. So I find myself looking all around on the different screens to try and figure out what something is. And you can catch things from the corner of your eye as well.
Any idea on group?
Halascara. Although I always said Halascara.
I did too, but Halicratidae is the
Yeah.
level above
Google
that.
says Halascara, so, you know, can't argue with that.
Do you know the bigger group for them?
The Halicratidae.
Let's see.
Skyphomedusa, Narcomedusa,
They are
Tracheomedusa?
Trachea.
Thank you. I don't know about spelling. Give it a go.
Halicreatidae.
Thank you.
And then since we're also on the Fathomverse Discord streaming through now, it's maybe a good time for maybe Emily will tell everyone what the special things we're doing on this cruise with all the dive data, because it's a first for us.
Yes, thanks, Joost. Yeah, something very exciting is that as we're doing these ROV dives, sometimes 2, even 3 times a day, we're looking for that over there. That data is going straight into Fathomverse. So with a barely 24-hour turnaround, the dives that you can see on these dive streams are showing up in the game for both classification in Pulse and Identify and Illuminate. They're also in Bound. So you can see all these images that we're seeing and help us annotate at home.
Might be another one of those Halicratidae.
So I was thinking tricky. It looks similar.
Yeah.
We're gonna keep searching, so I'll keep blabbing. But pardon me, I did just wake up. Sorry, everybody. I am on nights. But we also have playtime badges available, a variety of tiers, because folks start earning them quite quickly. So any playtime in Fathomverse counts towards them. The first, I think, is only 5 minutes. So check out Fathomverse and earn a Falkor badge, or 3 or 9 of them, and play along as we're diving, because it really helps us process this data. And you're really doing a lot of what we do in the control room by covering those annotations of the exact data we're looking at.
Let me actually show that in like.
Oh, yeah.
We're gonna go here.
Oh.
Just a minute.
Oh, okay.
Right, so there's a number of annotation tools. This one. Sea log, right at 500, so
Yeah, they can.
Golden
They're
five.
pretty good. Yeah, they said normal.
Bring the camera over.
and then there's another annotation
That was
tool
sea
that's
log,
being
yes.
used. This other one, other station. Elia's here. And so basically all of the data and input is there.
Let's see, I'll do it.
Of course, we have our own systems.
Welcome back. Okay, how are we pushing the data so quickly? Well, basically what happens is all these
There.
dives
There you
are recorded as we're
Just
going.
one at 500?
Fired. Yep, bottle number 2, just 1.
So just really quick, we're firing a Niskin bottle. You can see the numbers on screen. It's just a water bottle that can be opened at a precise depth. So since we're hanging out at 500 meters right now, we're going to open a bottle and collect the water from that depth, close it again, so that when we bring it to the surface, it's just water from this depth.
You got it. Thank you. And then 2 at 50 and 2 close to the surface.
And Julieta, one of the scientists on board from Argentina, and she works at a university in Brazil, and she is doing filtration on this water. It's called vacuum filtration, where you suck the water through a small filter, and then you can study the plankton that was in that water.
How long do you
chlorophyll.
guys need to get to the surface? It's 6:35.
Probably boogie up
Right.
about now.
Right. That's what I was asking because I think it is right.
Yeah, that would get us up about 7.
Yep. That's the game plan. So, all right. There you go.
Alrighty.
Nicely done, team.
A few question about the max depth
Hey,
and
coming
how
up.
we determine dive
Yeah.
depths. Well, a lot depends on how far away the bottom is, obviously. Where we've got instruments on board the vehicle, like DPIV and IRIS are rated to 4,000 meters.
Right.
There are some imaging system that Dougal's also added to the vehicle. I believe one of them is rated to 1,200 meters, so that's kind of setting our max depth right now. And so at least when we're planning dives, we're using that as the maximum depth. And I think today we went to just deeper than 1,000 meters. So I think that's kind of been our floor so far.
Is there a certain amount of water we need between us and the bottom?
No. So question about whether we need a certain amount of water between us and the bottom. I mean, of course, we want to make sure the vehicle is aware of what's below us so we don't run into a seamount or anything. Of course, we have diagnostics on board to ensure that. But scientifically, at least the research questions people are interested in, we don't have to operate with a certain amount of distance between where we are and where the bottom is.
The ROV, Sarah, what is the longest the SuBastian can stay underwater?
24/7.
Yeah.
Round the clock.
until it breaks.
Until
something
people run out of
happens,
steam.
really. We don't
Doesn't
have
run
to
out of
worry
gas
about
or
like
anything.
battery life
Right.
or anything like that.
Yeah.
So,
The power is connected to the ship. No need to recharge. So technically they can
Sometimes the
do
equipment
24/7 ops.
Yeah, the equipment
Yeah.
may need it. The batteries on the equipment, perhaps.
Yeah. I
Usually the limiting
sample
factor.
space.
right,
Yeah, in
Joey?
sample space.
Right.
Yeah, go ahead.
We are starting to come up for recovery, probably right around 1900.
Okay. I will come to a stop at the moment then. If you guys need me to move for recovery, just let me know.
Awesome, thank you. And our Medusa, that was the same one again.
Then we're also quite
Uh-huh.
privileged to have, 8 ROV pilots on board? So they're working 24-hour shifts. So there's always something to be done on the ROV, getting it ready for the next dive, but we've also really been able to take advantage of having a lot of time underwater, doing multiple dives a day to, unload the samples and just almost straight go back in. And in between, we're also doing trawls here out on the ship while the ROV's on deck, so we can catch critters in a different way. And I think Emily was explaining how we're pushing data through. We got a little bit of excitement cutting you off. You want to continue that
Sure.
conversation?
Yeah, so in addition, so Schmidt is recording all of these dives as they're happening, not just on the dive streams, but internally with all our different cameras and things. So once the dive is completed, we're already on board, which helps. So we're next to these servers, and we can upload them into our system. It basically goes through an inference process where we do an initial pass through them. Obviously, in Fathomverse, we're not seeing videos, we're seeing frames, static frames. So we go through and sample a certain amount of frames per minute, per second. And we're also trying to do an initial pass in terms of object detection to see, to try and eliminate if there's a ton of frames with no animal in the frame, that's that pre-filtered. And then it goes to either me or Kakani for review to make sure we have good images to go in the game. Basically, each dive forms what's called a labeling run or two. And those kind of are pushed into our general algorithm of what images you see per batch as you open a game. And then we make it live in the game. We have a content management system where we look through all these images and connect it to the game and where those data pathways go. So it's a bit of a soup to describe without any visuals, but it's a pretty quick process at this point. We've been turning them around within pretty much as soon as the download can complete. That inference runs quickly. Our review is just a quick pass because really you're doing the review as a game player. For the question currently up, it says, it seems like a lot of the animals are undescribed, but many have been observed before and have tentative IDs, not formal description. How many actually new ones have been found? So that is a constantly changing answer because we're still diving and still working through all our samples. So it's hard to say for sure. It also depends on genetic testing. So we have folks running genome sampling, and that process takes a little longer. So we're taking notes of the different morphology, the different physical features on the animals once they come up on deck with microscopes and macro cameras and so on.
Don't be afraid to.
But it'll take a little bit to get those DNA results back to confirm or deny that it is something that's been described or not.
And I can add to that is these new imaging techniques, DPIV and IRIS, are all relatively new. This was, of course, not part of the description process for animals in the past. So now that these techniques are becoming available, we need time to demonstrate them as a viable way to help identify these features and describe the morphology. Dougal talked earlier about how line drawings are often part of the description of new species. But line drawings are inherently two-dimensional, so if you want to really describe these varied three-dimensional structures we're encountering here in the Midwater, three-dimensional techniques are probably better. So we're trying to make sure we overlap those techniques to sort of validate these newer methods, and hopefully in the future be able to describe, leaning more heavily on these in situ methods, describing animals as they are happy and alive and swimming around in their natural state. Because as soon as they come up, they often are, a slightly different shape. They might have lost a certain a number of tentacles, if you want to count how many there are. So these in situ observations are incredibly important, and knowing more of their three-dimensional arrangement can only help.
It's going to speed up.
Great. And also, so we have a couple more questions in the YouTube. It says, are most new images in Fathomverse and Bound? Illuminate, identify, or pulse. So for those who've not played Fathomverse before, those are our mini games, different style of gameplay to see these images. And the good news is it's in all of them. We have them in Bound for localization, which is where you draw bounding boxes around the animals in the frame, which helps determine where animals are and what these look like as objects. And then we also have the classification mini games where you ascribe animal mission labels to them, like bony fish or Cydippid Ctenophore or Siphonophore. And these images from our dive streams are currently in all the mini games, ready for classification or localization. And another person is asking, what do I look at to tell where the deep scattering layer is? Well, your best first pass for whatever region you're looking for is a clock, because this happens really in tune with sunrise and sunset. And so that is kind of your biggest indication that it has started to scatter or head back to the deep. But in terms of the dive stream, Alex also does share oftentimes our echo sounder data, which is our sonar, where you can see that scattering layer dispersed or thick at the bottom. And so keep an eye out for that. It's now on the stream. Yep. So these are 3 different panels. Each is a different frequency of echo sounder. But basically what you're looking at, the x-axis along the bottom is time, and then the y-axis going up and down is the depth. And so if you look towards the top, we're getting into night now, and so you can see bright bands across all 3 frequencies of sonar between the surface and 250 meters, that first kind of inch or so. And so that's what it kind of looks like as things are migrating towards the surface. And then during the day, when it's bright out, you'll see it as a thick band right around 500 meters, though it can vary based on where you are. When sonar was first starting to be used, particularly with different navies, this was thought to be the seafloor incorrectly. So it was so thick based on how many different little lanternfish and shrimp and jellies, like all the things we're seeing, based on how thick that layer is during the day, that was actually thought to be a false seafloor, a false bottom. And it was only later that they realized that this was animal life. So pretty cool.
Come check.
Got you. 1, 2.
I got you.
So is localization bound usually done before identification? Yes, it is. This is actually one of the first times that we are putting data into both zones at once.
Quick question, Zulu 1.
I'm just going to pause real quick for engineering. We're doing some checks with the pilots currently. But yes, so bound is usually done first, and then those boxes that are created by the community consensus go into our identification, our classification mini games. But this time, Yoast has done something clever where those images are graduating as soon as they are ready, as opposed to when that whole dive is ready and bound. So we're getting it moved over much quicker by image as opposed to by dive. So and because these midwater animals, these midwater images are not quite as dense as a lot of our benthic scenes, as you may have noticed, they tend to reach consensus a lot quicker.
Yeah, the long-term players might know that it's oftentimes 3 or 4 week process for something to be done with localizations, because it makes sense, right? To first say, oh, where are the things in the scene before you start naming them, if you don't do it all at the same time. So yeah, that's been a nice change here to hopefully accelerate that process a little bit. But if you do see any issues, call it out on the Discord. This is a little bit of experimental because we really wanted to make sure this makes it through quickly, and those folks watching the live stream can see those cool images of the galaxy Siphonophores the next day. And especially those more iconic images seem to be making it through bound or the localization minigame extra quickly, so you should be able to find those as well, and it more rapidly allows us to get more diverse imagery and more different animals, because many of you have seen the plots too of what types of animals are available in the various mini games, and oftentimes we're a little sea fan heavy, sometimes bony fish heavy. Emily keeps a close eye on which animals need the most help in classification, and hopefully this introduces a little bit more diversity, and especially should be good on the Ctenophore jelly front from these past couple dives. But stay tuned for the barracuda that swam by at the end of, what was it, the day before yesterday? It's in there too. Oh yes, oh yes.
And a fabulous glass squid.
And a
Minor
fabulous
detail.
glass squid.
But we are at 215 meters, and I just want to mention that, pretty quickly we're going to get near the surface. Pilots are going to need to, start talking with each other, making plans to pull the vehicle, so we might not have much time to keep chatting, but, feel free to continue watching and we'll try to answer any questions we can before we have to go silent.
Kakani, Lily's asking if you're making DPIV scans faster in the new way.
We're definitely trying to process the DPIV data faster. This has been part of the work that we've been doing as part of the Sasakawa Foundation funding we received, the Ocean Shot. Again, a big reason why we're also out here. And so we are trying out some new software and processes that will allow us to get from scans to 3D models as quickly as possible.
Copy. Ready for command.
And perhaps this question was referring to some of the background conversations happening earlier. We're always dreaming about ways to do better and do new things. But it's a lot of work implementing that in housings that go to 4,000 meters and making them robust enough that they actually work. So lots of ideas, but not all of them make it to fruition.
Our schedules are always changing, so we try not to put out a specific time, but we will be back very soon, as soon as we can.
That was a tease.
Tentatively 2 hours, but we're never know.
Very soon. Might be sooner than you think if things go well. I love that. You know, soon.
We are a little forward, just kind of let roll forward to be straight up with you guys.
Copy.
Thanks so much for timing in. I would eat dinner. I'm gonna eat dinner, Jen, so you should eat dinner.
Oh, they're seeing Alex walk
Kakani
in over
actually
here.
set me aside a
Should have
plate
warned
up
people
in
we've got the
a
video.
mess, so I can't wait to see what surprise she prepared for me.
Delish.
I hope you like gnocchi.
I do like gnocchi. Hooray.
They're saying hi, Alex.
Something fun about our kind of round-the-clock ops is
by Emily.
Oh, what?
Emily's
What's
got
up?
my laptop up.
Oh,
You're being
yeah.
recognized
Say
from
hello.
afar.
Hello.
Yeah. The intrepid Alex Angle.
Oh, there's a mic. I could have said hi. Sorry.
Yeah.
I don't know why I was doing that in silence. Hi, everyone. Good to see you.
I think he's got some amazing drone shots.
Yes,
from the
can't wait
vehicle
to see
launch and
Alex's
recovery.
drone shots.
You can put pause and run when we're at 15 meters. Trigger off 2 biscuits.
Copy.
Do you want high
What's,
power?
yeah.
High power.
Hi, Bar.
Do you want 85%? 100%?
Master
Let's
trace.
do 85. We save 100 for the last one.
Okay.
100.
Last
What's
will
that?
be 100. I'll be 85.
Yeah.
So once we
I
are
wonder
ready
if
to
there's a
go,
scaling factor. Maybe we can actually go to 150.
Yeah, if we need a drift, we can ask them.
Not a drift, but just a lead to
Oh.
port. Obrigado. Yeah, if you need a lee from the wind or the waves, let me know as soon as possible so I can change before the vehicle's at the surface. If you want a drift, just let me know anytime you need it. Copy. This is about 30 meters. After we remove the last set of buoys.
Another angle hour for sure. They're saying we need another angle hour for sure.
What's an angle hour? Do you get in the seat?
Start changing now.
Once
Ocean
I have
recovery.
Nice. That's exciting. All right, so as you're seeing, we're doing a lot of different things with the folks on board. Our engineers are getting ready to get the ROV to the surface. So I think we might be signing off for now.
Boys to surface.
Floats on surface.
I can use the water automatic vessel very close.
Yeah.
Thank you. That way you'll be able to get a belly and see it there.
Thank you.
Position's good from in here. Oh, yep. Start backing up.
See what I'm typing? I'm like, what am I doing?
Who? Where?
Oh yeah. So we're gonna start our recovery then. Floats are on surface. They're gonna be moving those back to load them off, and then we're out of the water. I'll keep the cameras running until then, but yeah, the mics are gonna come down while we're having recovery chatter. So we'll see y'all soon.